How are you all doing? Welcome to PW English. Hello everybody. So the last leg of revision for your NEET 2025 examination for biology, in which of course we are starting with botany today. This marathon is going to be divided into two parts. The first will be the morning session and then the afternoon session. Hello everybody. Hello. Yes. Hello Likhita, Kamalamma, Krupa Venkat, Yashaswini, Unknown, Akhila, Sakshi. Hello. Upin, Akshata, Uma, Suvarshi. Hello Ashu. Sparkle Spectacle, Ramalinga, Kusumarti, Kirti, Farhana, Inchada, Janani, Basaya, Gopika, hello. Good morning. Good morning, Maitri. Hello. Oh, that's sweet of you. Anusha, Preetish, yes. Nayana, Dr. Afsha. Malaya, yes. Manisha, Adish, Dr. Pratham, Usha, Daksh. Good morning, everybody. Suvarshi. Yes, total, the entire syllabus in one shot. Nikita, hello, will get covered. Dev, hello, hello, hello. Shall we begin? Nafisa, hello. So before we, Nafisa Anjum, I don't know which one of your name it is. Hanshita, Shweta, Santosh. All right. So in one shot, we'll be covering the entire syllabus, and yeah, it is going to be divided into two parts. The first morning session and the afternoon session. Rahul, hello. Sneha, good morning. So what we will do? Of course, we will revise. We will do as many questions as possible. Duration will be, 9 hours. Probably, because the entire botany has to go. Kusuma, ecosystem is difficult. I don't think so. Ecosystem is the easiest. Don't break your head around ecology unit. Yes. Hello. Palak, Shivam, MR Sir, Physics. Hi. Okay. So like I said, this entire session is going to be divided into two, the morning and the afternoon session. We are going to be doing all high weightage units first, okay, and then we will be doing, of course, we will go in order. First of all, in class 11th, what are the high weightage units? Now that is important. High weightage units in class 11th for botany, if I were to say, is plant physiology, and then you can expect considerable number of questions from cell and then cell cycle. On an average, if you look at the pattern of NEET, every chapter at least one question they try to cover. One question at least, even from a chapter like Living World, at least one question they do ask. So you can expect at least one question. However, plant physiology unit if I take, that is photosynthesis, respiration, and then you have of course plant growth and development. You can expect somewhere about, on an average, 3 questions from each chapter, 3 questions from each chapter. Same goes for cell and cell cycle. Cell you can expect 2 questions, cell cycle around 2 to 3 questions. Yes. So these are the important units that we are going to be dealing with. So what we will do, we will start with, we will complete first 11th and then we will go to 12th. So we will start with plant physiology first, then go to cell cycle. Then we will come to plant kingdom, biological classification, living world, and so on. The questions are somewhat jumbled and mixed. Purposely we have kept it that way because that is how your NEET question paper is going to be. Okay. Yes. So let's start. Before I begin, my name is Chaitra and I am, of course, a biology teacher specializing in botany. So we are going to start with the revision. I hope all of you all are ready. Your PYQs are something which you have already dealt with for a very long amount of time. I would say here and there, everywhere you have dealt with your PYQs. Even in the series that was conducted on PW English, we have dealt with PYQs only. So what shall we do? We will pick up questions along the PYQs, the same format, the same pattern, and we will start with our first unit, that is plant physiology. Shall we begin? I hope all of you are ready to sit for at least 8 to 9 hours. Yeah, most expected. Isha, I'm really sorry, I will not be using Hindi because this channel is purely English. Okay, sorry about that. Yes, we will be giving a poll to, yes, correct. We will be giving a poll to try to solve the questions within the given frame of time. I hope you know how to participate in poll. Duration would be 8 to 9 hours. Try to participate in the poll, of course. How will you do that? The answer will be A, B, C, D. You will not be putting A, B, B, C, D or D, ma'am, such things. If you put, your name will not get captured here. Just put A, B, C, D in whatever capitals or small letters, your name will get captured. Okay, fine. So, shall we begin with the first question of plant physiology? And the question is in front of you. Good morning. For those of you who joined just now, starting the poll, look at the question. I'm starting the poll, look at the question. Yes. Very good. Don't put A, B, B, C, D, don't put the alphabets twice or more than that. Very good. See, Kopika has put B, 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 your name will not get captured in the poll if you do that. Very good. Easy. Wavelength of photosynthetically active radiation varies from somewhere if you want any conceptual explanation, please let me know. But otherwise, we will try to. Only botany will be covered in today's session. Okay. Zoology will be dealt separately on a separate day. So, what was I saying? If you want any conceptual description, let me know. If there are some short tricks and tips and all which I can help you with, and I will do that. Okay. So, wavelength of photosynthetically active radiation, you have to remember that this is in relation with VIBGYOR, the visible spectrum that we have. So, VIBGYOR is the visible spectrum and this is a spectrum in which blue and red wavelength mainly get utilized for photosynthesis. So, blue falls somewhere between 400 to 500 and red falls between 600 to 700. But in total, if I were to generalize, I could say that 400 to 700 nanometer wavelength is the wavelength where photosynthesis occurs. Good job. And in this particular wavelength, 400 to 700, you have the visible spectrum, the 7 colors of rainbow. Okay, 7 colors of rainbow. Yeah, I will not be explaining anything in detail. Don't worry. We don't have time for that. Photosystem which is concerned with non-cyclic photophosphorylation is the one that shows non-cyclic photophosphorylation, the Z scheme. Okay. Okay. Shiva, I will be there in Kannada channel soon. Don't worry. Yeah, I'll be dealing in class 11th. In the afternoon session, class 12th will be dealt with. Very good. Good job. Even if, yeah, I said if you want some conceptual explanation, you could let me know. Very quickly, we will do a revision. Or else you can make the note of the concept that you're weak in and then you can deal with that later. Do a quick revision by yourself. Photosystem which is concerned with non-cyclic photophosphorylation, which is also called as the Z scheme. Okay, what is it? Both photosystem A and photosystem B. I mean the sense option that they have given, photosystem 1 and photosystem 2, both. Good job. They both are involved in Z scheme and that is why you call it as anyways non-cyclic photophosphorylation. What are the things that get produced in non-cyclic photophosphorylation? You have production of NADPH, you have production of ATP. There's also liberation of oxygen that is happening. So these are the three things that get produced in non-cyclic photophosphorylation. But when it comes to cyclic, we say that only ATP gets produced. ATP gets produced. Okay. Right. Let us talk about this question. Light reaction produces assimilatory power in the form of. Light reaction produces assimilatory power in the form of chemiosmosis. Okay, we'll come to that. The moment a question comes on chemiosmosis, we will start with that. Explanation I'll give you, a basic explanation I'll give you, which might help you remember chemiosmosis and understand chemiosmosis. So, light reaction produces assimilatory power in the form of what? ADP NADH2, ATP NADPH plus H plus, NAD plus, NADP plus. What is the answer? The light reaction is nothing but cyclic and non-cyclic photophosphorylation. So, what is the assimilatory power that it produces? In the sense, the molecules that can be utilized for something bigger like dark reaction, like production of glucose. So, for that, what exactly is used? ATP, NADPH plus H plus is produced using the light. Okay. For those of you who got confused, NADH. Okay, here's something you have to remember. Okay, something that you have to remember. NADPH. Correct? P for photosynthesis. P for photosynthesis. You will see that NADPH production or even the mention of NADPH happens only in photosynthesis. The moment that NADPH's P, that is phosphate actually, is lost, then comes NADH, and this is seen in respiration. Please remember, this is seen in respiration. I am talking in terms of your syllabus. So remember that way, don't get confused. Those of you who got confused, a small tip for you. NADPH, when you say, P stands for phosphate, but you can also remember P stands for photosynthesis. Okay. So light reaction produces assimilatory power when they say, then NADPH gets produced there. Alright? Yes, I gave you a trick for this. When they give NADH, phosphate is not produced, not a part of this, then you will see that it is seen in generally respiration. It is seen in respiration. Clear? I hope this is clear. I hope this is clear. Okay, let's go to the next question. Photolysis of water is related to. Photolysis of water is related to what? Photolysis of water is related to what? Yes. Photolysis of water is related to what? Good. Try to answer. Photolysis of water is related to what? Photolysis of water, H2O splitting into 2 protons, 2 electrons plus half a molecule of oxygen. Correct? This is what is photolysis of water. Which particular part is involved with it? Yes, photosystem 2 is involved with it. Photosystem 2 is continuously losing its electron during non-cyclic photophosphorylation, and it is worrying that I am losing my electron, the whole day light is falling on me, and I am losing my electron for the sake of production of ATP and NADPH. Fine, for the greater good and for the survival of the plant, but who will look after me, it worries. Who will look after me? Because it is losing its electron every time an electron gets excited, because the photosystem 2's chlorophyll A is exposed to sunlight. Now that's the problem. So that is when water molecule comes into picture and says, don't worry, we will not allow you to die, because if you die, photosynthesis will stop. So please take my electron. I am a water molecule. I am available anytime, any day. Nobody cares about me. That means I am available in abundant is what it means. So you can take my electron. Yes, splitting of water. Correct? Now, Cytochrome is involved in electron transport. So you can call it as an electron carrier. And NADP plus is something which will become NADPH in non-cyclic photophosphorylation. Correct? Yes. I will show the leaderboard, but after a certain number of questions, because otherwise it consumes a lot of time. Correct? Yes. Let us start with this question. To reduce 1 carbon dioxide in C3 cycle, the assimilatory power needed is to reduce 1 CO2 in C3 cycle, the assimilatory power needed is, assimilatory power needed is how much or how many? Yes, come on. See, for those of you who are asking me for chemiosmosis, if you want, we can do a small short on that and I will upload it probably today or tomorrow on the same channel, PW English. You can utilize that. Yes, we will come to that. In photosynthesis, you don't have many diagram based questions. Most of the diagram based questions come from other chapters, like it could be plant kingdom or sexual reproduction in flowering plants, such chapters. Okay, yes. Done? To reduce 1 CO2, they are asking you. Please recall the cycle. You have 3 steps: carboxylation, reduction, and then regeneration. Okay, clear? PYQs are already done, my dear students. There was a series run in the same channel in which all the PYQs were covered. So, in regeneration, 2 ATPs, but the questions are same in line with the PYQs. Language is changed. That's it. 2 ATPs are utilized, and then what is utilized here? In one round of cycle, 2 NADPH in the regeneration phase. Then, in the reduction phase, not in regeneration, in the reduction phase. Then in regeneration phase, you have 1 ATP being utilized. Correct? So, in total, you have 3 ATPs and then 2 NADPH utilized. Correct? Yes or no? Yes. Then the answer is 3 ATP and 2 NADPH2. I think most of you have answered this. Very well. Good job. Be proud of the answers that you're getting right, but don't get demotivated by the answers that you're getting wrong. I am going to modify this question a little bit and ask you something else. Okay, you are going to answer this question very quickly now. Very quickly you will answer this question. If I change this question to one glucose molecule, if I ask you for one glucose, how many ATPs and NADPH2 would be needed? What your answer will be? Tell me. For one glucose, if they ask, then what your answer would be? Yes. Tell me very quickly. Very quickly. Think and answer. Think and answer. Yes. Yes. So, when you take one glucose, see here, what is the formula? C6H12O6. That means for 6 C6 to come in, that means 6 carbon dioxides have to be fixed. That means the cycle will run how many times? 6 times. So, if one cycle would consume 3 ATP, 6 into 3 would be 18 ATP and 6 into 2 would be 12 NADPH2. Understood? Is this clear? Fine. That is one glucose. This is one carbon dioxide. One small homework for you. Try to solve this. If they ask one sucrose, what will your answer be? If they ask one sucrose, then what your answer would be? Then what would you answer if they ask one sucrose? Okay. In the same line, you have to take this. No, not same. Not same. Sucrose is a disaccharide. I gave you a hint. It's a disaccharide made up of fructose and glucose. Glucose has 6 carbon, fructose has 6 carbon. 6 and 6, 12 carbon dioxides are needed. 12 carbon dioxides are needed. Yes. Now do 12 into 3 and 12 into 2. Very good. Yes, exactly. Correct. All right. Let us come to this particular question. Carbon dioxide acceptor and carboxylating enzyme in C3 plants are respectively. Try to answer this question. Yes. Try to answer this question. The idea here is to do as many concepts as possible so that your revision is good and thorough, and then use certain concept explanation if you want. You can ask me, I repeat. Carbon dioxide acceptor and carboxylating enzyme in C3 plants are respectively. Who's accepting it and who is carboxylating it? So the acceptor is RUBP and the enzyme is Rubisco. How many carbon does RUBP have? How many carbon does it have? It has 5 carbon molecules. Yes, it has 5 carbon dioxide molecules. Okay, the enzyme is Rubisco. 3-PGA is the product that gets formed. It's a product and it has 3 carbons present in it. PEP is an acceptor, yes, but where? In the C4 pathway. How many carbon molecules does it have? 3 carbon, okay. Oxaloacetate, it has 4 carbon and it is again the product that is formed in C4. Alright, so the answer is option B. Option B. Okay, few of you have got this wrong. Some of you have marked option C as the answer. Wrong. Okay. Alright, I'll try to go faster. Okay. Cran's anatomy. Cran's anatomy is, what is Cran's anatomy? What do you mean by Cran's anatomy? What do you mean by Cran's anatomy? Having peripheral reticulum in chloroplast, presence of distinct bundle sheath, presence of same type of chloroplast, large vacuole present in mesophyll cell. Large vacuole present in mesophyll cell. What is the answer? The answer would be presence of distinct bundle sheath. Where? Around the vascular bundle. Around the vascular bundle, of course. Please remember the distinction between the two. Please remember the distinction between the two. Please, when they say Cran's anatomy, Cran's refers to ring-like arrangement of the bundle sheath around the vascular bundle. Correct? There are 2 different types of chloroplasts that you find in the bundle sheath and in the mesophyll. Please remember, the mesophyll has something called as a pepcase enzyme, phosphoenol pyruvate carboxylase, and the bundle sheath has an enzyme called as Rubisco, but it does not have pepcase. No pepcase. Can you give me example for Cran's anatomy? What are the plants? Sugarcane, maize and sorghum. SMS. Sugarcane, maize and sorghum are the example. Yes. Yes. Correct. Clear? Alright. Let us come to this particular question. In C4 plants, the first product of carbon dioxide fixation is. In C4 plants, the first The first product of CO2 fixation is what? What is the first product of CO2 fixation? In C4 plants, the first product of carbon dioxide fixation would be what? Oxaloacetate. And then oxaloacetate will convert into malic acid after that's the next step. Yes, this would be the first product of C3 cycle. This would be the product of C3 cycle. Okay. Please remember the number of carbons, 4 carbon, 3 carbon, 4 carbon. Here again, glutamic acid you need not remember, forget it. Okay, glutamic acid is a type of amino acid, The most common limiting factor for photosynthesis is, what? The most common limiting factor for photosynthesis is what? CO2, O2, H2O, temperature, what? A direct question from your NCERT. Try to answer this. Very good. The most limiting factor is CO2. First of all, it is present in the lowest of the concentration in the atmosphere. Yes. Why? Why the confusion? Why C? Why B? Why D? Simply for the sake of it, carbon dioxide is the most limiting factor. The rate of photosynthesis gets altered depending upon the carbon dioxide availability. Oxygen is not directly involved in any type of photosynthetic activity. Correct? Oxygen is a byproduct, something which is not needed by the plant and released during photosynthesis. Yes. Incorrect statement in relation to 𝐄𝐍𝐆𝐋𝐈𝐒𝐇 hypothesis. I'll give you 45 seconds because the statements are, yes, Blackman's law of limiting factor. This is easy. 𝐄𝐍𝐆𝐋𝐈𝐒𝐇 hypothesis. Primary electron acceptor is located towards the outer side of membrane. NADP reductase is located on lumen side of thylakoid membrane. Splitting of water releases protons in the lumen of thylakoid membrane. Decrease in pH of thylakoid lumen due to proton accumulation. For this, I will have to give you some kind of a theory explanation. So, kindly bear, have some patience, like 10 minutes. Yes. Incorrect, they are asking you. Incorrect statement. Incorrect statement is this, yes. NADP reductase is located on lumen side of thylakoid membrane is wrong. It is located towards the stromal side. See, stromal side. Please understand, you have a thylakoid membrane. I am drawing a single thylakoid. I am not drawing a grana as given in your book. So, this is the thylakoid membrane. Imagine, okay. What are they asking you? The first statement says that the primary electron acceptor is located towards the outer side of the membrane. What does outer side of the membrane mean? Okay, what does it mean? What is primary electron acceptor? First of all, it's a molecule called as pheophytin, which is the first electron acceptor in non-cyclic photophosphorylation. Non-cyclic photophosphorylation. It is located towards the outer side of the membrane. Pheophytin. So, imagine this is the thylakoid, this is the lumen of thylakoid, okay. And this thylakoid is located where? In a larger chloroplast. I'll just draw in a larger chloroplast, something like this. So, this is what the outer part will be what? The fluid filled stroma, correct? This is going to be the stromal region. It is going to be the stromal region. Yes or no? Yes, I am just erasing this because I just wanted to give you a visualization. We don't need it. So, this is the stromal side of it. They are saying the primary electron acceptor is present towards the outer side, means attached to the thylakoid membrane, but here, which is correct. It is facing the stroma, so towards the outer side. Okay? Clear? No, first pheophytin comes. Okay. In non-cyclic it goes like this. Photosystem 2, in the theory part they have given, electron gets excited, gets expelled. Who accepts it? Pheophytin. Alright. And then comes plastoquinone, that is the hydrogen carrier. Then comes cytochrome B6 bar F complex. Then comes plastocyanin. Then comes photosystem 1. All the reaction happens through electron carrier. Ferredoxin will come and then the enzyme NADP reductase, which will help in production of what? NADPH. From NADP, and the electron that came from here, that electron will be taken up and 2 protons will be picked up from the stroma. Meanwhile, photosystem 2 is crying, where's my electron? Give me my electron back. And that is when the water splitting happens so that the electron can fill the electron gap created in photosystem 2. And these protons are again liberated into the lumen. Correct? Liberated into the lumen. Meanwhile, cytochrome B6 bar F complex is also pumping H plus with the help of plastoquinone. Yes or no? Yes. So, this molecule is pheophytin, which is present towards the outer side of the thylakoid membrane. Towards the outer side of the thylakoid membrane. Clear? Where is NADP reductase present? Also present towards the outer side of the thylakoid membrane. Attached to what? Is it stuck to thylakoid membrane? Yes. Where is it facing? So imagine I am NADP reductase. Behind me is the lumen. In front of me is the stroma. Understood? So I am continuously looking at stroma, though I am attached to the thylakoid membrane. That is what it means. Okay? 8 to 9 hours class duration. Understood? Splitting of water releases protons in the lumen of thylakoid membrane. Yes, I just now showed it to you. It goes into the lumen of thylakoid membrane. Decrease in pH of thylakoid lumen due to proton accumulation. Yes, inside the lumen H plus increases. So H plus increases means this is getting acidic, and in the stroma there is drop in H plus. How is it happening? With the help of non-cyclic and cyclic photophosphorylation. All the events happening in cyclic and non-cyclic are pumping H plus into the lumen of the thylakoid, and the thylakoid is getting more and more acidic. More and more acidic, and the thylakoid is literally screaming, the insides of me are becoming acidic, somebody help me, otherwise I will get destroyed. And it is continuously crying for help. Who comes to help? Who will come to help? A molecule called as FOF1. FOF1 molecule. It says that here I am, I will help you. And this FOF1 particle will now send all the H plus back to their home. From where have they come? They have come from stroma, and FO particle of the FOF1 says that go back to your home so that the acidity of the thylakoid can be reduced. But the H plus are actually happy because they are going back home. But they say because you are sending us home, we will help you create ATP. So the head part of it ends up synthesizing ATP. All this is possible because of a proton gradient that is created, because H plus is increasing in the lumen and then the H plus is dropping in the stroma. Clear? Understood? That is chemiosmosis. Yes, come to this particular question. Rubisco has a much greater affinity for dash when the CO2O2 is nearly equal. Done? Okay, easy. This one's easy. Rubisco will have a much greater affinity for dash when CO2 and O2 is nearly equal. Greater affinity for what when both of these are equal? Same for both, depends on temperature. What is the answer? Answer is CO2. Answer is CO2. Good job. The problem is when CO2 is low and O2 is high, then Rubisco is a confused enzyme. It wants to bind to CO2 also, it wants to bind to O2 also. But the plant wants it to bind to CO2. So it makes sure that CO2 levels are high. But there comes a point sometimes when CO2 drops and O2 will increase. That time Rubisco gets a chance to bind to O2 and then cause trouble in the form of photorespiration. Correct? Yes. Let us come to this. Let us talk about photorespiration. In the photorespiratory pathway, there is, read it, read it, read it. Read it. Done? Photorespiratory pathway is called as a wasteful process. It is called as a wasteful process. Correct? Neither synthesis of sugar nor of fat, nor of ATP. Why am I saying fat? Synthesis of ATP but not of sugar. Synthesis of sugar but not of ATP. Synthesis of both ATP and sugar. The answer is neither sugar is synthesized nor ATP is synthesized. Good job. Very good. Along with this, you can add one more point. Yes, also called a C2 pathway. Yes. You can add another point that CO2 is released instead of being fixed. Normally, we say that CO2 is getting fixed. Correct? But here, CO2 is getting released instead of it being fixed. That's why we call it as a wasteful process. And you have to remember the first equation of photorespiration, that is RUBP attaching with oxygen in the presence of Rubisco, giving us a 2-carbon what? Giving us a 2-carbon phosphoglycolate. Please remember this molecule's name, and giving us a 3-carbon PGA, phosphoglyceric acid. Yes. Clear? All right. Let us come to this particular question. The bundle sheath cells are rich in dash while lack dash. Done? Done. The bundle sheath cells are rich in dash while lack dash. Bundle sheath cells are rich in Rubisco and they lack pepcase. Option A. What happened? Why? Flipped. How? Where are the bundle sheath cells present? The bundle sheath cells are present around the vascular bundle. Aren't they rich in Rubisco? Are they not rich in Rubisco? Yes or no? Why are you confused? This diagram will help you. That's why when I give explanation, you guys tell me, ma'am, go fast. So then I have to rush. Then I realize maybe you need a little bit of conceptual understanding. I mean revision more than understanding. This is a mesophyll cell. Very roughly I am drawing. Okay. And this is the bundle sheath cell. Now this bundle sheath cell will have Rubisco. This has Rubisco and this here has pepcase. Both are fixing carbon dioxide. Both are fixing carbon dioxide. Remember that. Okay? Clear? Both are fixing carbon dioxide. Remember that. But the enzymes involved are two very different enzymes. Two very different enzymes. Now is it clear? Now is it clear? Yes. Alright. Rubisco in BS cells, bundle sheath cell. Pepcase in mesophyll cells. Meso cells I am writing. Alright. Let's go to the next question. Read it. I'll give you 45 seconds. Incorrect statements they are asking. Incorrect statements they are asking. Even in NEET, try to finish your biology paper in 55 minutes. 50 minutes maximum or 55. And I am saying you to finish all the easy ones. There could be few difficult ones or confusing ones which you are scared to attempt. Leave that. Excluding that, you should be able to cover the entire paper in 50 to 55 minutes. Okay? Done? C4 plants lack Calvin cycle. Remember one thing, Calvin cycle is more like a universal pathway of photosynthesis. Okay, C3 cycle has to occur. In addition to C3, C4 can be there. But without C3, sugar production will not occur. C4 plants lack photorespiration. C4 plants have more productivity than C3 plants. C4 plants cannot tolerate higher temperature. Wrong, they can. So, how many statements are incorrect they are asking? 1 and 2. So, 2 statements are, or rather, this is also wrong in a way. But yeah, we will go with 2 statements as per the answer key. I'll tell you why. One negligible amount of photorespiration does occur in C4 plants, so low that the plants don't care. They don't care that photorespiration is occurring. So, it is very, very, very low. Okay. Completely zero, no. That's why we say that C4 plants lack photorespiration. Okay. Let's come to this question. Enzyme responsible for primary CO2 fixation. Primary CO2 fixation. Done? Alright. Primary they are asking you. Primary CO2 fixation. So, the first level of CO2 fixation occurs in mesophyll and the enzyme is pepcase. And the enzyme is pepcase. Very good. Very good. Secondary will occur in bundle sheath cell with the help of Rubisco, carboxylation. Secondary carboxylation. Okay? Okay? Next. In Hatch-Slack pathway, the primary carbon dioxide acceptor is in Hatch-Slack. In Hatch-Slack pathway, the primary carbon dioxide acceptor is done? Alright. Hatch-Slack pathway is another name is C4 pathway. Who's the primary acceptor there? Phosphoenol pyruvate. PEP. Phosphoenol pyruvate. Why the confusion? They're asking acceptor. Oxaloacetate is the product. It's the product of Hatch and Slack pathway. It's the product. Okay? Yes, Atif, it's done. Okay. Let us come to this. If a chemical process is affected by more than one factor, then its rate will be determined by the factor. Done? Yes. If a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value, farthest, nearest to its maximal value, both B and C, which is nearest to its minimal value. Whichever is available at the lowest quantity, that is the one which is going to affect the rate of reaction, correct? Suppose photosynthesis is occurring and CO2 is at the lowest. Everything else is properly available, but CO2 is available at the lowest. Then if you want to increase the rate of photosynthesis, you have to increase CO2 availability. CO2 availability, okay? Next question. Light saturation occurs at dash of the full sunlight. At what percentage of the full sunlight light saturation occurs? Done? This is done? Yes. So it is 10%, correct? It is 10% because that is how even on the cloudy days the plants are able to photosynthesize. This is from factors affecting photosynthesis. Easy, very easy. Law of limiting factor, which scientist gave law of limiting factor? Option B, Blackman. Okay. Hooke, Robert Hooke you would have heard of, who observed dead cells. Fleming you would have heard of, Mendel you would have heard of. So Blackman's law of limiting factor, quite famous. Okay. At what concentration do C4 plants show saturation? At what concentration do C4 plants show saturation? They are asking you. Yes. C4 plants show saturation at what concentration? Yes, 360 and 450 would be C3 plants. C3 plants. 360 microliter per liter of carbon dioxide. This is for PEP case. For C3 it is 450 microliter per liter for the enzyme Rubisco. That much amount of CO2 they need. So option D would be the answer here. Option D would be the answer here. Yes. Next question. Okay, just a minute. So photosynthesis, we are, I think, almost done. Almost done with photosynthesis chapter. Do you have any doubts? If you want, we'll display the leaderboard very quickly now. I'll just display the leaderboard very quickly. These are the top 10 names currently. Okay? Yes. No doubt in photosynthesis chapter? No doubt in photosynthesis chapter? So, what and all you will be covering? Of course, light reaction, the entire chapter is important, but most often the questions are asked from C3 cycle or the dark reaction part of it. We have covered factors affecting photosynthesis also. That also we have covered, but pay attention to the dark reaction part of it, okay? And the difference between C3 and C4 plants, very often asked, very often asked. Yes, let us go to the next chapter then, that is respiration in higher plants. Look at the respiratory substrates, and in the descending order of their respiratory quotient, you have to arrange these substrates, respiratory substrates. Okay. Done? Okay. Descending order they are asking you. So first we'll write it down. 1.9, 0.7, and oxalic acid is more than 1, greater than 1. So how would you arrange it? You would go B, A, C and D. Option A. Respiratory quotient is what? Respiratory quotient is what? Respiratory, even if you didn't know this, A, C, D you should have known. Automatically the answer would be option A. Automatically the answer would be option A. Yes or no? There is another one which they have not given, but then they might ask you. For anaerobic respiration, if they ask respiratory quotient, for anaerobic respiration, if they ask respiratory quotient, then how will you answer it? Volume of CO2 released divided by O2 that was consumed. Correct? So, in anaerobic respiration, CO2 is released. That is for sure. But is oxygen consumed? No. Then that is 0. Anything divided by 0 is what? Yes, correct. Infinite or infinity. Understood? So, this is another question that they could ask. We don't know. But yeah. Let us come to this question. Enzyme of Krebs cycle which is found attached to the inner mitochondrial membrane. It's an enzyme of Krebs cycle found attached to the inner mitochondrial membrane. Inner mitochondrial membrane. What is it? What is it? Done? Done? Yes. Malic dehydrogenase, succinate dehydrogenase, citrate synthase, isocitrate dehydrogenase. The answer is succinic acid dehydrogenase or simply succinate dehydrogenase. Very good. Which is also a part of complex 2 of ETC. Correct? Or ETS. Remember, citrate synthase is involved in citric acid formation in Krebs cycle. Citric acid formation in Krebs cycle. Whereas succinate dehydrogenase involved in the formation of FADH2. So, when here succinate converts to fumarate, you actually see involvement of this enzyme, because this is where FADH2 gets released. FADH2 gets released. Yes, good. Very good. Substrate level phosphorylation does not occur in which of the following of aerobic reaction? In which of the following aerobic reaction substrate level phosphorylation is not occurring? Okay, let's start. Try to solve this. Substrate level phosphorylation does not occur in which of the following aerobic reaction? When 1,3-bisphosphoglyceric acid converts to 3-PGA, you will see 1 ATP formation occurring. If there is a single reaction, that is called as substrate level phosphorylation. When one substrate converts into another, you see release of ATP. Yes or no? When 3-PGA converts to 2-PGA, you are seeing simply a rearrangement. The phosphate that was present in the third carbon jumps to the second carbon. That's it. There is nothing being released there. No molecule is being released. Just that the third carbon's phosphate goes to the second carbon. That's it. When PP converts to pyruvate, you again see ATP formation. Succinyl coenzyme A converting into succinic acid. Here also you will see formation of GTP. Krebs cycle, GTP. Then the answer is option B. Substrate level phosphorylation does not occur in which of the following aerobic reaction? They are asking you. Why? Answer is B. In B, I repeat, there is simple rearrangement. There is simple rearrangement that is going on in option B, nothing else. Okay? Clear? This is clear, you have no doubt. 1, 2, 3. ABC are part of glycolysis. Succinyl coenzyme A to succinic acid is part of Krebs cycle. Okay? Let us come to this particular question. What are they asking you? Identify the correct site of glycolysis and Krebs cycle. This is very simple. This is very simple. Sites are very important. Okay? Very important. Good morning. Hello. Yes. Done? Done. Site of glycolysis and Krebs cycle. Very easy. Very easy. Glycolysis occurs in cytoplasm and Krebs cycle will occur in mitochondrial matrix. Correct? Mitochondrial matrix. Yes. Easy? Easy. Concentration of alcohol in a sugar solution inoculated with yeast, after which the yeast cells are killed. The yeast cells will die after a certain concentration of alcohol. What concentration is this? What concentration is this? Easy. Yes. Easy? Yes, good. Option B. 13% is the answer. Very good. 13% is the answer. One more thing I would want to tell you. When it is anaerobic respiration, this line is also there in NCERT. I don't know how many of you have paid attention to it. When it is anaerobic respiration, the energy yield that you get, right? Like how much of energy is produced? Only 2 ATPs are produced. That too, this is from the event of glycolysis. This you remember, correct? So what is this energy yield? It is literally less than 7%. 7% or less than 7%. This number also they have mentioned. So you please go through that particular statement, okay? Let's come to this one. Match the following. They have given you complex 1, 2, 3, 4, 5 and then A, B, C, D. From where do you get this information? From the diagram that is given in ETS. They have mentioned, and of course from the theory part also. This is mentioned. So please answer. You want, I'll tell you one simple trick. In NEET, the moment you get the question paper, open it and finish match the following first. Match the followings are most often 99.9% easy. You will be able to solve them. It will boost your confidence. Mentally, you will be a little relaxed and then go for the next set of questions. That would be my advice. The next time you take up a mock test, you can try it because you have good number of match the followings in NEET. Then complex 1, also called as NADH dehydrogenase, has FMN and FES, iron sulfur protein. Complex 2, also called as succinate dehydrogenase, has FAD, flavin adenine dinucleotide. Full forms nobody will ask, so don't worry about that. Complex 3 is made up of cytochrome B and cytochrome C1. So E here. Complex 4 has cytochrome A, cytochrome A3 and has 2 copper centers. Please remember, this is the only complex to have 2 copper centers. No complex will show 2 copper center. Quite a unique property. Okay. And last, you have complex 5 FOF1 particle. So DCEBA. DCEBA, option C. Answer is option C. Correct. Confusion. Here also few of you are confused. Option B and A and all you have put. So please go through this particular part. Okay. Akhila, there's sadly no trick to remember here. I would, those of the students who remember it, they have remembered it because it's just drawing. Like complex 1, you can put 2 circles, then write inside it. So that could probably help. But yeah, sadly, I don't have any trick for this. Okay. Which of the following is not an intermediate in Krebs cycle? Not an intermediate in Krebs cycle. Yes. Hello, Anam. Pranay. Hello. Yes. Good. Good. Done? I hope all of you have read Krebs cycle. Not an intermediate. Acetic acid, succinyl coenzyme A is formed, malic acid is formed, citric acid is formed. That is the first product, right? Then the answer is acetic acid. Common name of acetic acid is vinegar. Vinegar, commonly called as vinegar. Yes or no? Yes. Where is it? It's not part of our Krebs cycle at all. Succinyl coenzyme A. Why do you say succinyl coenzyme A is not a part of it? Come on, guys. Alpha ketoglutarate first converts into succinyl coenzyme A, and then succinyl coenzyme A converts into succinate. Where is this? The theory part of your Krebs cycle, this information is mentioned. You have your book in hand. Please read it once very quickly. Please read it once very quickly. Shall I proceed? I hope there is no confusion here. There is no confusion here. Chalo, in any case, you know that succinyl, succinate, something like this is part of Krebs cycle. Na? Acetic acid is nowhere to be seen. Yes or no? If you didn't know this, leave it. Acetic acid is like a new term, nowhere related to Krebs cycle. Okay? Yes. This is one tricky question. Now, don't think that the paper will be difficult. Never. We can actually predict the paper, but then I don't want to, send a rumor across saying that we can predict the paper, we know how the paper will be. We don't know exactly how the paper will be, but the concepts are repeated every year. And in biology, difficult questions are generally 5 or below, like difficult for everybody, I'm saying. Confusing will depend upon student to student. Like some students might find examples very confusing. Some students might find statement-based questions very confusing. So that will differ from student to student. That will decide the paper. That's why biology paper, no matter how it is going to be different for each one of you, because your confusing concepts are very personal to you. Na? Like I might get confused in ETS. There might be somebody who's very good in it. That way. You might be very good in calculation. Like, of course, come on, guys. Basic calculation in biology. Plus, minus, division, multiplication. Only this much is asked. Thank God. Okay, and let us not say this out loud. Let us not say this out loud and let us not, spread this so that then they might include it. Who knows? Yes. What is the answer? Which of the following biomolecule is common to respiration mediated breakdown of fat, carbohydrate and proteins? What's the answer? Why do you say it is C? Fat converts into acetyl coenzyme A. Doesn't it convert into something else also? When fat breaks down, you get two components. One is glycerol. The other one is fatty acid. Now, fatty acid is more than happy to become acetyl coenzyme A. It becomes acetyl coenzyme A. Yes. What about glycerol? What about glycerol? It converts into, I'll write it here, glyceraldehyde 3-phosphate, which is an intermediate in glycolysis. So, doesn't it become pyruvic acid then? So, shouldn't the answer be A, because they have asked fat, they have not asked fatty acid. Yes, indirectly pyruvic acid. Carbohydrates anyways can form pyruvic acid. Proteins can also do that. Not all amino acids, but they are asking proteins. Then the answer turns out to be A. A, my dear students. A. You got the concept? You got the concept? They are asking fat, and fat has two components. Glycerol and it has fatty acid. Fatty acid will convert into acetyl coenzyme A. Please read what they are asking. Which of the following biomolecules is common to respiration mediated breakdown of fat, carbohydrate and proteins? What is the answer? No. How? What? Well, I don't have the reference, but see here, what am I saying is, fat has two components. When it breaks down, you get glycerol. Another component is fatty acid. Fatty acid, yes, will break down and eventually give rise to acetyl coenzyme A. That acetyl coenzyme A then will enter Krebs cycle. Okay. Now, what about glycerol? Glycerol converts into 3-phosphoglyceric acid, or rather, indirectly if I was to say, 3-PGAL, that is 3-phosphoglyceraldehyde it converts into. Correct? Yes or no? Yes. So, when I say 3-PGAL, then it ultimately moves forward and becomes pyruvic acid. Correct? Yes or no? ChatGPT says acetyl coenzyme A. Then very good. ChatGPT might be, yeah. Carbohydrates will of course eventually break down, and then ultimately the glucose or whatever monomers you have will give pyruvate only. Proteins have a lot of amino acids. Few can directly convert into alpha ketoglutarate. Few can also directly convert into pyruvate. Again, I'm going as per the answer key which is given, and I am pretty much convinced with that answer key. Okay. Yeah, but isn't all three also converted to pyruvic acid? You are saying that all three are converted to acetyl coenzyme A before entering the Krebs cycle in aerobic cellular respiration. Fine. Then isn't all converted into pyruvic acid also? A component of fat is getting converted into glyceraldehyde 3-phosphate, ultimately pyruvate. Carbohydrate, ultimately pyruvate. And then fatty acid, protein also ultimately into pyruvate. PGAL, okay, phosphoglyceraldehyde converted into acetyl coenzyme A. Fatty acid of course will not convert into pyruvic acid, it becomes acetyl coenzyme A, correct. Acha, you are saying that this will also ultimately be converted into acetyl coenzyme A. Okay, I got your point. So this is also ultimately converted into it, and this also is converted into this. So the answer will be acetyl coenzyme A. This is what you are saying, correct na? Yes? Yeah, that makes sense. That makes sense. Yeah. I take it back. Option C. I was stuck with the answer key that was given. So yeah, we'll go with option C here. ChatGPT ki jai ho. All right. Yes. Yeah. Got it. I got it. What you are saying. Yeah. Because the answer key was given pyruvic acid. I still entirely don't feel it is wrong, because glycerol does convert into pyruvic acid, but whatever logic you're applying is also right. Not wrong. I would see that logic is better. So yes. Okay. Let's come to this particular question. How many ATP molecules are produced from the complete oxidation of one molecule of acetyl coenzyme A? Quick. Please read it. What does it say? One molecule of acetyl coenzyme A. How many ATP molecules are produced from the complete oxidation of one molecule of acetyl coenzyme A? Okay. So that means you're going to skip what? You're going to skip link reaction. If you had added link reaction, you would have got 4 NADH. So if you take it from acetyl coenzyme A and then oxaloacetate undergoing condensation to give us citric acid, from here if I take this entire Krebs cycle, I'm not writing the entire cycle, let's say. So that means you have to take this cycle alone. So this cycle alone, if I take, it will produce 3 NADH. 1 FADH2, correct, and it produces 1 GTP. 1 NADH is equal to 3 ATP, so 3 into 3, you get 9 ATP here. 1 FADH2 is equal to 2 ATP and 1 GTP is equal to 1 ATP. Correct? So, what is the answer? The answer will be 12 ATP in this case. Yes, 12 ATP in this particular case. Please understand, they are asking you one molecule of acetyl coenzyme A. So, that means only the citric acid cycle you have to consider, not even the link reaction. Okay? Yes, not even the link reaction. Don't consider the link reaction here. 38 ATP or 36 ATP would be starting from glucose up until the end of Krebs cycle. Yes, whatever you are saying is right. Understood this particular question? All good? Shall we go forward? Thumbs up, thumbs down, whatever. Yes, all right. Good. Read this question. How many ATPs are produced? All right. When a succinate is converted to fumarate, during oxidation of one molecule of acetyl coenzyme A in Krebs cycle. They are being very specific here. They are being very specific. They are not asking you the entire cycle and the entire total of what ATP are produced. Succinate is converted to fumarate during oxidation of one molecule of acetyl coenzyme A. How many ATP are produced when a succinate is converted to fumarate? Succinate to fumarate. Yes, succinate to fumarate, you get only, see here, one molecule of FADH2. And if I take one molecule of FADH2, then 2 ATPs are generated. Option B would be the answer. What happened? How 3 ATP? Only succinate to fumarate. Succinate to fumarate. Succinyl coenzyme A to succinate 1 GTP. Succinate to fumarate FADH2. Understood? Clear? They are asking you succinate to fumarate 1 FADH2. That means 2 ATP. Okay? Indirectly asking respiratory quotient. Indirectly asking respiratory quotient. Of groundnut and castor. Think a little. Groundnut and castor, why are they asking you? Why can't they ask something directly? Huh? Think. Yes, correct. Groundnut oil, castor oil. That means their endosperm is rich in castor endosperm. The seeds in general are rich in stored fats. Oil production is happening because of that. And fats have a respiratory quotient of lower than 1. Tripalmitine has 0.7, but in general we say it is lower than 1. Now I hope none of you have confusion with greater than 1 and lower than 1 sign. Okay. This is of carbohydrate. Greater than 1 is of organic acids. Give me example of organic acids. Okay, I will give you example of organic acid. oxaloacetate, alpha-ketoglutarate, pyruvate. These are organic acids. They have greater than 1. Okay. Clear? Yes. Malic acid, yes. When the evolution of CO2 is more than the intake of oxygen, the respired substrate should be. Think. Again, indirectly asking a question. Evolution of CO2 is more, but oxygen consumption is less. Pari, good morning. Yes. What is the answer here? When the evolution of CO2 is more, CO2 released is more. Very good. That is why you are getting it as greater than 1. Greater than 1, why? Because, very good. It is greater than 1 because CO2 releases more. You put a little oxygen, CO2, little oxygen, CO2 releases. Fatty acid, like put oxygen, it will be like, put a lot of oxygen. Correct? Yes or no? Polysaccharides or glucose is not like that. You put oxygen, CO2, oxygen, CO2, same energy. I don't even know if it makes sense, whatever I did, but yeah. Okay. In the electron transport chain during terminal oxidation, the cytochrome which denotes electrons to O2 is, think. I'll show you Chirag, leaderboard, yes, soon after this chapter. Coming soon. Very soon. Done? Okay. Class duration 8 to 9 hours. Electron transport chain during terminal oxidation, the cytochrome which denotes, the moment they say terminal oxidation, we know that it is cytochrome A3 complex, which is complex 4. So, which is the one which gives this last one to give received by copper center A, then it comes to cytochrome A, correct? So, then it transfers the electrons. The last one to have it, the electron is cytochrome A3, and all of this is within the complex 4. And the complex 4 is thinking, the cytochrome A3 within the complex 4 is thinking, what do I do with this electron? Somebody take it. How many electrons can I collect? Chemically speaking, it becomes unstable, right? So, somebody has to take it, and that is where oxygen comes into picture, and it says, give me the electron, you don't worry. I will create a water molecule which is fairly harmless molecule. That is called as terminal oxidation. The oxygen receives the electron. Electron is electronegative. Oxygen doesn't like negativity. What does it do? It thinks and says, let me neutralize the negativity by picking a proton. It picks up a proton, it has an electron, and it is there. Of course, there's an oxygen, and you get a water molecule. Correct? You get a water molecule. Next question. Think and answer a very basic question. Which of the following process CO2 is not released? CO2 is not released in which of the following? Yes. Aerobic respiration in plants, aerobic respiration in animals, alcoholic fermentation, lactate fermentation. Yes. What's the answer? Lactic acid fermentation doesn't release any CO2. Aerobic respiration in plants and animals are the same, poor thing. Both do cellular respiration in the same way. They will release CO2. Do you remember, I don't know if you have ever read a story where they would say, or ever spoken to your grandparents and they would have told you that they never used to sleep under the tree in the night because it releases CO2. There's no light for oxygen evolution. So light reaction releases O2, but otherwise it is releasing CO2 just like any other living organism. Okay. Lactate fermentation is lactic acid fermentation, which even our muscle cells show. Imagine if CO2 was being produced all over body. Imagine if we were, exercising or you're running to catch your college or school bus and your muscles are fatigued and they are not getting enough oxygen. What do? They start producing lactic acid. Imagine CO2 being liberated throughout your body. Would it be possible? Is it seen? No. Correct? Yes. Chemiosmotic theory of ATP synthesis in mitochondria is based on, or in general if they ask you, chemiosmotic theory of ATP synthesis even in chloroplast for that matter is based on what? Is based on what? Very good. Very good. Good job. It is H plus gradient. Very good. Good job. Good. Interesting way of using words in the question here. The energy releasing metabolic process in which substrate is oxidized without an external electron acceptor. Without an electron acceptor, external electron acceptor. Think. There are two conditions. It is an energy releasing metabolic process. Substrate is getting oxidized, two conditions, but there is no external electron acceptor. What is the answer? Fermentation, glycolysis, quite a tough war between the two. First of all, photorespiration is out of the race because it is not energy releasing. Correct? Glycolysis, could that be the answer? Substrate is oxidized, they say. In glycolysis, you are seeing partial oxidation, not complete oxidation. Correct? Read the statement properly. The energy releasing metabolic process in which substrate is oxidized without an external electron acceptor. What would that be? In glycolysis, what are you seeing? Partial oxidation. In fermentation, what are you seeing? You are seeing partial oxidation. Fine. But very specifically, we can say that there is no external electron acceptor here. Very good. In glycolysis, one external electron acceptor. No, it is also incomplete oxidation only. It is incomplete oxidation. But in glycolysis, what happens? There is one step where NADH formation happens. Do you remember NADH formation happens where? When glyceraldehyde 3-phosphate converts into 1,3-bisphosphoglyceric acid, a proton or rather a hydrogen molecule gets released. So there NAD plus will come and take that hydrogen and become NADH2. NADH plus H plus you can write, or NADH2 you can write. So here indirectly when H2 is released, electron release we can say indirectly. Okay. Fermentation is the answer because you are not seeing any NADH formation there. Aerobic respiration, who's the external electron acceptor? Oxygen. Oxygen. Okay? Remember. So the answer is option C. Which of the following statement is incorrect? Quite long statements. Start reading it, please. Yes. I'm starting the poll. Yes. Done? Done? Okay. Oxidation reduction. Oxidation reduction reaction produces proton gradient in respiration. Yes. So when the electrons are transported from one complex to another, there's gain of electron, loss of electron. What is that? The series of reaction of gaining and losing electron is redox reaction, reduction and oxidation. And because of that, a proton gradient is created is correct. During aerobic respiration, role of oxygen is limited to the terminal stage. The only spot where oxygen gets directly involved is in the last step, terminal oxidation. In ETC, one molecule of NADH plus H plus gives rise to 2 ATP molecules and 1 FADH2 gives rise to 3 ATP molecules. Wrong. It should be the other way. It should be the other way. Incorrect they have asked. So option C. Oh, very good. First 100%. See there. Very good. Complex 5 is synthesizing ATP. That is also correct. Yes. Shall we go to the next question and probably the last question? That is this. Name the enzyme responsible for oxidative decarboxylation during aerobic respiration. Oxidative decarboxylation. What do you mean but why? Basaya. What why? I didn't understand. What is the question? Done? Correct. Oxidative decarboxylation is seen in link reaction where pyruvate gets converted into acetyl coenzyme A. Acetyl coenzyme A. What does it do? It converts pyruvate, which is a 3-carbon molecule, into acetyl coenzyme A, which is a 2-carbon molecule. That means 1 CO2 is released. Correct? Yes or no? 1 CO2 is released. The enzyme here is pyruvate dehydrogenase, and along with that, you will also see production of 1 NADH plus H plus. I have not written it completely here. Coenzyme A plus NAD plus. Okay. This requires magnesium. Incorrect they are asking here. Yes or no? In ETC, one molecule of NADH gives rise to 3 ATP and 1 FADH2 gives rise to 2 ATP. They have written ulta. They have exchanged those two. 3 and 2 they have exchanged. Now I hope it is clear. Done? Last question. Last question in this chapter. The only 5 carbon acid in TCA cycle is? The only 5 carbon, yes. Think. Recall. Yes, correct. This is 6 carbon, this is 4 carbon, 4 carbon and 5 carbon. That means the only 5 carbon one is alpha ketoglutarate. Something to remember. This is the leaderboard. See the leaderboard. We are going to the next chapter, plant growth and development. Okay. All right. First question on arithmetic growth. Plant growth and development. You don't have to do anything. When the moment the question is displayed, if you feel A, B, C, D, D is the answer. Then you put D. Your name will get automatically captured. Done? Arithmetic growth is mathematically expressed as, how is a graph? How is the graph? For arithmetic, the graph is like this. Correct? Final length is equal to initial length plus RT, where the unknown is R that you have to find out. You know the initial length, you know the final length, you know the time period that you have chosen. Okay? Yes, you get a straight line. R is the growth rate that you should find out. Maybe you are doing an experiment over over 30 days. So you know your time span. Now over 30 days, what has been the growth rate? Like initially, what was the length? Final after 30 days, what was the length? How much it has grown approximately per day? That would be the R value. Correct? Okay. What is the answer for this question? Quite a long statement. Plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called. What do you call this ability? What is this ability called? Done? Okay. What is the answer here? Yes. Plasticity. Totipotency refers to the ability of the plant cell to generate the entire plant body. To generate the entire plant body. Okay. Growth is maximum in the zone of. Growth is maximum in the zone of. Read it properly. Growth is maximum in the zone of. Cell elongation, cell differentiation, cell maturation, all of these. Growth they are asking, cell elongation. The cells are elongating, naturally growth will be there more, right? The cells have already differentiated, they have already matured. That means they are not going to elongate any further. They are going to probably remain the same in size, but they will do some special activity that will help the plant body to survive. So, elongation, you will see maximum growth happening. Factors which affect or influence growth, I just wanted you to go through this particular paragraph. So, one question I have taken. However, if you go back to like last 5 years of NEET or even 6 years, you will not find any question on this. So, here I'm not even putting poll. All of these help in growth or influence growth. Yes, correct. All of these. Now, we start with, now what is the main thing here? Pay attention to the functions and hormones. Function and hormones. Also the precursor molecule of the hormones. Very important. Like for example, indole derivative is auxin. Purine derivative if they are cytokinin. Such things. Terpene, gibberellic acid, carotenoids, abscisic acid. So that and all is important, and the first intro paragraph only they have given it. You have to go through that. 2 or 3 lines are there. So let us start with the functions of hormones. That is the first function. The hormone which is responsible for apical dominance is, what did I say? I said terpene derivative, no? I said something else. Yes. If you put A and all, answer, your name will not get captured. Put A. Okay. Break I'll give you directly at 2 o'clock. Let's finish it off. Take a break, have your lunch, get ready for your class 12th portion. In between, if I give break, then this entire session will stretch up until late night. What is the hormone responsible for apical dominance? Indole acetic acid, a type of natural auxin. GA is gibberellic acid, ABA abscisic acid. Florigen is the hormone that is responsible for flower production. You haven't studied this hormone. Coconut milk contains which of the following hormones? In coconut milk, which of the following hormone comes? ABA, auxin, cytokinin, gibberellic acid. Yes. Okay. Zeatin is the one. Okay. Zeatin is the one which is present in coconut milk and then corn kernels and all. Kinetin was the one that they found out in herring sperm DNA. Yes. N6 furfuryl aminopurine, again something that they have mentioned in your book. So I just wrote the name, that's it. Related to cytokinin only. So it's an aminopurine derivative, they say. Adenine derivative, they say in few cases. Answer is option C. Option C. Okay. To increase sugar production, sugarcane crop is, yes, sprayed with what? Sprayed with what? Sugarcane crop will be sprayed with what in order to increase sugar production? Done? Easy? Sugar production will increase in sugarcane if its length increases, and to increase length only one hormone helps, like in general to increase length, that is gibberellic acid. There is another hormone which can help in elongation of petiole and internodes. Ethylene. Ethylene also does it. Elongation of petiole and of course internodes in deep water rice plants, but only in deep water rice plants, not otherwise. Otherwise, in general, if I take, it is gibberellic acid. One more thing, gibberellic acid does not change the genotype. Change genotype of, pay attention here, dwarf plants. It cannot change the genotype. It can only change the phenotype. A plant is 2 feet, it will grow to 4 or 5 or 6 if you apply gibberellic acid. But the gene inside it, now if you take this plant and then let's say self-pollinate it or so, it will come, the offspring plant will come out dwarf only because the genes have not been changed. You have done external application and it has increased in height. Understood? This is very important. Students shouldn't think that, okay, the plant became 6 tall because the gibberellic acid went inside and changed the gene. Can you imagine changing gene is like gene replacement. Biotechnology you have learned, right? That gibberellic acid cannot do. It's just a hormone, poor thing. Okay, leave this. We don't want that. Photoperiodism is anyways removed. Yeah, come to this. Come to this. Yes. I know, interesting, but it was there in, every year I say this in the class, but it was there in this time CBSE paper also. Bio paper. One of the codes, I don't know which one. Yeah. Which of the following growth regulator is known as stress hormone? Poor thing, these stress hormones, have a bad name, but they actually help us survive. I mean, even our stress hormone for that matter. If stress hormones are not there, we would never be able to cope up with the stress around us. Stress hormones are of course bad, prolonged period, but when they are released, they help us to cope up. The same with plants. The plants are in stress of temperature. They will need stress hormone, otherwise they'll die. They will die. Diagram based question is this, taken directly from your NCRT. Done? So they have given you two diagrams and they have asked you what does it indicate. It shows something called as environmental heterophily, the example for which is buttercup plant. See how the plant has evolved. When in touch with air, the leaves are like this because they know air will not be very damaging. We can keep the lamina complete and sunlight will fall. We can do a lot of nice photosynthesis and produce food. But these are the leaves. When the leaves are in contact with water, they know the water will come and hit and there will be a lot of wear and tear and we'll have to spend a lot of energy in repairing this tissue. So let us not resist the water. Let us create these patterns so that water can very freely pass through the leaf. Yes, correct. Developmental heterophyly. What will be the answer for developmental heterophyly? Coriander and cotton and larkspur. CCL. Coriander, cotton, larkspur. Okay? Yes. Chalo. Next question. Cytokinin and auxins are antagonistic to each other. Antagonistic, opposite to each other. Opposite to each other. Yes, correct. Done? Antagonistic to each other. What is the answer here? Antagonistic means opposite to each other in apical dominance. If auxins are promoting the tip to grow, cytokinins are pushing the lateral growth. So the branches, the canopy growth as such is because of the cytokinins. Correct? Yes, another interesting question. Parenchymatous cells, if you take, put it into the lab, of course under controlled condition, in the sense you're giving it the right temperature, the right nutrient, you're taking care of it basically. What will happen to these parenchymatous cells during tissue culture if you do this? What is the answer? They will become differentiated. Will they become de-differentiated? Will they become re-differentiated? Or will they become undifferentiated? So those of you putting B, 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 your name will not get captured. Put only once, A or B or C or D. Parenchyma cells, they became meristematic. Meristematic cells can divide. Parenchyma cells have given up on division and they are like, let us grow up. We want to take up a special function. Okay. Now, these parenchymatous cells which have gone back to being meristematic again. What do you call this growth as? We call it as callus growth. Then these cells are called as de-differentiated cells. Very good. If these callus cells become parenchyma again, you will call them as re-differentiated. Correct. Re-differentiated. In sigmoid growth curve, very good, the exponential phase will slow down, leading to a stationary phase due to. Done? Limited nutrient supply. Yes or no? In exponential phase, sigmoid growth. What is this phase or this growth curve? It is geometric growth that they are talking about. Yes, geometric growth that they are talking about. Correct? Yes. This is a little difficult question, but let's just take it up. So phototropic curvature is the result of uneven distribution of what? Uneven distribution of what? Yes. Phototropic, the moment they say, answer is auxin. Correct, auxin. Bending towards light. Bending towards light is due to auxin's response. It is because of auxin. Correct. Which of the following plant growth regulator is responsible for respiratory climactic? Respiratory climactic. What is this event of respiratory climactic? Respiratory climactic is when the rate of respiration increases when the fruit is undergoing ripening. When the fruit is undergoing ripening, then rate of respiration increases. So who's causing ripening here? Ethylene, option B. Oh, another 100%. Good. Tasnimul, thank you so much. Okay? Ethylene is the answer. Okay, thank you. You don't even realize if, no problem. Okay, thank you. To all of you, love from Bangalore back at you. Okay. Yes, let's start with this. Which one of the following hormone is concerned chiefly with root initiation? Okay. All right. Good to see a mix of students. I did live in Assam very briefly when I was very young. Yes. Like I said, love from Bengaluru back at you all. I lived in UP also very briefly. Gorakhpur. Okay. Which one of the following hormone is concerned chiefly with root initiation? The moment they say root initiation, the hormone is auxin, my dear students. Auxin is responsible. Okay? Not Dholakpur, Gorakhpur. Yes. Dholakpur is in Chhota Bheem cartoon, right? So, increased auxin, see in tissue culture, if you take more auxin, then in combination you have to take this, less of cytokinin. What do you get? You get root. Then if you take less of auxin and then more of cytokinin, what do you get? You get shoot. I'm talking about tissue culture, okay? But if you take auxin and cytokinin equally, if you take auxin and cytokinin equally, what do you get? You get callus. You get callus. Okay? Yes. Which one of the following induces, pay attention, femaleness in plants? What do you mean by femaleness? I'll give you one hint. Femaleness means female flower production. Yes, female flower production. Done? Okay. Yes. Female flower production in cucumber. Ethylene does this work. Ethylene does this work. Correct. So female, not ethanol. Ethanol, no. Next question. Weed killer. Weed killer. Yes. Ethafon. Correct. Commercial ethylene. Done? Two 4D, a type of a synthetic auxin. Very good. Another 100%. This is the third time. Good job. Bolting hormone. What do you mean by bolting? Just before flowering, there's a very specific event that happens. Yes. Gibberellin. Elongation of internode before flowering. Oh, another 100. Just missed. Okay. Assertion and reason type of question, more like statement based question. Try to solve this. Auxin prevent fruit and leaf drop at early stage. Auxin promote abscission of older mature leaves and fruits. Auxin prevents fruit and leaf drop at early stage, and it is the same hormone which will tell the older mature leaves and fruits to detach. It will not allow the younger counterparts to, both are correct. It will not allow the younger parts to detach from the mother plant. It will tell them to stay back. But at the same time, it will help the older ones to detach. So option C, both are correct. Last few questions from plant growth and development. Ethylene will initiate what? Flowering in pineapple, flowering in mango, synchronizing the fruit set in pineapple. All of these. All of these. Done? All of these. It can initiate flowering in pineapple and then synchronize it. It can also initiate flowering in mango. Hence, all of this synchronized fruit set production in pineapple. In most situations, ABA acts as an antagonistic to, as an antagonist to. Opposite of what? Opposite of what? Done? Easy? Yes. Good afternoon. It is antagonistic to gibberellic acid, especially gibberellic acid promoting seed germination, and then ABA suppressing seed germination. Suppressing seed germination. Answer for this. Read the following statements regarding cytokinin. Regarding cytokinin. Kinetin occurs naturally in plants. Kinetin was discovered from coconut milk. Zeatin does not naturally occur in plants. Zeatin was isolated from human DNA. How many of the statements are incorrect? Incorrect. Kinetin does not naturally occur in plants. Okay. It was discovered from coconut milk. Zeatin was discovered from coconut milk. Correct? And maize kernel. Zeatin does not naturally occur in plants. And zeatin was first isolated from human DNA. What should be the answer? Zeatin does not occur naturally in plants, is it? Zeatin does not naturally occur in plants? Or it does? What is the answer for this particular question? Check your book. Zeatin does not naturally occur in plants. What am I saying? Zeatin is part of coconut milk, part of kernel, corn kernel. Then it does occur, right? So this is wrong. Zeatin was first isolated from human DNA. Can we say? Can we say that? Was first isolated from human DNA. Human DNA, is it? Zeatin was first discovered in corn kernel, okay. Kinetin, yeah, that too human, derived from human DNA, isolated from human urine. That would make sense. So this is also wrong. So that means all the 4 statements are wrong. Option D would be the answer. Correct? Option D would be the answer then. All the 4 statements are wrong, na? Yes or no? Yes. All the 4 statements are wrong. So that is the end of plant physiology. Now let's go to which chapter? Shall we go to cell cycle, or shall we mix it up? Quickly we'll mix it up. Or shall we start with living world? Yes. Let's start with first question here. Let's mix it up a little bit. Yes, we'll mix it up, don't worry. Try to answer this question. One question you can expect, okay? One question. That is mostly from binomial nomenclature, or that wheat, human, housefly and mango chart what they have given from there, or this. As we go from species to kingdom in a taxonomic hierarchy, the number of common characters will remain, will decrease. Correct? Yes. Because as you go higher up the taxonomic hierarchy, number of similarities decrease. This is living world, first chapter. And as you come from kingdom to species, the similarities goes on increasing. The similarities go on increasing. Another question from this we will do, and then we will mix it up. Which of the following suffixes? Suffix means at the end of the word what is added is suffix. So, which of the following suffixes used for units of classification in plants indicate a taxonomic category of family? Recall all the families that you know in plants. Recall all the families that you know in plants, you will get your answer. In the end, what are the alphabets that are used? Solanaceae, then what was that? Poaceae, Anacardiaceae, correct? The answer is option C. Solanaceae, Liliaceae, Fabaceae, Brassicaceae, Anacardiaceae. Answer also C only here, option C. Correct? Okay. Let's go to the next question. A common characteristic feature of plant sieve tube cell and most mammalian erythrocytes is. Most mammalian erythrocyte is. Yes. Come on. Sieve tube cells are found in phloem and mammalian erythrocyte. Both of them lack nucleus and most of the organelles for that matter. Most of the organelles are absent in them. Okay. Next, which one of the following is not a eukaryote? Not a eukaryote. Euglena, Anabaena, Spirogyra, Agaricus. Which one of the following is not a eukaryote? Not a eukaryote. That means it's a prokaryote. Because rest of all the kingdoms are what? Eukaryotes. There's only one kingdom which is prokaryotic. Euglena comes under which kingdom? Protista. Anabaena comes under which kingdom? Monera. Blue-green algae. Spirogyra. Plantae. Agaricus. Fungi. The answer is option B. Blue-green algae. Correct? Yes. Next question. Which of the following stain is not used for staining chromosome? Little difficult. Little difficult. Not used for staining chromosomes. Done? Okay. What is it? Safranin. Safranin stains the cell wall. Cell wall, my dear students. Methylene green also stains the nucleus. Basic fission, carmine, all of these stains nucleus only. Okay. Clear? Let's take this question. Fucoxanthin is found in plants belonging to which group? Which group? Fucoxanthin is found in plants belonging to which group? Out of NCERT, yeah, but not out of syllabus because you have been doing practicals for so long. You have used such different types of stain. Do you remember using cotton blue stain? Where did you use that cotton blue stain? And if you're aiming at being doctors, then this is something that you should be knowing. Pathology. Anyways, fucoxanthin phaeophyceae. Fucoxanthin phaeophyceae, brown algae. Bryophyta will have chlorophyll A, B, carotenoids and so, yes, pollen tube. Rhodophyceae, red algae, phycoerythrin. Chlorophyceae, chlorophyll A and B. And again, carotenoids. Pyrenoids contain what and all? Pollen tube growth in stigma. Very good. Correct. That is why you use cotton blue stain. Done? Protein and starch together give you pyronoids, which is stored food material in chlorophyll. In chlorophyll. Correct? Yes. Now, next question. Order for convolvulaceae. Which of the following is order for convolvulaceae? Hello, Shivu, hello. Yes, convolvulaceae. Done? Order polymoniales. Yes, correct. Select the mismatched pair. Select the mismatched pair. Just a minute. One question from this chapter, you can expect one question. Very few years they have not asked any question. Yes. Done? Okay. Panthera, Mammalia, Musa, Mucidae, Triticum, Poales, Solanum solanaceae. Why Anacardiaceae? No. Anacardiaceae is mango family. Solanum is Solanaceae family. Okay. Fine, then we'll do that. Let's come to this question then. Match the following and choose the correct option. Match the following and choose the correct option. Done? Family. Solanaceae. Kingdom, of course, Plantae. Order, Polymoniales. Species, Tuberosum. Genus, Solanum. 5, 4, 2, 1, 3. 5, 4, 2, 1, 3. Option A. Very good, 100%. Good job. Which of the following is a defining characteristic of living organism? Yes. Yes, I know only one match will do, but I had to do the entire thing. Yes. Done? Okay. Which of the following is a defining character? Response to external stimuli. which is called as consciousness. Consciousness, response to external stimuli. Normally questions don't come from this because in a new NCERT they have kind of removed it, but in NMC they have like mentioned, so I thought let me take at least 1 or 2 questions. Reproduction and growth are not definitive characters or not defining characters, remember, because everything that grows cannot be living and everything that cannot reproduce cannot be, of course, called non-living. Remember that. So this is called as consciousness, which is a sure short feature of a living organism only living organism have it. Metabolism and cellular organization is another 2 you can add, and cellular organization are another. So in total you have 3 features which are like called as defining features. This that is consciousness, the same thing. Consciousness, metabolism and cellular organization are called as defining features. This much you remember enough. Okay. Last question, no, not last question, but yeah, maybe one more question on binomial nomenclature. Hello. Yes. Taxonomic unit phylum in the classification of animal is equivalent to which hierarchical level in classification of plants? Like they both are at the same level of what? Division. When you say phylum in animals, then you say division in plants. Correct? Oh, another 100%. Good job. Good job. Okay. Clear? Just a minute. There was another question. Okay, I think we'll do a thing. There's another question on binomial nomenclature. The slides have got a little mixed. Only those 2 slides are somewhere lost. So it might come up somewhere. Okay. How can I say sure? These concepts will be there for sure. Question no. Because they haven't, I am not the one setting the paper. Na? Yes. So binomial nomenclature, you can expect one question in between somewhere, okay? Then, which one of the following is not true for ribosome made up of 2 subunit? Yes, they form polysome called as polyribosome. They may attach to mRNA. They have no role in protein synthesis. Their whole life is protein synthesis. Oh, good job. Their whole life is protein synthesis. Without protein synthesis, they will not function. Who proposed fluid mosaic model? Scientist based question. Yes. Done? Yes. What is the answer? Singer and Nicholson. Robert Brown was the one who discovered nucleus. Clayton and Schwann, cell theory. Benda was the one who discovered mitochondria actually, but yeah, you don't have to remember. Which of the following statement is true for a secretory cell? A cell which is involved in secretion, continuous secretion. Golgi apparatus is absent. RER is easily observed in the cell. Only SCR is present. Secretory granules are formed in nucleus. Done? What is the answer? RER is easily observed, even Golgi apparatus is easily observed for that matter. Problem with option C, secretory granules. True, they are asking you, true for secretory cells, okay. Secretory granules are not formed in the nucleus. This is wrong, in nucleus. Next question. The non-membrane bound cell organelle common in both prokaryotic and eukaryotic cell. Both prokaryotic and eukaryotic cell. What is the answer? Yes. What is the answer for this question? Non-membrane bound, no membrane around, and found in both prokaryotes also and eukaryotes also. What is it? No membrane in centriole also, of course, but centriole is found only in animal cell. The answer is ribosome. What is mesosome? Infolding of the plasma membrane. Lysosome is an acidic organelle, we say. Lysosome is an acidic organelle. Plasmid DNA that you find in bacteria, the plasmid DNA is found in all eukaryotes, confers certain unique phenotypic characters to organism, is the genetic material of prokaryotes, is larger than genomic DNA. This class will go up to 2:15 probably, and then we end the session. This botany part 1 session, and at 2:30 or so another session, that is your class 12 syllabus session, will begin. Okay. So the plasmid DNA is what confers certain unique phenotypic characters to the organism. One example is antibiotic resistance. Antibiotic resistance is one character. It is not the genetic material of prokaryotes. If you remove plasmid, the bacteria will not die. Remember that. And it is a very small portion. It is smaller than the genomic DNA. Genomic DNA means the chromosomal DNA. Okay? Clear? Understood? Next question. The outermost layer of bacterial cell envelope is? Bacterial cell envelope is? Done? Outermost layer they are asking you is glycocalyx if present. Cell wall is the one which is composed of peptidoglycan. Cell membrane, of course, we all know. Plasma membrane or cell membrane is the same. Yes. Next question. Select the incorrect statement. In bacterial cell wall, bacteria cell wall determines the shape. Glycocalyx protects cell from bursting and collapsing. Loose sheath, outermost protective layer of bacterial cell envelope is slime layer. Glycocalyx differs in composition among different bacteria. What is the answer? The answer is, they are asking you incorrect statement. We know this is right. Loose sheath-like protective layer of bacterial cell slime layer is correct. Glycocalyx is different in composition among different bacteria. Correct. It can either be capsule or slime layer. It is a cell wall which prevents the cell from bursting. So the answer turns out to be option B, not glycocalyx. Good job. You guys are doing really well. Okay, really well. Wherever you're failing in the sense you're not able to answer, make a note of those concepts. And if you have answered some questions by maybe I think it is A, maybe I think it is A, maybe it is B, and you're clicking on the answer or you're answering and it becomes right, Don't consider yourself masters in those concepts. You've got them right by fluke. Make a note of those topics also, because you will not take that risk in NEET. There's negative marking, na? Yes. Mesosomes do what? They help in replication and respiration process, yes. They are not found in eukaryotes, they are found in prokaryotes. They are not extensions of slime layer, they are extensions of plasma membrane. And they do not contain pigments for photosynthesis. That work is given to chromatophores, which are found in blue-green algae. So, option A would be the answer. Mesosomes are infoldings of plasma membrane. What are inclusion bodies? What are inclusion bodies? Yes. Done? Yes. Inclusion bodies are or include store reserve food material would be the answer. They are not found in eukaryotes. They are single membrane bound. There is no membrane. There is no membrane. They include gas vacuole, okay, but not sap vacuole. They are found in eukaryotes, wrong. They are found in prokaryotes. Select the incorrect statement for lipids of plasma membrane. For lipids of plasma membrane. Done? Incorrect statement. Heads are polar, situated towards outer side. Mainly consists of phosphoglyceride, Non-polar tails are situated towards inner side. Tails are hydrophobic, not hydrophilic. So, the answer turns out to be incorrect. Hence, option D would be the answer. Option D is the answer here. Option D is the answer here. Which of the following is the component of endomembrane system that have ribosomes? Which of the following are components of endomembrane system that have ribosomes? Major site for synthesis of lipid, this is SER. Involved in protein synthesis and secretion, RER. Double membrane bound, you have mitochondria, chloroplast, and you have even nucleus. Discovered by Camillo Golgi is Golgi, and the answer is option B. Option B, correct, a part of the endomembrane system which has ribosome. Along with Golgi, endoplasmic reticulum, you have vacuole and lysosome also part of the endomembrane system. Identify the wrong match. Golgi body, dense reticular structure found near nucleus. Lysosome is formed by packaging in Golgi. RER forms SER. Vacuole is found in plants only. What is the answer? Think. Don't be in a hurry. Think. Don't be in a hurry. Think. Yes. Option D. Vacuole is not found only in plants. You find it in Protista also. You find it in Protista also. Contractile vacuole, food vacuole. RER, yes, it does form SER. This is correct. A lysosome is formed because of the packaging that happens in the Golgi. Yes, they arise from Golgi. Golgi is a dense reticular network-like structure around the nucleus. Yes, this is also right. All NCERT. All NCERT. Next. Important site for glycoprotein synthesis. Glycoprotein synthesis. Done? Done? Very easy. Glycoprotein synthesis. Golgi bodies. Yes. Ribosome does what? Protein synthesis. Nucleus is the one carrying the genetic material. RER is also protein synthesis. And of course, it's packaging and not packaging exactly, but then yeah, I would say secretion to an extent. Yes. Done? Next question. Read this. In plant vacuole, the dash facilitates transport of a number of ions and other materials against concentration gradient. Done? Okay. What is the answer? Tonoplast. It is a membrane of the vacuole which help in transportation of ions and other materials against concentration gradient. Good job. Another 100%. Identify the incorrect statement for mitochondria. For mitochondria, which of the following like incorrect statements? Not easily visible under microscope, inner membrane forms several infoldings. They divide by fission, the only double membrane bound structure of animal cell. What is incorrect? Option D. Am I right? Yes. Why? They do divide by fission. They form infoldings, the name of which is cristae. They are not easily visible under microscope. You would need electron microscope to visualize them and a special stain called as Janus green. Okay. Yes. This is clear. Two more questions from cell chapter here. In the below diagram, identify the part which connects the peripheral microtubule to the central sheath. To the central sheath. Yes. Which is the one which is connecting the peripheral microtubule to the central sheath. What is this? This is TS of cilia or flagella. TS of cilia or flagella. Answer is radial spoke. Correct? Yes. What is the arrangement of the microtubule? 9 plus 2. 9 plus 0 would be the arrangement of the centriole. Centriole. Yes. Central microtubule. Let us come to this question. Identify the part which is labeled as M in the given diagram. Yes. Identify the part which is labeled as M in the given diagram. M. Very good. Option? Yes. Option C. Satellite. Centromere is this. This is centromere. Correct? The tips are called as telomere. This is arm. This entire thing, if I take, is chromatid. This narrow portion is called as secondary constriction. Centromere is called as primary constriction. Kinetochore will be a plate-like structure around the centromere. Correct? Option C. Yes. That marks the end of cell chapter. I think cell is the one of the easiest chapters. So, let's come to cell cycle very quickly. But before that, I told you 2 questions on binomial nomenclature I wanted to do. So, I'll do this very quickly, okay? Yeah, telomere is the tip of the arm. The tip of the arm is called as telomere. Telomere, okay? Binomial nomenclature means what? Rules of binomial nomenclature also you have to go through. So, do remember you have to go through rules of binomial nomenclature. Don't forget that. Okay, one scientific name which consists of generic and the other will be the specific epithet. Very good, another 100%. Good going, guys. Very good. And I told you about the rule. Which of the following statements regarding nomenclature is correct? They are asking you. Correct. Generic name always begins with capital letter, whereas specific name with small letter. Scientific name should be printed in italics. Scientific name when typed or handwritten should be underlined. And the names are given in Latin. Answer is all of the above. All of the above. There is another point that I would like to add here, which is given in your NCERT. After species name, okay, name comes author's name, the person who for the first time described it. Author's name in short form in abbreviated manner. So, short form like Linnaeus means L-I-N-N. So, abbreviated manner. It should not be in italics. If you are printing it, the name will not be in italics. And if you are underlining it, then genus and species should be underlined, but not the author's name. Not the author's name. Remember this point also. Okay. Zoology, I think, if I am not wrong, 1st of May. I am not sure. Subspecies need not be entered. Subspecies need not be entered. 7 obligate categories are there. Kingdom, phylum, class, order, family, genus, species. Only that is needed. Subspecies, sometimes we don't even know subspecies. It's not a compulsion to write it. What is there in interface? There are 3 phases, G1, S and G2. So, does it involve DNA replication? Yes, it does. That's the answer. Constitutes less than 50%, 95% and more of the duration is taken up by interphase. Is the phase of actual cell division? M phase. Does not include RNA formation. It does include RNA formation. RNA formation happens when there is synthetic activity going on. Correct? Transcription is going on. If in G1 phase and G2 phase proteins have to be synthesized, then that means translation has to occur. If translation has to occur, transcription has to occur. And transcription is nothing but formation of RNA. Yes or no? So, answer will be option A. Okay, option A. Next question. A typical human cell divides approximately once in, complete question would be once in 90 minutes, 15, 90, 30, 24 hours. 90 minutes is east. The answer is option 4, 24 hours. What is the confusion here? You think our body cells divide in 1.5 hours? Come on guys, there are 46 chromosomes to replicate. If a cell has to divide, DNA has to replicate, no? So many organelles have to be synthesized. 1.5 hours is not enough. Resting phase of cell cycle is, think and answer. Resting phase of cell cycle is where the cell is resting. Resting phase of cell cycle. Yes. Resting phase of cell cycle is interphase. Why? Because the cell will show no division, not because it is not showing any metabolic activity. No division. Is it showing metabolic activity? Yes. Metabolism? Yes, it does. But no division. That's why you're calling it as resting phase, okay? Don't think that metabolism is not there. The moment metabolism stops, that means the cell is dead. As simple as that. The next question. Some cells exit G1 cell cycle and enter into a phase called quiescent stage, in which it can easily proliferate, where cells remain metabolically active. What is the answer for this question? Answer for this question. Done? Yes. If it exits the G1 phase of cell cycle, that means it is entering into a phase called G0. Is it called quiescent stage? Yes. Does it proliferate? Proliferate in the sense the cells divide? No. Cells remain metabolically active? Yes, of course. The moment metabolism stops, the cell will die. These cells actively living, but they choose not to divide. They don't want to divide. They don't want the hassle of division. And that could be, they could be resisting it or blocking it temporarily, or it could be permanent. So, A and C are true. Correct? A and C are true would be the answer. Please remember that. They are temporarily blocking the G0 cells are temporarily blocking that DNA, I would say replication, or in general division. Okay, they are simply resting. So, it could be temporarily, like parenchyma cells could temporarily block it. Permanent, okay, permanent blocking will be by neurons and muscle cells. Clear? Okay. Next, identify the wrongly matched pair. Identify the wrongly matched pair. No, I will not go fast. Prithviraj Chavan. Whoever you are, I don't care, and I will not go faster than this. If you want, you can get out of the class. Yes. Done? Next. Yes. Prophase ER not visible at the end of the phase. We'll change the color. Let's use red. Yes. In prophase, endoplasmic reticulum is not visible at the end of prophase. Yes. because all the organelles are going to get disintegrated. They are going to like literally become gayab. Metaphase. Chromosome are thickest and shortest. Yes, that is why we study chromosomes in the metaphase only, because that is when they become visible and we also come to know what is the position of centromere and so on. Anaphase. Chromosomes are clustered at opposite pole. Telophase, the mitotic spindle will disappear. Yes. But in anaphase, the chromosomes are in most of the late anaphase, they have already reached the opposite pole, we say. But then the majority of the anaphase is seen when the chromatids are moving towards the opposite pole. Like they've just started their journey towards the opposite pole. Yes or no? Yes. So here when I say anaphase, they don't cluster. Now this could be, this is for telophase, it would be apt. Yes, for telophase this would be apt. Then the answer turns out to be option C. Yes or no? What is the confusion? Why have you marked A and D? Spindle will disappear in telophase because the work is done. The chromosomes have moved to opposite pole. Now spindle fibers are not needed. So in telophase, naturally they will disappear. See, whatever is not needed by the cell will not store it. It will get rid of it. It's a very clean system. Mitotic spindle's work is done, digested, gone. Needed again, it will recreate it. Okay, that's a different story. Right. Thank you guys. Thank you. The best stage to study shape of chromosome is. Yes. Done? Vidya, Prashu, thank you. What about this? This is easy. Just now I explained, so I think you will be able to solve this. See, whatever chromosomes we draw, right? I showed you one chromosome diagram and we did a question. That was metaphase. Why do we always draw like that? When it is, see, remember one thing, the chromosomes are not always lying like that. If this is a nucleus, when the cell has decided not to divide, this is how the chromosomes are lying. They are very thin, frail, they are not condensed, and we cannot see them properly, so we do not draw this. Why do we always draw chromosomes like this? Because this is how it appears in metaphase, and that is when it becomes visible to us. So whatever is visible, only that we will draw, na? Yes or no? If tomorrow you become a or if you are maybe a portrait sketching kind of a person who sketches very well, now that whatever visible to your eyes, that you will draw, right, in the portrait. That person might have had a dog at home. Will you draw that dog unless and until you are told? You will not, right? Because right now in front of you that person is sitting, you have to draw the portrait of them. The same. Chromatin fibers, how they appear often we don't know because they are not visible properly. But we know when the cell plans to divide and then in metaphase it becomes very thick and short. Okay, this is how it appears for me, then I will draw it the same way. Remember this. Which of the following occurs during cytokinesis of an animal cell? Okay, Shashi, thank you. Tasmia, alright, I will take that as a compliment. Thank you, Vidya. Yes. As long as it helps you guys. Yes. Which of the following occurs during cytokinesis of animal cell? In animal cell, you will see that there is cell furrow formation. I'll just show this to you. So, imagine there are two nuclei already formed. Now, this cell, what it will do? It will start creating a kind of a furrow. Furrow in the sense depression. And this depression is moving from the periphery to the center like this. So, it is moving towards the center. So, it is not centrifugal, it will be centripetal. Phragmoplast formation, whatever they have given, this is in plants. Of course, that will in future, phragmoplast will become middle lamella. Okay, this becomes middle lamella, not cell wall, remember that. calcium pectate in middle lamella. Cell wall formation of course happens in plant cell. Centrifugal also in plants. In animal it will be centripetal because it moves towards the center. Okay, towards the center. So option A would be the answer. Even if you didn't know all of this, furrow formation you know, na? Mitosis is not required for. Where is mitosis not required for? One session of expected questions of NEET. Anam, this is almost similar to that because the questions that we have taken is kind of in line with whatever portions and whatever portions have appeared in NEET regularly. Okay. Vidyashri. Okay, I'm from Andhra Pradesh. I'm from Karnataka. Yes. So, not required for healing the wounds. It is needed. Growth of multicellular organism, growth the moment you say it is mitosis. Maintenance of cell size, introduction of genetic variation. For if you want genetic variation to get introduced, rely upon meiosis. Mitosis will not help. Then ma'am, maintenance of cell size, does it? Of course it does. Imagine a cell becomes too big and the nucleus becomes very small. Now the cell starts to worry because there is no balance here. Nucleus is very small and it is not able to control the cell. Cell is too big. Now the cell will decide to divide. When it divides, the nucleus to the cell size ratio, what is there, that will be balanced. Okay. Metaphase is the best stage to study chromosome. Anaphase is the best stage to study the position of centromere. There's a difference between the two. Okay. I'll give you 45 seconds for this. In anaphase, when the chromatids are moving towards the opposite pole, you will be able to study the shape of the chromosome. L shape, I shape, J shape. That will tell you if the chromosome is metacentric, submeta, acro or telocentric. But then in metaphase, you will study the chromosome structure, shape, anaphase with respect to position of centromere, because every chromosome has a very different centromeric position. Suppose Y chromosome is telocentric, we say. That way. Okay. Total number of mitotic division required for formation of 32 daughter cells from one cell. Let us derive, the formula is n minus 1, where n is equal to number of daughter cells. Let us derive the equation. You have one cell, divides into two daughter cells. This divides further into two daughter cells. So, total how many daughter cells you got? 4. Let us count the number of divisions. One division here, second here, 3 here. So, 3 divisions. So, what would be the formula? N minus 1. So, for us to get 4 daughter cells, 3 mitotic divisions are needed. Then, for us to get 32 daughter cells, how many divisions would be needed? 31. 31, my dear students, 31, because it says from one cell, from one cell. Okay. Tasmia, don't be scared of assertion and reason. Only 1 or 2 will be asked. So, assertion and reason, let's see. And most of the assertion and reason come from your class 12th topics. So, class 12th, when ma'am is doing, she will be doing assertion and reason as well. So, don't worry much about assertion and reason. Just 2 questions are asked. One small tip is, when the assertion and reason are related, I mean you read when both are correct first and they are related, then the answer is most often that reason is the correct explanation of assertion, most often. This is one short tip that I can give you. Done? Good afternoon. Yes. Prophase 1 is similar to prophase 2 as both occur in gametes, both include 5 substages. They don't. Both occur in gametes. Nuclear membrane disappear in both. Both are complex and prolonged stages. Prophase 1 and 2 are similar as both occur in gametes. Do both include 5 substages? No. The nuclear membrane will disappear in both? Yes. They both are complex? No. Prophase 1 is complex but not 2. They both occur in gametes. Now they may occur, may not occur. Correct? They may occur, may not occur. That we don't know. But we very surely know that if it occurs, the nuclear membrane will disappear for sure. Correct? Yes. That is unavoidable. Where do you see the activity of enzyme recombinase? Recombinase activity, enzyme recombinases activity is seen where? It is seen in meiosis 1, to be very specific, in pachytene. In pachytene. Correct? Seen in pachytene. Okay. Next question is this. Statement based. I am giving you 45 seconds. True or false? Synaptonemal complex dissolves at pachytene stage. Synapsis occurs between non-homologous chromosome. Diplotin may last for months or years in oocytes of some vertebrates. Solve this. Yes. So, in leptotene, you will see that the condensation begins. In zygotene, you will see that the homologous chromosomes come together. And because they have decided to come together, why have they come together? First of all, there should be some reason, na? Because there is some kind of a genetic exchange happening here, going to happen here. And till it happens, they are scared that they will be separated. Till it happens, synaptonemal complex will hold them together. It says, I am there, you do your work. And then in pachytene, what will happen? The exact exchange of the genetic segment happens. And here you will see the event of recombination or crossing over. Once that is done, in diplotin, what will happen? The synaptonemal complex will dissolve. But the point of crossing over becomes visible to us because synaptonemal complex has dissolved. The way these two have, crossed over, that point. So, chiasmata visibility is seen in diplotin. Why? Because the synaptonemal complex dissolves. Remember that. And then in the diakinesis, what are you seeing? You are seeing there is terminalization of chiasma. Terminalization of chiasma. Is here. So then the answer should be what? Synaptonemal complex dissolves at pachytene stage? No, false. Synapsis occurs between non-homologous chromosome? False. It has to occur between homologous chromosome. Diplotin may last? Yes. For several human beings are a very good example for that. So false, false and true. Option D. Option D. This should have been homologous chromosome, not non-homologous. What is a bivalent? What do you mean by a bivalent? A bivalent is a pair of homologous chromosome. It's a pair of homologous chromosome. Correct? Yes. It is called as a tetrad. Yes. Is it called as a bivalent? Yes. Non-homologous chromosome? No. This is correct. Includes 2 chromatids? No. 1, 2, 3, 4. That is why the name tetrad. So, this is wrong. Includes 2 homologous chromosome? Yes. Because there are 2 homologous chromosomes, we call them as bivalent. So, this is correct. So, B and D are true. B and D are true. Option D. Very good. Good. Alright. So this is clear. Last two questions we will take. Separation of the homologous chromosome. When does it occur? When does the homologous chromosome separate? The homologous chromosomes are separating when? In the homologous chromosomes will separate when? This is happening in anaphase 1. The reductional division exact phase is anaphase 1. In anaphase 2, sister chromatids separate. This difference you should know. Very important. Still last moment in cell chapter, what to study? If you ask me, I would still suggest meiosis 1. Last is interkinesis. See a question on interkinesis. Interkinesis lacks. Interkinesis first of all between what? Between meiosis 1 and meiosis 2 comes interkinesis. Is there DNA replication there? Certainly not. RNA formation, yes. Protein synthesis, yes. Tubulin synthesis, yes. Centriole duplication, yes. But then DNA will not replicate. Centriole will duplicate, but DNA will not replicate because it is already done in interphase. Okay? Okay? Already done in interphase. So again another round of DNA replication will not happen. That happens in interphase, then meiosis 1 happens, then interkinesis, then meiosis 2. All right. So, I hope cell cycle and cell division is clear. No doubt. Let's very quickly go to, now we will go in order, biological classification, plant kingdom. Let's take one of this question. This is plant kingdom. Now plant kingdom. Done. Agar is commonly used in culture medium and is obtained from what? A red algae called as Gelidium. A red algae, very good, called as Gelidium. Next, isogamy with non-flagellated gametes is found in. Isogamy, non-flagellated gametes. Isogamy with non-flagellated gametes is found in what? Isogamy also and non-flagellated gametes. Answer is Pyrogyra. Correct? Very good. Eulothrix, flagellated. Chlamydomonas also flagellated, and this is oogamy. Volvox, fucus and all show oogamy. Correct? Yes. Phycoerythrin is one of the major pigments. A, D and phycoerythrin. One of the major pigments in what? Major pigments in what? Correct. Erythros, erythrin. Erythros means red. Answer is option C. Very good. Very good. Fucoxanthin, brown algae. Okay. Another question. Two more questions on algae. Red algae differ from green algae as they lack. See, in PW English only, top 50 topics of NEET, I have done one video. You please refer to it. That is based on last 5 years of question paper. If you see plant kingdom, most of the questions come from algae part. You please see that. In that, zoology is also there, so that you can, last moment pay more attention to these topics. Red algae will differ from green algae as they lack. Who lacks? Red algae will lack flagella throughout the life. Red algae does not contain any motile structure including their gamete. Even their gametes are non-motile. Okay, remember this. Remember this. Yes, another, probably the last question. This information important. In red algae, that one chart they have given in NCERT. Please go through that chart. Their cell wall material is mentioned, pigment is mentioned, these kinds of spores also they have mentioned. So, a class of algae which is characterized by pyriform. Pyriform means pear shape like this. Like one tip will be narrow, the other one is broad. Pear shape, nucleus is here. Okay. With two lateral means sides, and unequal flagella unequal. This is a unique feature of brown algae, my dear students. Brown algae. Volvox is green, first of all. Green. Eudorina, green. Porphyra, red. So, green algae shows spores like this. Apical flagella 2 to 4. 2 to 8. So, 2, 3, 4, 5, 6, 7, 8, anything. This is in green algae. I repeat, top 50 topics I have done. If you go through that, you will come to know what should you pay attention to. In plant kingdom, most of the questions have come on algae part only. I am not saying other topic questions will not come at all, but be thorough. The chances are high. Probability of them asking questions from here is high. Select the incorrect statement with respect to bryophyte. Bryophyte. Commonly grow in moist, shaded places. They are dependent on water for sexual reproduction. Yes, that is why we call them as amphibians. Amphibians of plant kingdom. They lack true root, stem and leaf. Zygote undergoes reduction division. Wrong. This happens in algae, not in bryophytes. The zygote will undergo meiosis and directly produce spore. Here, the zygote undergoes mitotic division and produces sporophyte. Okay, main plant body of bryophyte is, what is the main plant body of bryophyte? Funaria, Mercantia, you would have seen. What is this? This is a gametophyte. On this, the zygote is produced, which we cannot see, and the zygote then develops into a foot seta and capsule containing sporophyte. So, sporophyte is 2N and then gametophyte is N. Option B. Yes. Next question. One question on protonema we will do very quickly. Protonema is formed in mosses and liverworts. Protonema represents sporophytic stage of bryophytes. Protonema is found in mosses and liverworts. Not in liverworts. Mosses, yes. They represent the sporophytic stage? No. They are representing the gametophytic stage. So, they are haploid in nature. So, both the statements are incorrect. Both the statements are incorrect. Okay. Remember that. Liverwort has another unique structure, asexual reproductive structures of buds called as gemma or gemma cups. or gemma cups. Correct. Heterospory is not found in. Heterospory is not found in. You don't find heterospory in which of the following? Heterosporous nature is not found in. Yes. What is the answer? Remember, liverworts and mosses are homosporous. Algae is also homosporous, we say. Angiosperms, pteridophytes, few of the pteridophytes, and gymnosperms. All the gymnosperms, all the angiosperms are heterosporous. Pteridophytes, few are heterosporous like Selaginella and Salvinia. So, heterospory is not found in bryophytes. Remember, not found in bryophytes. Okay. Next, the giant redwood tree is the largest in the entire plant kingdom. Sequoia. This is the smallest plant, or rather smallest angiosperm. Option A. Yes, good. In gymnosperms, the male gametophyte is highly reduced and is known as. In gymnosperms, the male gametophyte is highly reduced and it is known as what? Done. Male gametophyte is highly reduced and correct. It is called as a pollen grain. Embryo sac. This is in embryo sac word is in angiosperms and it is called female gametophyte. In gymnosperms, you have archegonia inside the ovule. Okay. Inside the ovule, you have archegonia when it comes to gymnosperms. So, option D would be the answer in this case. Yes. Let's go to the next question. And this is the question. Select the incorrect match. Yes. First embryophytes, bryophyte. First time embryo in the entire plant kingdom. First time you will see vascular tissue in pteridophytes. That's why they are called as tracheophyte. Archegoniate spermatophyte. They produce archegonia also and seed also. Gymnosperms. Yes. They are seed plants without ovary. Angiosperm is wrong. They have plants with, they have ovary and they have fruit production also. So, this would be the answer. This is the incorrect match. Ovary is there. Yes or no? Okay. So, I think, yeah. Shall we go to the next one? Let us go to the classification system, biological classification, that is. Done? R.H. Whitaker's classification is or are based upon what? All of this. He considered the cell structure, that is eukaryotic or prokaryotic. He took into consideration unicellular and multicellular nature, Mode of nutrition, heterotrophic, autotrophic, he did consider that. The type of reproduction that the organism shows, an evolutionary relationship or phylogenetic relationship. Okay. Based on life cycle questions, based on life cycle, even if they do ask, it will be life cycle in the sense, I did one question, no, where the zygote undergoes directly reductional division or not. That kind of question they ask. Like they will not give you a life cycle and then ask you to identify something. Life cycle based questions are asked like after gametophyte, a sporophyte is formed, in the sporophyte spores are formed. That way questions are asked, which we covered. Done? Easy. A is coccus, B is rod shaped, C is spiral and D is comma shaped vibrio. So the answer is option B. Easy question. This is easy. In this also 37% have marked wrong. Why? Respiration chapter I did. Column 1 and column 2, halophiles, thermoacidophiles, methanogens. Halophiles, thermoacidophiles and methanogens. Generally when archaebacteria they ask in terms of habitat only most often, or methanogens found in ruminants, the gut of ruminant, that way they ask. Okay, so halophiles would be salty area, thermoacidophile hot water springs, methanogens marshy area. So B, C and A. Option C. Correct? Yes. Next, archaebacteria differ from other bacteria in having, archaebacteria differ from other bacteria in having what? They differ in the cell wall structure. Yes or no? Yes. That is why they are able to tolerate a highly hostile condition. Chief producers in ocean are pseudopeptidoglycan, you can say something similar to peptidoglycan. Chief producers in ocean are what? Easy. Chief producers in ocean are diatoms, not even desmids, but diatoms. Correct. Yes. Good. Look at this. Try to read this. Instead of a cell wall, they have a protein-rich pellicle. The moment they say pellicle, you should mark the answer. The answer is euglena. Mixotrophic nutrition, 2 flagella, 1 long, 1 short. They are heterotrophic in the absence of light. In the light, they are photosynthetic. The connecting link between plants and animals. So, option D would be the answer. Correct? Yes. The moment they say pellicle, it is euglena. It's a very unique feature of it. Let us look at this. Which of the following statement is correct about slime mold? Its thalloid body, plasmodium, has pseudopodia for locomotion and engulfing organic matter. During unfavorable condition, Plasmodium differentiates and produces fruiting body sporangium. Spores possess no true cell wall, wrong. They have cell wall, cellulose. They are dispersed by air current, yes. Being extremely resistant, the spores survive for many years and Plasmodium can grow up to several feet. Correct? So 1, 2, 4, 5, 6. 1, 2, 4, 5, 6. Option A would be the answer. Yes, option A would be the answer. Paramecium. Paramecium. What is paramecium? What feature does it show? Paramecium is a ciliated protozoan, shows water current movement by cilia which helps the food to be stirred into gullet, has a cavity gullet that opens into the outside by outside of the cell surface. Then all of these are correct. Comes under protozoa of protista, that is ciliata group. Correct? Bacteria as a group show most extensive metabolic diversity. Yes. And they have a very simple behavior but complex structure. This is ulta. This should have come here and this should have gone here. They have complex behavior but simple structure. So this is wrong. A is correct. Option A is correct. Okay, option A is correct. NCERT statement directly. Diseases, in case they ask, they can ask. You should be aware of this. Vibrio cholerae. Vibrio cholerae causes what? Cholera. Salmonella typhi, of course, typhoid. Clostridium tetani, tetanus. Xanthomonas citri, citrus canker. So 3, 4, 1, 2. 3, 4, 1 and 2. Option C would be the answer. Confusion in diseases. All the names are giving away the name of the diseases it causes. Which of the following is a fungi which is parasite on mustard plant? It causes something called as white spots on mustard leaves. What is the answer? Albugo. Correct? Albugo. Comes under Phycomycota. Important. All the three in fact come under Phycomycota and Aspergillus comes under Ascomycota. Ascomycota. All are the given features which are common between the members of Ascomycota and Deuteromycota except production of sexual spores. Deuteromycota are called fungi imperfecti. Why? Because they don't show any sexual stage. Rest of all of the things are common between Ascomycota and Deuteromycota. Okay? Unknown. For that, the class will get extended, and for anatomy part, I'm really sorry. In this session, I will not be able to do it. Okay? Because see, by 2:15 it has to end the session. So that you can start with class 12th. There was a series, I think Umid series, which was ran on PW English. In that you can check out anatomy of flowering plants if you want. You can also adjust the speed of it, right? Like at what 1.25x if you want to, 1.5x, whatever. So that you can refer there, it will help you. All are sexual spores of fungi except which of the following is not a, this is an asexual spore, very important. Remember that it is motile, that is why they have put a prefix as zoo, and it is endogenous, it is produced inside a sporangia. Remember that. Ascospore is also endogenous, Basidiospore is exogenous, oospore is also a sexual spore, that's the only information that they have given. So just remember that. Option B is the answer for this. Zoospore is the asexual spore, rest all of them are sexual spore. Asexual spore you have sporangiospore or aplanospore and even conidia for that matter. Even conidia gets included there. Members of Basidiomycota usually lack, what do they not have? Members of Basidiomycota, yes, lack what? Chitin in their cell wall, fruiting bodies, sex organs, long-lived dikaryophase. Dikaryophase means where the cells have two nuclei which do not want to fuse. They are kind of fighting with each other. Then like we are not going to fuse. The cell says, are you, cytoplasm is mixed, why are you not fusing? They say, we don't want to fuse. Who told you to fuse? They are asking the cell. The cell says, we have to continue the life cycle. Then you fuse. Let the cytoplasm mix, but we are not going to fuse. And they remain n plus n. So that entire phase, when they remain n plus n, they remain n plus n. Dikaryotic phase. Correct? Basidiomycota don't have a sex organ. They don't care. The hyphal cells directly fuse. They don't want to create any structure which will then produce a gamete and the gametes will fuse. Who has time for all of that? In the soil, two hyphae are growing. Both of them are compatible. They come in contact. They fuse directly. They fuse directly. The cells will fuse, but the nuclei keep fighting. They say, we are not going to fuse. Stay away from me, says one nucleus to the other. Let us come to this question. TMV is similar to HIV. TMV is similar to HIV. In having RNA as genetic material, as both lack capsid, as both infect humans. In having lipid envelope. Answer is, they both have RNA as genetic material. One is plant infecting virus, tobacco mosaic virus. The other one is human infecting virus. They both have capsid. Lipid envelope is there with HIV but not with TMV. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells in the members of. Just now I told you. Just now I told you. What happens? In which one of the higher fungi, a very common fungi that we eat at home, we enjoy it as a delicacy. Yes. What is the answer? Deuteromycota is out of question anyways because it does not show any kind of sexual reproduction. Then the answer is option A. The moment they say sex organs are absent, it is Basidiomycota. Remember that. Because, and of course Deuteromycota, but the ones that show sexual reproduction and still sex organs are absent is that. Red tide is caused by whom? Who shows red tide? Which organism is responsible for red tide? Dinoflagellate, and an example under dinoflagellate, goniolax. Option B. Very good. Very good. Clear? This is clear. Also go through prions once. Okay, very quickly go through prions once. Prions in the sense diseases that it causes, and also viroids. I didn't take those because they are like fairly straightforward questions that are asked. This also was straightforward, but I felt you were more confused here. Prions and viroids, you are not confused. Okay? Clear? Lucky, Mauni, whoever you are, I am not teaching. I am solving questions. Yes. Let's come to this question, anatomy of flowering plants. Bovine spongiform encephalopathy, Creutzfeldt-Jakob syndrome, prions. Yes. Let's come to this particular chapter, anatomy of flowering plant. For those of you who need a little bit of description, please watch the series that is there on the English channel. That will help you. Okay? Yes. Shape of guard cells in Graminae family. Graminae family is what? Monocot family. Then the shape is going to be dumbbell shape. There are only two shapes you have studied. Kidney shape is in dicots and of course dumbbell shape in, yes. Next question. Ring arrangement of vascular bundles is the characteristic of what? Done? Ring arrangement of vascular bundle is the characteristic of what? Dicot stem. The only one to show vascular bundles arranged in ring form is dicot stem. Okay? Yes. The next question. Types of vascular bundle in dicot root they are asking you. In dicot root. What is the answer? Done? Done? In dicot root, the moment they say root, you don't get a bundle here, correct? You get radial arrangement of the vascular tissue and the condition of the xylem is exarch. These two things will not change. So it is radial and exarch. Radial and endarch combo you have not studied anywhere. Conjoint and open, you have studied this only and only in dicot stem. Conjoint and closed will be in leaf and then in the monocot stem. Okay, option A. Good. Done? Find the correct match. Few more questions. By 2:15 we'll wrap it up. You take a break and then you can start with class 12th preparation. Okay? Don't be scared of the paper. Don't be too anxious. Don't be too nervous. Do your preparation. Have a very good sleep on May 3rd. And then on May 4th, write your exam. Once exams are done, don't worry about how it is done. It is gone, over. Forget it. Alright? Yes. Correct match. Monocot root, tetrarch xylem bundle. Monocot root, pith is small and inconspicuous. Dicot root, no. Monocot root is polyarch. So this is wrong. Oh, this is wrong. Dicot root, it should be pith is small and inconspicuous. Dicot root pericycle involves in vascular cambium formation. Yes, this is also wrong. Monocot root, this should have been dicot root. Dicot root. So the answer turns out to be option D. In dicot root, pericycle will involve in vascular cambium formation. Dorsiventral and isobilateral leaf are similar in. Yes, Shweta. Oh, incorrect. Correct match Deva. So this is correct, na? Yes, answer is option D. Dorsiventral and isobilateral leaf are similar in dorsy. Dorsy, D for dorsy, D for dicot. D for dorsy, D for dicot. Isobilateral is monocot. Type of vascular bundle. The answer is there. The type of vascular bundle is conjoint and closed, having large empty and colorless cells on adaxial surface. Adaxial means upper. They're talking about bulliform cell. Many a times they've asked question on this, okay? So remember this. And they're found only in monocots. Distribution of stomata also is different. In dicot, you have more number of stomata on the lower surface. In monocot, you have equal number. Differentiation of mesophyll tissue you will see in dicot, but not in monocot. Correct? Differentiation of mesophyll tissue, where do you see? You see this in dicot, palisade and spongy. Monocot, it is simply mesophyll. That's it. Hypostomatic leaf with stomata distributed more on the lower surface is an example of. Why? Okay. I want to finish the portion by 2:15. That's the reason I'm kind of focused here, so that all the concepts are covered. And I'm paying attention to that. That's why I'm not I didn't smile. Okay, hypostomatic leaf, when more number of leaves are present on the lower epidermis. Correct? Then, what kind of leaf would it be? It would be dorsiventral. D for dicot, D for dorsiventral. Yes or no? Yes. Bulliform cells are, where are they found? Where are they found? Sorry. Where are they found? Bulliform cells. They are found on the adaxial epidermis of the grass leaf. Adaxial means upper. They will try to confuse you. Abaxial is the lower epidermis. And in monocots only, along the veins also they have mentioned in your book. They are the ones which are filled with water. The screen is already bright. It is your mobiles or your screen's work. What were they saying? So they are filled with water. When the water is lost, they kind of shrink. How air goes out of balloon. How the balloon will shrink, the same way bulliform cells will also shrink. Okay. Ground tissue includes what and all? Yes. What and all is included in ground tissue? Ground tissue. Yes. Ground tissue will include everything other than epidermis and vascular tissue. End of it. Hypodermis, yes. Cortex, yes. Endodermis, yes. Pericycle, yes. Pith, medullary ray, you name it. All of those come under ground tissue. So, no epidermis and no vascular bundle. Major characteristic of the monocot root is the presence of. How will you identify monocot root under microscope? How will you identify it under microscope? Scattered vascular bundle, vascular tissue without cambium, no. Cambium sandwiched between phloem and xylem along the radius, no. Open vascular bundle, no. Then what is the answer? Monocot root you will find vascular tissue without any cambium. Correct? Yes. Scattered vascular bundle in monocot stem. Cambium sandwiched between phloem and xylem along the radius. Dicot stem, this is conjoined and open to secondary growth. Conjoined open to secondary growth. Open vascular bundle means cambium is present and that is seen only and only in dicot stem. Where are the Casparian strips? Casparian strip. The name Casparian is given because of the scientist who found it, Caspari. There was one character, na? Casper the Friendly Ghost. Yes. Casparian. Suberin deposition in the endodermis, and that too of both monocot and dicot roots. Option D. Yes. Correct. Polyarch condition of vascular bundle with a well-developed pith. Done? Polyarch. Polyarch. Only one and one chose, that is monocot root. Very good. Monocot stem, scattered word, dicot stem, ring-like arrangement and cambium is present. Dicot root, tetrarch. 2 to 4 we say, tetrarch condition. Yes, last question. Hypodermis is dash in sunflower stem. Last question in this chapter. Morphology is pending. Yeah, after morphology, 2:15 sharp, you will get a break. Hypodermis is dash in sunflower stem and dash in maize stem. Done? 𝐂𝐍𝐼𝐿 and sclerenchymatous. Remember, 𝐂𝐍𝐼𝐿 is a tissue which is found in the dicot shoot only. First of all, not found in monocot, and then even in dicot, in the petiole and in the stem, yes, but not in the root. Never. Never. Okay, remember that. Never. Sclerenchymatous, hypodermis in maize. The sunflower is of course dicot and this is monocot. So here you have sclerenchymatous, hypodermis. Done. Last chapter for today in class 11th, morphology of flowering plants. Done? Easy? Yes. Proximal to distal. So imagine if this is the root. This is a soil and this is a root which is growing proximal to the plant. Where is the plant? Proximal means near the plant. Okay, so above the soil. So proximal, when they say, then this is the proximal part, proximal to the plant above the soil, and this is the distal part. Okay, this is distal. Distal means away, far. This is proximal means near. Okay. So now proximal to distal, can you now arrange? Near to what? The plant. Far from what? The plant. Understood? Proximal would be zone of cell maturation, then elongation, then zone of cell division would be the farthest and the most away. Away. Oh. Yes. Leaderboard I'll show. Okay. I don't know why everybody is behind my smile today. Never in my life have been complimented and asked to smile so much. Nobody has ever told me, ma'am, your smile is good. Never. They've told me, ma'am, you're humble, you're sweet or whatever, but beauty lies in the eyes of the beholder. Yes. Asymmetric flowers, the flower that cannot be cut into two. Examples, morphology of flowering plants, I cannot stress the examples. How important. All of a sudden, one fine year, NTA decided that no, morphology of flowering plants has to be very important in class 11. So actually from here, 3, 4, in one of the years, 5 questions they had asked. 5 questions they had asked. Then they came to their senses, I suppose, and then now 3 questions they ask. Okay. Done. Asymmetric flower. Canna is the answer. Bean and pea would be zygomorphic, that is bilateral symmetry. Datura would be actinomorphic, that is radial symmetry. Option D would be the answer. Correct. Reduced leaf found at the pedicel is known as. Found at the pedicel. What? You also time traveled. Okay. Reduced leaf found at the base of the pedicel. I thought you were asking a doubt and I read it. Okay. This is a leaf. At the base, you will find sometimes, not always, a leaf like small chotusa structure which is called as stipule. If it is at the base of the flower, it will be called as bract. It will be called as bract. If there is no stipule, you call it as ex-stipulate. Correct? Will you match and keep this and then we will come to the correct options, or simply match and keep it? Who cares? Hypogynous, epigynous, perigynous. Hypo, below, gynous, gynoecium. Who is below the gynoecium? Calyx, corolla, and rhizium. Epigynous, epi, above, gynous, gynoecium. Who's above the gynoecium? Or the ovary rather in this case? Calyx, corolla, and rhizium. Perigynous, around the gynoecium or the ovary, half inferior or half superior. So in hypogynous, gynoecium occupies the highest position. Gynoecium is situated at the center in perigynous. Margin of the thalamus grows upwards, enclosing the entire ovary, that is in epigynous. Mustard, brinjal, china rose are hypogynous. Peach, plum, rose, perigynous. And guava, cucumber, ray floret of sunflower, very important. I cannot stress the importance of this, is this. Okay, understood? Clear? Done? Yes. Over? So you can then match from this. I think we are done with this. So this doesn't matter much because anyways I have not put the, yeah. With 1 A and X will go. With the 2 B and, no, C and Z will go, correct? Yeah, C and Z will go. Option B would be the answer. Okay, yeah. Which flower possesses superior ovary? Come on, now, superior ovary, superior, hypogynous condition. Hypogynous condition, all of these would be the answer. Just now we did this. Entire concept we finished. Come on, guys. Mustard, china rose, brinjal, all of them are hypogynous. Okay. Let's come to estivation, which is false for vexillary or papilionaceous estivation. Vexillary estivation is how? You have standard, then you have the sides, wings, then you have keel, and they are fused. This is anterior, that is posterior. This is anterior, that is posterior. Okay. So, false, they are asking you, this is anterior, this is a head, keel is the head. Like in our body, this is anterior and the legs are posterior, the same way. The head is anterior, this is posterior. Okay. Two smallest anterior petals are known as keel, two lateral petals are known as wings. The largest standard petal is posterior, largest standard petal is, nope. Answer is option D. Yes, good job. Aestivation, like I said, very, very, very important. Aestivation, valvate, twisted, imbricate, vexillary. Correct? Okay. Valvate is cassia. Valvate is calotropis. So, this is wrong. Cassia should have been imbricate. Twisted is, yes, China rose, lady's finger, and calotropis is wrong. This should have been valvate. Cassia and gulmohar are, this is right. So, this would be the answer. Vexillary is P and bean, okay, but not aloe and tulip. That is wrong. So, answer is option, I didn't put a poll, sorry. Answer is option C. Let's go to placentation. Very important, asked many a times. Very important, placentation example. Done. Marginal placentation in P, parietal would be in mustard and argemone, axile would be in China rose, tomato, lemon, central and free central in dianthus and primrose, basal placentation in sunflower and marigold. 1, 2, 4, 3, 5. 1, 2, 4, 3, 5. Option A. Yes. Next question. Given is a floral formula. Okay. I don't know if you are able to see this. Let me re-put it here. Yes. They have given you this floral formula and they are asking you to identify. Yeah. To which of the examples does it belong to? Floral formula belongs to which of the following? Done? Done. Allium is actually belonging to a family called Liliaceae. Allium is nothing but onion. Sesbania is a relative of Fabaceae family. It's a part of Fabaceae family. Petunia is the answer here because it belongs to Solanaceae, ornamental plant called as Petunia. Brassicaceae is different. We will do a question on that. Brassicaceae is different. See, Solanaceae, how to identify? I'll tell you how to identify. See here, main thing is this. Main thing is this. This line indicates epipetalous stamen. That means the stamens are attached to the petals, corolla. That is the key which will give away the Solanaceae family to you. Yes, correct. For Brassicaceae, now we come to this. Are you able to see the options properly? This is minus here, this is plus, plus, minus, plus, 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 plus again. I hope you're able to identify here. Now you tell me Bressicaceae formula. Yes. Done? Yes. What is the answer? Option B. Very good. What is wrong here? 2 minus 2. What is wrong here? 2 minus 4. What is wrong here? They've put a bracket here. This is wrong. Okay, there's no bracket. Bressicaceae mustard family. How many leaves are present at the node in opposite phyllotaxy? In opposite phyllotaxy, how many nodes are at nodes? How many leaves are present? Yes, tell me. This is Calotropis example. Guava is another example for opposite phyllotaxy. So you have two, right? One would be in alternate phyllotaxy. For alternate, you can take example of hibiscus, even sunflower for that matter. That can be also an example. Many will be for world phyllotaxy, correct? Alstonia and Nerium is another example. Alstonia and Nerium. I think Nerium is not given in your book. So yeah, stick to Alstonia. 2. How 2? 2. Correct. Correct. 2. Okay. Very important. Diagram based question. Very important. Economic importance of Solanaceae. Very important. That also is important. Yes. They have given you the diagram. Identify, they have told. Saw it? Now, A is hypogynous, B is perigynous, C is also perigynous, and D is epigynous as given in your NCERT. But they need not give it in order. They can give B first and then they can give this diagram first and then the next one. Don't think they will give it in order. So even if they have given, such a diagram based question, properly see it. Don't be under an impression that they will give you in order and you are able to answer very easily. Not always. Not always. Okay. Be very careful. Be very careful when attempting such questions. Estivation A, B, C, D. Peri, epi or hypogynous? Yes, I will tell you. We'll just do this and then I'll come to that. Okay, the first estivation, I am not going to tell anything here. See, pay attention to the diagram. Look at the diagram, accordingly match it. Look at the diagram, accordingly match it. Don't be in a hurry. The order given in your book is different. They have given valvate first, then twisted, then imbricate, then vexillary. Here they have changed it. Only vexillary's position is as in NCERT. So, A is twisted, B is imbricate, C is valvate, D is vexillary. Option D. Correct? That's what I said. What did you do? Same order as NCERT you marked. See there. Just now I told you, they need not give it to you in order. Just now I told you and you did the mistake. How to identify? I'll tell you how to identify here. See here. Pay attention to thalamus. I'll just outline it. This is thalamus. See here. Thalamus is here. Hypogynous. Here the thalamus reaches to a certain length. This is perigynous. Okay. See, the thalamus is like this, cup shape. The red outline one is thalamus. This is also perigynous. Okay. This is also thalamus. This is thalamus. This is epigynous. So here actually they have shown you a kind of a section. Here also of course thalamus will be there. They have shown you a section. So it looks like a half cut to you. Look at the thalamus. Thalamus. Okay, position of ovary also can help you, but thalamus would be the best thing to identify. Or you can pay attention to how ovary is. But the problem with ovary, unless and until you don't understand where thalamus is, ovary's position you will not be able to understand. Now you can say ovary is above. Here ovary is partly covered, ovary is partly covered, ovary is completely covered. Okay? Yes. Correct. Placentation, very important. Very important. Diagram they have given and asked you to identify the placentation. I cannot stress how important it is. We have done only one here. You should know the diagram of all. I will draw it. The best way to remember and learn. Draw it, even if it is rough, but draw. Axile they have drawn. So the answer is axile. For marginal, what is the example? Pea plant. Fabaceae, leguminous, if you take. For axile, we did examples we have completed already in one of the questions. China rose, tomato, lemon, correct. For parietal, how does the diagram look like? Brassicaceae family, argemone, mustard and all is parietal. Cucumber is parietal. And in Brassicaceae, you will get a false septum because of the placenta being extended inward. Okay. Basal placentation, how does it look? It looks like this. Then free central, basal placentation is sunflower, marigold. Free central is like this. This is primrose, dianthus. Okay, is an example. Yes. One more thing you remember, in Brassicaceae, a false septum called replum arises. Axial placentation it will show. Imagine this is the ovary. This is a placenta. The placenta comes inward so much, it looks like there is a false septum. It looks like the ovary has two locules. It looks like two chambers. Why? Placenta has grown inward so much. So this false septum is called as replum. Called as replum actually. But remember false septum, that's enough. I suddenly remembered that point. So this is very important. Please remember the examples. Non-NCERT family. I'll apply a simple logic. As long as a portion is not there in NCERT, how can anybody ask questions? At least old NCERT, it should be there as per me. Even then you should not ask. When CBSE has released a new textbook on their website, they've removed all the old textbook and there is no information. How can they ask questions? They should not. It is unfair to students who don't have any means of studying in such coaching, or maybe the schools are not catering and they're using new textbooks, right? So I would suggest something which was not part of old NCERT and is not part of current NCERT, don't prepare that. New families, I did it last year. Actually, the whole session, whatever we did, I've done it last year. I've uploaded notes and then it made me realize that only because students were asking. But I said even then, no question will be asked from that particular part. So don't worry about it. Last year there was no question asked. The year before that there was no question asked. So don't worry. The stony part of the mango represents what? Endocarp. Mango is a drupe along with coconut, and drupe develops from a monocarpellary hypogynous flower. Correct? Mango, and then you have coconut. In coconut, the mesocarp is fibrous, and mango, the mesocarp is juicy and edible, we say. Option D. The name of the fruit is drupe. Probably, yeah. The last question for today, floral formula, diagram they have given, sorry, and they are asking you to identify the floral formula. Will you be able to? Will you be able to? Pay attention. Very important. Minor differences will be there, which you should not overlook. So be calm. Be calm. Done? Oblique septa, swollen placenta. See here, this part is swollen. Oblique means this is anterior posterior axis. The septa is diagonal to that, like, you know, slanted towards. If I consider this should to be the anterior posterior axis, the placenta, the septa is like this. That's why we say oblique septa. Okay. And one more point I am adding, persistent calyx. Calyx is seen even in fruit formation. Even in fruit formation, calyx is there. Normally, calyx falls off, but here it is not. The answer is option D. Option D. Very good. What is wrong with option B? C is not in bracket. They should have put a bracket. Gammocephalous condition. Okay, gammocephalous condition. So that would be our last question. This is Solanaceae family here for you. Yes. Last question for today, and that's a wrap of class 11th. Solanaceae is there. Solanaceae is part of your syllabus. Thank you and all the best. All the best, guys. Be calm in examination hall. Do not be anxious. The moment you feel your heartbeat is increasing, take a deep breath and drink some water. I'm talking about examination hall. I'm talking about examination hall. That's your leaderboard. Thank you, everybody. All the best. Do well. I hope all of you score well. Get the college that you want and the ones that you're aspiring. Yes? Okay. This is your leaderboard. Take your break. Get ready for the second half of the session. Yes. Bye-bye.
I'm back. Is everyone back? Hello, hello. Welcome to PW English. Is everybody ready? Hi, Yadav. Hi, Ramya. Good afternoon. How are you doing? Hi, Moni. Sunidhi. Lakshita. Okay. Hi, Arav. Okay, so until a few minutes ago, this marathon was conducted by Chaitra ma'am. She has discussed questions from the 11th grade syllabus. I hope all of you attended and you've figured out where you stand in your revision and which concepts you were weak in, which one you have to revise and go back and study and all of that, right? So we'll start with 12th grade syllabus. Is everybody prepared? Hi Praveen. Kanika. No friendships here in the chat box. Everybody is here to learn. You don't want to know anybody's age here. What is your Instagram handle? None of that will be tolerated in the chat box. We're going to be discussing questions and solely focus on the revision for the upcoming NEET exam without any distractions. All right? Arav is asking, Kevin kahan hai? Ghar pe hoga shayad. He must be preparing. Okay, hi Kashish. Good afternoon. Okay, so we will turn on the poll. For those of you who have just joined who have no idea how the poll works, I will be displaying questions on the screen and you just have to identify which is the correct answer and type the options in the comment box. Okay? Everybody ready? I want to see thumbs up in the comments. We'll get started. You might already be aware, but these are the chapters that come under botany in 12th grade syllabus. We're going to start with sexual reproduction in flowering plants, principles of inheritance and variation, molecular basis of inheritance, microbes, organisms, population, ecosystem, biodiversity, conservation. Okay? Okay, Moni, thank you very much. All right. Let's get started with the first chapter. I'm going to go chapter wise and just one piece of advice before we start. Please pay 100% attention. Do not get distracted. Approach each question as you would in the actual NEET exam. I want you to practice reading the questions carefully, reading all of the options before you type the correct answer in the chat box. Instantly it might seem like option A is correct. You might overlook options B, C and D. Don't make that mistake. This is a preparation before your NEET exam. So read all the options carefully. Doesn't matter if you're not the first person to answer the question, but as long as you've answered it correctly, it's fine. Okay? Please pay 100% attention. Mute all other app distracting notifications and let's get started. Okay? So here is our first question for today. I'm going to give you 30 seconds for most questions unless it's really lengthy. And I generally read the questions myself. This time I will not read it. I want you to read the question carefully and answer it. Only then I will read it. मुझे हिंदी नहीं आता, बिलाल रहमान. नहीं आता, आती, I don't know even if that is correct or not. Okay. The word 𝐑𝐄𝐍𝐋 refers to option A, occurrence of two bundles of stamens in a flower, two lobed anthers of a flower, occurrence of stamens of two different lengths, anther of two types in a flower. Actually, what 𝐑𝐄𝐍𝐋 means is in each lobe there are two theca or two chambers, right? That is what two theca 𝐑𝐄𝐍𝐋 means. So, most of you have answered it as option B. Option B seems like the most appropriate option here, but it is because each lobe has two theca. Okay? Two theca in each lobe of an anther. You've just finished morphology of flowering plants as well. Whenever the stamens are in two bundles, we use a technical term. What do we call it? When the stamens are in two bundles, what do we call it? What do we call that term? You see that in Fabaceae family, there's a total of 10 stamens, of which 9 unite to form one bundle and there's one separately. What do you call that condition? Yes, Divya is saying diadelphys. That is the correct answer. And in morphology, you also studied how in certain plants the length of stamens vary. There will be few short stamens, few long stamens. Does anybody remember example for that? At least one example where the stamens vary in length from morphology in flowering plants? Morphology of flowering plants. Bilal Rahman, this is the maximum speed I can talk in. I don't have a motor in my mouth. Okay, I am human. So, salvia and mustard, good job. Okay, so the correct answer here is option B. Good job, most of you answered correctly. Moni, Khushi and Sunidhi, top 3. Let's take a look at this next question. Cells of tapetum generally have dense cytoplasm and more than one nucleus, dense cytoplasm and single nucleus, dense cytoplasm, no nucleus, vacuolated cytoplasm and a single nucleus. Correct answer, like most of you are saying, option A, dense cytoplasm and generally it has more than one nucleus. Can somebody let me know in the comments what the function of tapetum is? Moni, Bilal, Yadav, top 3. What is the function of tapetum? Nourishment. What does it nourish? It nourishes the developing microspores. What are the 4 layers in the wall of the anther, outside to inside? Epidermis, endothecium, middle layers, tapetum. Tapetum is closest to the sporogenous tissue which will in the future become pollen grains. So as the sporogenous tissue develops and matures to become the pollen grains, it derives nourishment from the tapetum which is lying very close to it, right? Okay. Now, here is our next question. I want you to please read the question carefully. Just don't copy what one person says. Please read the question carefully. What is microsporogenesis? It's a process. Microsporo relates to microspore. Genesis relates to generation or production. Generation. The process by which microspores are produced is called microsporogenesis. Most of you are saying it's option D. In option D, they've given formation of microspore mother cells and not microspores. From microspore mother cells, microspores forming, that is microsporogenesis, right? So, from the diploid microspore mother cells, meiosis will happen, and then haploid microspores are formed. They are formed together in a tetrad. Then they get dehydrated, they separate, and then pollen grains are formed. So the most appropriate option here would be option B and not option D. The formation of pollen grains is correct. Very few of you have answered that question correctly. Suhas, Darshan, Preeti, good job. You did not get confused. Top 3. Hi Amar, good afternoon. How are you? I'm okay. Just please pay attention. Sometimes I've seen a lot of you make mistakes because you don't read the question properly. There could be 1 or 2 students who are attending this who are regularly in the top 3 leaderboard. So if one of them answers one option is correct, you assume that probably it's correct and you all start typing the same thing. Please use your own brain. Don't go follow people like sheep. Use your brain, read the question completely and only then answer questions. Okay? Hello Anshuman, good afternoon, welcome to the class. Here is our next question. Please read carefully and answer. Which among the following is a wrong match? Every time you come across a question which has correct statement, incorrect statement, mismatch, correct match, please underline it when you're doing it in a physical copy. When you're answering questions online like that, please take like a mental, put a mental dabba around it. So you are very clear about what you should look for in the options, okay? So here we have in option A microsporophyll stamen, microsporangium anther, megasporophyll carpel, megasporangium ovary. Okay. We are required to identify which is not correct. What is not correct here is option D. See, when flowers form on plants, different leaves get modified and take up different functions and become the plant. One set of leaves at one node will become the calyx, another set of leaves will contribute to become the petals, another whorl of leaves will contribute to become the stamen. Generally in lower plants what happens, what is microsporophyll? Microsporophyll is a leaf that bears microsporangia. In flowers what bears microsporangia? It is the stamens that bear microsporangia. So stamens are equivalent to microsporophylls. What is a microsporangium? Sporangium is a structure within which spores are formed. Microsporangium is a structure within which microspores will form. Where exactly do microspores form? In a flower, it forms within the anther, right? Within the anther is where the 4 microsporangia are located. So that also pretty much is correct. Megasporophyll is carpel. What is megasporophyll? Megasporophyll, you've learnt about it in pteridophytes and gymnosperms. They are specialized leaves which will bear megasporangia. So where is the megasporangia? In a flower, it is the carpel, right? The pistils is what is megasporangia. That is where the megasporangium ovule is present. So here they've given megasporangium is ovary, megasporangium is not the ovary, megasporangium is a structure within which megaspores are formed. Now where exactly do megaspores form? They form inside the ovule. So technically megasporangium is equivalent not to the ovary but to the ovule. So option D is the incorrect match here. Very few of you have answered correctly. Darshan, only one student. Good job, Darshan. I hope it is clear now for all of you who answered it as A, B, C and all incorrect answers. Here is our next question. It is an assertion and reason type of question. Please read the question carefully and answer. Don't follow what others do. Yashoda, KCET is already over. How will you write KCET now? You can't. KCET happens only once a year and it's already over. Okay, so here is an assertion and reason type of question. A lot of you struggle with assertion and reason type of questions. First of all, we need to get over the fear and just understand that assertion and reason type of questions are true or false questions that are slightly more complicated. We will have two statements. Instead of immediately linking the two statements by ourselves, we should first take each statement independently, analyze both the statements, find out whether it is true or false. For as long as one statement is true, one statement is false, the question will be very easy. The complication arises only when both the statements are true. Then you have to figure out whether the reason correctly explains the assertion or the reason does not explain the assertion. So that is what, then what we do is we add a because between the assertion and reason and find out if that standalone statement will make sense. Okay, so let's do the same thing right now. We'll look at the assertion statement, find out true or false, reason statement, find out whether it's true or false. So here assertion statement reads, pollen grains are well preserved as fossils. Is it true that you can preserve pollen grains very well as fossils? Are they very well preserved as fossils? Yes, it is true. What is the reason? Pollen grains contain sporopollenin in their wall. Is that the reason? Is that a true statement? Forget whether it is the reason for this or not. Look at the reason statement as a standalone statement and find out whether that statement is true or false. Pollen grains contain sporopollenin in their wall. Is it true? Yes, it is true. Sporopollenin is one of the most resistant biological substances present on our planet, and it is present in the exine of pollen grains. So this also is true. Now we have to find out whether this is the correct answer or this is the correct answer. So we will add because here and read the question. Pollen grains are well preserved as fossils because pollen grains contain sporopollenin in their wall. Is that true? Yes, it is true. So option both A, assertion and reason are correct, and reason is the reason is the correct explanation for A. This will be our correct option. Another mistake that most of you make in assertion and reason kind of questions is you will assume that this is the first option and this is the second option, because in most cases as an assertion and reason kind of questions, assertion first option will be both assertion and reason are true and reason is the correct explanation. Generally the second option will be both assertion and reason are true and reason does not correctly explain the assertion. So don't already assume that in the same order even in your paper assertion and reason options will be given. Last year's NEET we saw that these options were on top, these options were below. So please read the question carefully before you answer. Okay, every question matters. You know how crazy the competition is in NEET. So please read questions carefully and correctly. So the correct answer here is option B. See, most of you may have assumed that this is the first option and in a hurry you may have made this mistake. This is the worst kind of mistake to make in your paper. It's not like you don't know the concept, guessed it and got it incorrect. You knew the concept very well, you made a silly mistake of not reading the options properly, and if you lose out on marks like this, it is very, very sad. Then we have Bilal, Khushi and Moni, top 3. Next question. Pay attention, read carefully. I will give you 45 full seconds. Okay, Yashoda is asking if you have to write both NEET and KCET. For those of you who don't know, KCET is Karnataka State Common Entrance Test. That is for engineering and veterinary sciences. But if you want to get a medical college in in Karnataka, and if you have to get through the counseling process, you have to at least register for KCET even if you don't write it. If you write it, well and good. But registration is mandatory if you want through KCET to get into medical colleges in Karnataka. Okay. So, what is the first thing we are going to do with this question? It is a true or false kind of question. Select the correct statement is what they've asked us to do. So, we will look for which one is true over here. In over 60% of angiosperms, pollen grains are shed at 2-cell stage. It is correct. This is a statement right out of NCERT. We've already arrived at the correct answer, but just for Safety, we'll go through the other options. Generative cell of pollen grain has more abundant stored food than vegetative cell. This is false. They've ultified it. Vegetative cell divides by mitosis inside the pollen tube and gives rise to the male gametes. This is also false. It is a generative cell that gives rise to the male gametes. Exine is continuous outer covering of the pollen grain. It of course is the outer covering, but it's not a continuous layer. It is discontinuous. It has germ pores. So the correct option is A. Good job. Darshan, Shahana, Yadav, top 3. See, sometimes what happens is, when you're answering these kind of questions, the first option might seem true and you might mark it correctly. But sometimes we may have realized that the first statement is true for the instinctively. But if you go through the other options, sometimes you will come across more than one correct option. Then you will realize maybe the first option was incorrect. You misread it, or you read it in a hurry, or you misunderstood that statement. So always read all of the options. Even though the first option itself seems like the most appropriate correct answer, you've arrived at it right away, please still read all the 4 options before you answer the question. Okay? Now let's move on to our next question. 30 seconds will suffice. Alright, so what are we going to do first thing when we read this question? With respect to angiosperms, which of the following is incorrectly tagged? We have to find out which is wrong here. With respect to its chromosome number, with respect to its ploidy basically, megaspore diploid, microspore haploid, primary endosperm nucleus triploid, egg is haploid. Here what is incorrect is option A. Megaspore is not diploid, it is haploid. All spores and gametophytes are haploid. Primary endosperm nucleus, primary endosperm cell, endosperm, this is triploid. Everything else is diploid. Okay? Rahul, Preeti, Ajay, top 3. Hi Ajay, good afternoon, how are you? How is preparation going on? Okay, here is our next question. When you read this question, you have to put a box mentally around something. Read the question carefully. Which among the following is a false statement? Option A. Lemurs, arboreal rodents, gecko lizard, garden lizard have also been reported as pollinators. This is a correct statement. It is straight out of NCERT. Grouping of small flowers into inflorescence is an adaptation for insect pollination. Okay. Because even in grasses you see that. Let's take a look at the other options. Flowers pollinated by flies and beetles secrete foul odors, unpleasant odors. True. Yucca flowers are pollinated by bees and butterflies. This is outrightly incorrect. It is pollinated by a specific species of moth, which is also called the yucca moth. So this is also true. Whenever the flowers are very small in size, in order to attract the pollinators, which could be insects or birds or any biotic pollinator, it will aggregate and form an inflorescence so that it's more, it can attract the pollinators better. Okay, so what is incorrect is what we were required to find out here. So option D is false. That is the correct answer. Not bad. Danish, Darshan, Khushi, top 3. Next question. Please read carefully and answer. Hi, Tenjin na Tengin na ಏನು ಹೆಸರು ನಿಂದು? Which among the following is not an outbreeding device developed by plants? Which is not an outbreeding device? Pollen release and stigma receptivity is not synchronized. That is an outbreeding device. Anther and stigma are placed at different positions. That is also an outbreeding device. Developing self-incompatibility, which is a genetic mechanism, that also is an outbreeding device. What are outbreeding devices basically? They are devices or mechanisms that flowering plants have evolved which will help discourage self-pollination and encourage cross-pollination. So to make sure cross-pollination happens and inbreeding depression can be avoided, prevented, these are the things that different species of plants have come up with over the course of evolution. Option D, developing 𝐒𝐍𝐆𝐋𝐈𝐒𝐇 flowers. 𝐒𝐍𝐶 flowers are flowers that are closed forever. They don't bloom at all. If the flowers don't bloom at all, then there's no way cross-pollination or outbreeding can happen there. So this is not an outbreeding device. Correct answer is option D. Chaitanya, Darshan, Danish, top 3. Next question has many statements, so let me give you 45 seconds. Read carefully and answer. Okay, so a lot of you are saying 3. Oh my god. How many of the following are wrong statements is what they've asked. Castor and maize prevent autogamy but not geitonogamy is true, right? Castor and maize, you have flowers, plants that are monoecious, but it will produce unisexual flowers. So autogamy is not possible because you have separate male and separate female flowers. But geitonogamy is still possible. That makes it a true statement. Second, papaya prevents both autogamy and geitonogamy. This is also true because papaya plant is dioecious. When you have a separate male plant and a separate female plant, it's obvious that autogamy cannot happen, geitonogamy also cannot happen, only xenogamy can happen. That is also true. Next, underground flowers in Camelina prevent geitonogamy and allogamy. Allogamy is another name for xenogamy. So what are these underground flowers? They are the 𝐒𝐍𝐿 flowers that are produced below the surface of the soil in Camelina. So here they do prevent geitonogamy and allogamy. How is geitonogamy or allogamy supposed to happen when the flowers don't open at all? They are underground, they do not open, they are 𝐒𝐍𝐿. So there is no way geitonogamy or allogamy can take place. These 𝐒𝐍𝐿 flowers are invariably autogamous, which means only self-pollination can take place in them. So this is also true. Viola, oxalis, camelina produce 2 different types of flowers, chasmogamous flowers and 𝐒𝐍𝐿 flowers, right? So this is also true. There is no false statement. There is absolutely no incorrect statement here. So what should the correct answer be? The correct answer would be option A. There are no wrong statements at all. It's disappointing how I don't think even one person correctly answered. Okay, there's one person probably. Danish. Okay, good job, Danish. I'm proud of you. And a lot of you thought 2 and 3. How could you even think 2 and 3 statements were incorrect? Like, this is the first chapter in your 12th grade syllabus, and the questions related to pollination are so frequently asked. One mandatory question you will have from pollination, and you can't not know these facts. Next question. There's a lot of you typing B in the comments. There is some C. Okay. So the question is, when you're performing artificial hybridization, when do you perform emasculation? Emasculation is to be done if the female parent plant selected. When do you perform emasculation in a plant? See, if the plant is already female only, if the flower is already female, if it is unisexual, it has only carpel, it does not have stamen, there's nothing for you to cut there, right? What is emasculation? Emasculation is removing the stamens, right? So if there is no stamen at all in the flower, there's no way you can emasculate it. The plant is already unisexual, you can't perform emasculation on a female flower. So when is emasculation necessary? Emasculation is necessary only when the flower is bisexual. It also has androecium, it also has gynoecium. So you want to designate it as the female plant. So that is when you will remove the maleness out of it. You will remove the anthers out of it. So if it is already unisexual, that is not a requirement, because all it already has is just the female reproductive part, right? So whenever it has unisexual flowers, it's not necessary. Only when there are bisexual flowers, it's required. So option B is the right answer, like most of you said. 91% of you all have answered correctly. Priya, Lucky, Danish are on top of the list. Here is our next question. 30 seconds should suffice. Hi Adesh, good afternoon. So suspensor is a part of. There is no mention of suspensor anywhere in the text of your NCERT textbook, but it is mentioned in as a label in the embryo diagram. You've learnt about how embryo develops from the zygote. You have the zygote, the proembryo, globular embryo, heart-shaped embryo, mature embryo. So over there is a label called suspensor. What is suspensor? Basically, for example, if you consider like this globular embryo, you have this bottom part, right? This is the embryo proper, and then you have something like this that is labeled as suspensor. What does this suspensor do? Suspensor penetrates into the endosperm and makes sure the nourishment is absorbed and transferred to the embryo proper, which is going to become the future plant. So suspensor is part of the embryo. Option B is the correct answer. If you've seen the diagrams in NCERT properly and learnt the labels, you would answer it easily. Priya, Danish, Preeti, top 3. Here is our next question. What you are thinking about NEET 2025 paper difficulty level? Reply. I actually have no idea. But what I can say is, compared to last year, this year can be slightly difficult because you don't have options this year. All 45 questions, there's going to be only 45 questions. So unlike last year where there were 50 questions, so that way you'll have lesser number of questions and no options to choose from. So that way that it will be more difficult. And otherwise, I suppose it will be as it was over the last few years, most questions from NCERT, nothing from outside. And also difficulty of a paper depends on one's preparation. It's very subjective. If somebody who's prepared really well takes an exam, even the most difficult paper can seem very easy. And somebody who's not prepared at all, for them even the easiest paper can seem difficult. So it's a subjective thing. Yeah, it depends on your preparation. And preparation not just with respect to concept clarity, how well you can manage time, how quickly you can read a question correctly, all of that, all of those skills matter. In which of the following groups all the three show complete utilization of endosperm before seed maturation? Basically, they're asking about which of the following are non-endospermic seeds. In non-endospermic seeds, or ex-albuminous seeds or non-albuminous seeds, you can see that in the mature seed, endosperm will be completely utilized, it will not be present. So in castor, endosperm will be there, coconut endosperm is there, beans it's not there, pea, groundnut, bean, wheat, rice, maize, groundnut, wheat, castor. So in all these three, endosperm is completely utilized. So they are non-endospermic seeds. Reserve food will be stored in the cotyledons instead of the endosperm. So option C is the correct answer. Oh my god. Oh my god. Oh my god. Sorry. My bad. They had asked for non-endospermic seeds. So I was supposed to tick B. I made a mistake, a blunder. So option B is the right answer. So sorry, you all are not the toppers. Whoever they are, I can't find out right now. I'm very sorry. Next question. Apomixis refers to production of seeds without fertilization, product of fruits without fertilization, occurrence of more than one embryo in a seed, none of the above. Correct answer, option A. Some of you might get confused between options A and B because both of them are processes or phenomena that are happening without fertilization. When seeds are produced without fertilization, it is apomixis. When fruits are produced without fertilization, what do we call it? We call it parthenocarpy. Basically, we will get seedless fruits if they are developing, if fruits are developing without fertilization. Occurrence of more than one embryo in a seed, what is this called? If there's more than one embryo in a seed, what do we call it? Very simple, it's called polyembryony. Okay? Option A is the right answer here. Dhanish, Divya, Chaitanya, top 3. Here is our next question. ಯಕೀನ್ ಯಾವ ಯಕೀನಲ್ಲಿ ಲೋಕೇಶ್ ಅಲ್ವಾ ನಿನ್ನ ಹೆಸರು ಏನು ಹೇಳ್ದೆ ಯಕೀನ್ ಕನ್ನಡದಲ್ಲಿ ಇರಲ್ಲ ಯಕೀನ್ ವಿಲ್ ಬಿ ದೇರ್ ಇನ್ ಇಂಗ್ಲಿಷ್ classes will start I think immediately after Rupesha sorry class will start immediately after NEET 4th I'm sorry May 4th after NEET exam. I think it will be launched, and maybe around May 10th now classes start ಮಾಡ್ತೀವಿ. Which among the following statements is correct? We have to identify. Option A, wind pollinated flowers produce non-sticky pollen. This is a true statement. If it does not produce non-sticky pollen, if it produces sticky pollen, then it will get attached to random surfaces and will not be able to get carried by wind currents. Gatonogamy is genetically similar to xenogamy is false, because gatonogamy, though it is happening between two different flowers, both the flowers belong to the same plant. Moni, please stop spamming. I will answer your question. You're distracting the others. Please understand. Gatonogamy is genetically similar to xenogamy. Yeah. So in xenogamy, it happens between two different plants, pollination. So genetically different pollen can be brought to the stigma, whereas in gatonogamy that is not possible. Even though it's from a different flower belonging to the same plant, it will have the same genetic constitution, same kinds of genes. So this is incorrect. Vallisneria flowers are pollinated by wind. No, it is pollinated by water. Chthogamous flowers are unisexual, underground and closed. If they are unisexual and closed, they can never get pollinated. So they are unisexual, they are closed, yes, generally underground, yes, but not unisexual. So this is false. The true statement here is option A. Chaitanya, Vidya, Dhanish, top 3. Here is our next question. It is easy to score 300 to 350 marks in NEET 2025 paper. Is it possible? Yes, it is possible to get 300 to 350 in NEET out of 720. It is actually pretty easy. But 300 to 350 will not fetch you a seat. I mean, for most categories at least. Unless you're like super rich and you can afford a management seat. In Vallisneria, the stalk is coiled in male flowers, female flowers, bisexual flowers, all of them. The correct answer is female flowers. What is Vallisneria? Vallisneria is an aquatic submerged plant. Plant is produced inside the water body, it's submerged. So Vallisneria is a dioecious plant. You have a separate male and a separate female plant. Male plant produces male flowers, female plant produces female flowers. The female flower stalk or the pedicel is coiled like a spring and present under water. So when it matures, it uncoils and comes to the surface. It does not get detached, it comes to the surface while the pedicel uncoils. Male flowers are completely detached and released into the water body. They will passively float in the water current. If by chance they happen to meet the stigma of the female flower, pollination will happen, fertilization will happen, and it will coil back and go down the female flowers, and then development of the fruit will happen inside the water body itself. Okay, so they do not produce bisexual flowers, separate male, separate female flower, and it is the female flower stalk that coils. So option B is the right answer here. Suhas, Priyanka, Priya, top 3. Next question. Okay, Moni. ರೂಪೇಶ್, ಇವತ್ತು ಫಸ್ಟ್ ಇಯರ್ ಆಲ್ರೆಡಿ ಮುಗ್ದಾಗಿದೆ. ಸೆಕೆಂಡ್ PUC ನಾನು ಕಂಪ್ಲೀಟ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಆಫ್ಟರ್ ದಿಸ್ ಲೆಕ್ಚರ್ ಇಸ್ ಓವರ್, ಯು ವಿಲ್ ಗೆಟ್ ದಟ್ ಫಸ್ಟ್ ಪಾರ್ಟ್ ಆಲ್ಸೋ. Select the correct statements among the following. First statement, most of the grasses are pollinated by insects. Most grasses are not pollinated by insects, they are pollinated by wind. Right? That is why in grasses the flowers are not very pretty, attractive, colorful, because wind cannot see. Right? Second, transfer of pollen grains from anther to stigma of another flower of the same plant is called gatonogamy. This is true. This is false. Chthogamous flowers are autogamous. Yes, this is also true. If they don't open at all, 100% they will have to undergo self-pollination. Statement D, in xenogamy, genetically different types of pollen grains are brought to the stigma, which is also true. In xenogamy, you have pollen from different plants coming onto the stigma. So, genetically different pollen will be dropped on the stigma. B, C and D are correct. So, option C it is. Okay, a lot of you answered it as C and some of you as B. What is the difference? Okay. Statement A is outrightly correct, incorrect. Shobha, Rahul, Lork, top 3. Next question. Pay attention and answer. Think carefully and answer. Nikita, what is your question? Yeah, it is possible, Nikita, but you have to find out where you're making mistakes and correct them. If you're doing the same things that you're doing while you're getting 400, 450 or 450, 460, if you repeat your mistakes, you will get the same score. If you change and correct your mistakes, then there is possibility of improvement. For that, you have to first identify where you're going wrong. Are you not reading the question carefully? Are you not have you not understood certain concepts? So what is your issue? You have to identify and correct it, then there is a possibility. All right. How many cell divisions occur in a functional megaspore to form a mature embryo sac? How many times divisions will happen? 3 times divisions will happen. Are those divisions meiotic or mitotic? They are mitotic divisions. There are 2 options which have meiotic division written. So we can eliminate these 2 options. There are 2 options that have mitotic divisions written. So the number is varying here. So first you have the megaspore. Megaspore will undergo mitotic division, only karyokinesis, no cytokinesis. So you have a 2 nucleate stage, then 4 nucleate stage, then 8 nucleate stage. So first division, 2 nucleate, second division, 4 nucleate, third division, 8 nucleate. Totally 3 mitotic divisions will take place. Initially only karyokinesis happens, later cell walls are laid. So option C is the correct answer. Priya, Shadow, Suhas, top 3. Here is our next question. Please pay attention, read carefully and answer. अभी तुम स्टार्ट करोगे तो गोविंद ज़्यादा कुछ हो नहीं सकता, टू बी वेरी ऑनेस्ट. अभी स्टार्ट करके क्या ही पढ़ोगे? और आई थिंक लेस देन अ वीक इज लेफ्ट और मे बी या वेरी फ्यू डेज आर लेफ्ट. सो अभी स्टार्ट करके तो कुछ नहीं हो सकता है. There are people who are studying from 4 years, 5 years and you're competing with them, and it is not fair, no? If somebody who's studying 3 days before the exam, if they can score that much marks, if it is possible even, how is it fair for those people who are studying from so many days? It's not. Your result will be proportionate to how much effort you have put. Okay, so if you worked hard consistently through the last few months at least, then there is a possibility. If you want to start right now, then no. In angiosperms, how many microspore mother cells are required? How many microspore mother cells are required to produce 100 pollen grains? From 1 microspore mother cell, we get 4 pollen grains. If we need 100 pollen grains, then how many microspore mother cells? So if 1 will give rise to 4, 100 divided by 4, 25 microspore mother cells are required. Okay, so option A is the correct answer. Again, what mistake most students do when such questions are asked is, you will assume that they're always asking for how many microspores will be produced. Sometimes in the question they might give, there are 50 microspore mother cells in a microsporangium. How many male gametes will it produce? So because you're used to solving questions like this, you will assume you just multiply it by 4 or divide by 4 and arrive at the answer. But always pay attention to what is given, what is being asked. Microspore mother cell is given, pollen grains could be asked, microspore mother cell could be given, and male gametes could be asked. So if in the same question, had they asked how many microspore mother cells are required to produce 100 male gametes, let's say 200 male gametes, what would be your answer? I need 200 male gametes. How many microspore mother cells do I need? Of course, there is a possibility, Avinash. The possibilities are endless. But how much are you willing to give? Have you identified where you're going wrong so that you can work on it? It depends on a lot of things. We will still need 25 microspore mother cells because 24 microspore mother cells is going to give us 100 pollen grains. Within each pollen grain, there will be 2 male gametes. So, 200 male gametes if you require, you need 25 microspore mother cells. Okay? So, the answer will be the same even over here. Lork, Rupesh, Chaitanya, top 3. Next question. Simple. The correct answer is endosperm. Endosperm develops from primary endosperm nucleus or the primary endosperm cell, which is a product of triple fusion. If triple fusion doesn't happen, this will not form. If this doesn't form, this cannot form. So option D, endosperm, is the correct answer. Chaitanya, Priya, Ajay, top 3. Next question. Please read carefully, apply whatever you know and answer correctly. Okay. The number of meiotic and mitotic divisions that occur during the formation of 50 embryo sacs starting from megaspore mother cell. We'll first see how many are required for one embryo sac. For one embryo sac, you have one megaspore mother cell that will undergo meiosis to produce one megaspore. This megaspore will undergo mitosis 3 times to produce one embryo sac. If this is the case for one embryo sac, then for 50 embryo sacs, multiply that with 50. We will need 50 times meiosis and 150 times mitosis. Option B is the right answer. Coconut, Arunachal Pradesh. Okay. Rocky, Anam, top 3. Is this clear? Here is our next application type of question. Try to answer this. Class duration Raha. RHA, okay. I don't know what time this will end. We've started like 45 minutes. Wait, we started 1 hour ago almost? Okay. So we're still in the first chapter. We have 6 more chapters to finish. It will take as long as it takes. Probably it will get over at around 6 o'clock or so, approximately. If number of chromosomes in an endosperm is 36, endosperm is triploid. So if the triploid chromosome number is 36, what is the number of chromosomes in the root cells? What is the ploidy of root cells? Root cells are diploid. So 36 is how much? 36 andre? 36 divided by 2. I don't even have my smartwatch. 12 I think. Divided by 3 is 12. 12 twice is 24. So option C is the correct answer. Shobha, Danish, Preeti, top 3. Now, if I ask you for the ploidy of perisperm, assuming that triploid is 36, in the endosperm with 36, assuming that, how much will it be in perisperm? How much will it be in alluron layer? How much will it be in integuments? How much will it be in endothecium? How much will it be in pollen mother cell? Let me know. And how much in vegetative cell? I want you to type A, B, C, D, E, F in the same comment and give me the answer. Perisperm is nucellus itself that remains as a thin layer in some seeds. Since the nucellus is diploid, perisperm also will be diploid. So that number will be 24. Alluron layer is the outer proteinaceous layer that you find in the endosperm. So that also is part of the endosperm. Since the endosperm is triploid, that will also be triploid. So this will be 36. Integument is diploid 24. Endothecium is diploid 24. Pollen mother cell is diploid 24. Vegetative cell is haploid, so it is 12. Right? गोविंद बस एक बार पढ़ने से एनसीईआरटी दिमाग में रजिस्टर नहीं हो जाएगा. I don't want to demotivate you, but try whatever best you can and be very mindful when you're reading the questions and let's just hope for the best. Start now. 1 week, in 1 week you have the exam, and if you're starting now, there's no way I can think that you can get 300, 350 without any preparation. सब कुछ भूल गया तो कैसे याद होगा एक ही वीक में? नहीं होगा ना. Pallavi, I've created a video on this very channel saying how you can improve your score from the mock test. It is available on this channel. Just scroll up a little bit, you will get how you can improve your score. It's probably a 5-minute long video. You can get your ideas from there. Okay? Yeah, 4 days. Like Ajay is pointing out, you have less than a week. It's already Tuesday today. So, 4 days. 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 30 seconds should suffice. Mauni, video is there. How many nuclei are present in a fully developed male gametophyte? Fully developed male gametophyte is the three-celled pollen grain. Male gametophyte is the pollen grain. In the three-celled pollen grain, you have 3 nuclei, 1 nuclei of the vegetative cell and 2 nuclei of the male gamete. So totally there will be 3. Option C is the correct answer. Oh my God. How? What made you think it was A? How have so many of you thought it is A? How many nuclei are present in a fully developed male gametophyte? They are not asking about the gamete, they are asking about the gametophyte. Ajay, Darshan, top 2. Only 2 people have answered out of what? 54 people that are watching. Here is our next question. Read carefully and answer. Pallavi, I am not setting the paper. I have no idea about the difficulty level of the questions. How you can improve you in your mock test, that I have told you in the video. Okay? If we knew everything about the difficulty level, how much you can improve and all that, there will, I mean, everybody can do well, right? Moni, it's called do this to improve your NEET score or something like that. The leaf cell of a flowering plant has 22 chromosomes. Then the number of chromosomes would be. The leaf cell here is diploid. So if the diploid chromosome number is 22, then will there be 22 in gametes? No. Will there be 11 in gametes? Yes. Will there be 44 in embryo? No, it will be 22 only. Will there be 44 in the cell of a stem? No, even here it will be 22. So what among this is correct? Option B is correct, right? Danish, Preeti, Basavraj, top 3. Next question. Please read carefully and understand and answer. I will give you 1 whole minute to answer this question. Please read carefully and answer. Okay. The number of meiotic divisions required to produce 400 seeds in plants. To produce 1 seed, we need 1 male gamete and 1 female gamete. To produce 1 female gamete, you need 1 meiotic division. To produce 1 male gamete, for 1 pollen grain to form, because After one meiosis, we get 4 pollen grains. So for one pollen grain, 1/4 of meiosis does not mean it will happen 1/4. I'm just saying for one pollen grain, you'll need 1/4 meiosis. Okay. Because from one meiotic division, we'll get 4 pollen grains. Right? In order to get 400 seeds, I will need 400 male gametes. To obtain 400 male gametes, 100 times meiosis has to take place, 1/4. To produce one female gamete, one meiotic division takes place. So to produce 400 female gametes, 400 times meiosis has to take place. So 400 plus 100 is 500 times. Option B here is the correct answer. Has everybody understood? Divya, Rakesh, Kashish, top 3. If you need 400 seeds, you need 400 male gametes, 400 female gametes, 400 male gametes. If you need 100 times meiosis has to take place for microsporogenesis. For 400 female gamete, 400 times meiosis has to take place for megasporogenesis. So 400 plus 100, 500 is the correct answer. Okay? Option B it is. Next question. Correct, Joshua. Anam, that's exactly what we're doing right now. If you're still waiting for any channel to tell you like the most expected questions and paper leak sort of thing, don't waste your time. Go study instead. Filiform apparatus, where do you find it? Is it present in the synergids? Yes, it's present in the synergids. Is it present at the micropylar part or the chalazal end? It's present at the micropylar end and it guides the entry of the pollen tube. So option A is the correct answer. It's not present in the egg cell. It's not part of the embryo. It is present in the synergid but not at the chalazal side but the micropylar part. So option A is the correct answer. Danish, Wahida, Divya, top 3. Let's take a look at our next question, which is a match the following type of question where you have examples that you have to match. Under column 1, we have polyembryony, perisperm, false fruit and parthenocarpy. Read carefully and answer. Polyembryony, you will see in citrus fruits like lemon. So A is paired with R, perisperm, black pepper and beet. P, false fruit, apple, cashew and strawberry. Parthenocarpy, banana. Q, R, P, S, Q. Option C is the right answer. Hritika, Priya, Preeti, top 3. Okay, apomixis we already answered. We'll answer this question. It is diagram based. You have to match the correct parts. Last day revision heg madodu, Rocky is asking. He's asking how you can revise the last 2 days. See, everybody will have their strength and weakness. Everybody will have their strong areas and weak areas. For example, I can teach sexual reproduction in the middle of the night. You wake me up and ask me to teach. I can teach all of the concepts without even looking at the book. Principles of inheritance and variation I can teach like that, but biodiversity and conservation I cannot teach without preparation because there's so much data. So I have to revise it before I teach so that I don't get the numbers wrong. Morphology of flowering plants I have to revise before I teach because there are so many examples like that. You will have some chapters which you know very well and some chapters that you kind of struggle with a little bit. So on the last 2 days, make sure that you go through those specifics once, revise all of those concepts once. Very shortly, there will be a video uploaded on this channel where I will give you for botany all the important specifics of your NEET botany syllabus, complete short notes where you have scientist names, their contributions, which year what experiment was performed, what is the example for polyembryony and which seed was discovered where, after how many years did it germinate and flower, all of those specific details that you need to remember, all of that in one document you will get. It's around 50 pages. So you can revise using that document and just relax yourself. You've struggled so hard for at least the last 2 years. So relax yourself. When it might seem like you don't know it all, you're forgetting things. But in the actual exam, if you're patient and calm and focused and mindful and present, then the right answers will start appearing. Okay, you will remember the answers by looking at the questions. Just stay calm, don't be anxious. Okay. So what do we have? One here is this little part. This is the plumule. There are two options that have plumule as A. The other two we'll eliminate. Two things here. This is a dicot embryo. These two are the cotyledons. Then what do we have here? We have 3 and here we have 4. Okay, so this portion of the embryonal axis, 3 is the hypocotyle. 4 is the radical, tip of the hypocotyle is the radical. This here is the root cap. Okay, so root cap is not labeled here. Plumule, cotyledons, hypocotyle, radical. Option D is the correct answer. Rocky, Shirisha, Suhas, top 3. 𝐒𝐿𝐻 Suhas. Okay. Okay, let's answer this question. Read the question carefully. Which of the following is correct is what they are asking. The generative cell is bigger. No, there only it's wrong. Vegetative cell is bigger. Yes. With food reserve, and generative cell is smaller and floats in the cytoplasm of vegetative cell. This is correct. Vegetative cell is bigger without any food reserve is wrong. Generative cell is bigger itself is wrong. So option B here is the correct answer. Rocky, Hritika, Dhanish, top 3. Next question. Synergid, zygote, primary endosperm nucleus respectively are. Simple question. Synergid is part of the gametophyte, so it will be haploid. Zygote is a product of syngamy, it will be diploid. Primary endosperm nucleus, product of triple fusion, it is triploid. So option A is the correct answer. Wahida, Rocky, Danish, top 3. Okay. We'll start with the questions from Principles of Inheritance and Variation. Before that, let's take a look at our leaderboard. Ajay is on top of our list. Second, we have Fatima. Third is Preeti. Then we have Sakshi, MR, Vimala, Solo, Anam, Danish and Nafisa, top 10. Is everybody ready for Principles of Inheritance and Variation? Goat, ನೀವು ದೇವರ ಅಂತ ಕೇಳ್ತಾ ಇದೆಯಾ? ನಾನು ದೇವ್ರಲ್ಲ, ಹಿಂಗೆ ಸ್ಪ್ಯಾಮ್ ಮಾಡ್ತಾ ಇದ್ರೆ ದೆವ್ವಾಗ ಬಂದು ಸಾಯಿಸ್ಬಿಡ್ತೀನಿ ನಿನ್ನ. Is everybody ready? I want to see thumbs up in the comments. Okay. Here is our first question. Poor thing, the option C is not visible, D is not visible. Okay, let's get started. Match the terms given under column 1 with the meaning given under column 2. Choose the answer which gives the correct combination of alphabets of the two columns. Genotype, T. Recessive, it remains hidden in the heterozygous condition. Hybrid, organism produced by a cross between two individuals differing in certain characters. Phenotype is observable characters. So TRPQ. Option A is the correct answer. This was a simple question for starters. As we move on, you'll have more difficult questions. Sharon, Noman, Hritika, only 3 of you all have answered correctly. Here is our next question. Sure, Ajay, I will. என் பேரு படையப்பா இல்ல மட்டும் நடையப்பா என்னோட இழந்தன படையப்பா படையப்பா படையப்பா. When a member of a pair of alleles expresses itself fully in the hybrid, it is called. There's one pair of alleles, it is in the heterozygous condition, that allele is expressing itself fully. What do we call it? We call it dominance. Option B is the right answer here. Not bad. Wahida, Hritika, Danish, top 3. Okay, let's answer this question. A parent that produces only one type of gamete for an allele is called. If it produces only one type of gamete, that means it has only one type of allele. If it has the same type of allele, then what do we call it? If both the alleles are similar and of the same type, what do we call it? We call it homozygous. So option B is the correct answer. If it is heterozygous, it will produce two different types of alleles. Heterozygous means it will carry two different alleles, dissimilar alleles. Whenever it is in heterozygous condition, it will produce more than one type of allele. If it's producing only one type of allele, that means it is homozygous and has only one type of allele. Okay, option B is the right answer. Prajwal, Rahul, Sharon, top 3. Here is a match the following type of question. Let me give you a little extra time and show the options properly. Don't worry, Govind Purohit, डर के आगे जीत है. So you've studied, you will remember on time. If you are anxious and nervous, then whatever you've studied, you will forget. Monohybrid phenotypic ratio. Monohybrid phenotypic ratio is 3:1. Oops. One mini earthquake only happened. Typical dihybrid phenotypic ratio is P. Typical monohybrid genotypic ratio is 1:2:1. Typical dihybrid test cross ratio is T. QPST. Option C is the right answer. This is the monohybrid test cross ratio. Okay, so option C is the right answer. Divya, Prithvi, Hritika, top 3. Next question. All right. So basically what they've asked in this question is they have described a monohybrid test cross. So what they're saying is they've crossed a plant that is tall, homozygous tall, and they've crossed it with a plant that is homozygous recessive, dwarf plant. So whatever F1 hybrid they obtained, obviously it will be heterozygous tall. If they've crossed it with dwarf plants which is homozygous recessive, what will the progeny be? What is this cross? This is a test cross. So whenever you have a heterozygous plant crossed with homozygous recessive, you will get 1:1 ratio of tall and dwarf. So correct answer is option D. मैंने तेरे लिए. Oh my god, a lot of you thought it was B. You've assumed that this was, they're asking about the F2 generation of monohybrid cross, but that is not what they've asked here. Have they mentioned F2 generation of monohybrid cross anywhere? Why did you assume that it is 3:1? Please read and understand the question carefully. Rocky, Ajay, Prajwal, top 3. Unless you've made that assumption, you would not have answered it as B. Next question. Okay. So how many types of gametes are obtained from a plant of genotype this? Whenever genotype is given and they've asked you about how many types of gametes it produces, we will apply the formula 2 to the power of n, where n represents number of heterozygous genes. Over here, Y is homo and R is hetero. So you have only one heterozygous gene. So 2 to the power of 1 will be 2. Correct answer is option B, 2. Hritika, Shweta, Divya, top 3. A lot of you answered it as C. You think that whenever there are 4 letters in the genotype, it will be 4 different types of gametes based on dihybrid cross. So in the dihybrid cross, what happens? F1 generation, both the genes will be heterozygous. Since both the genes will be heterozygous, you get 2 to the power 2, 4 different types of gametes. In the genotype that's given here, even though we are considering 2 different genes, we have only one gene that is heterozygous. So n will be equal to 1 and the total number of gametes will be 2. I hope it is clear. Hritika, Shweta, Divya, top 3. Now answer this question, which is very similar. How many types of gametes? How many types of gametes are expected from the plants with genotypes? With the genotype this one. If you look over here, how many heterozygous genes are there? This is homo, this is homo, this and this is hetero. So 2 to the power of 2 is 4. Option B is the right answer. Good job. Hritika, Joshua, Sangeeta, top 3. Next question. Law of independent assortment is applicable to all genes on all chromosomes, applicable to all dominant genes, applicable only when genes are on the same chromosome, not applicable when genes are on the same chromosome. It is not applicable when genes are on the same chromosome. Whenever they are on the same chromosome, there are chances that it will get crossed over together. The chances will vary depending upon the distance between two genes. When the two genes on the same chromosome are located very close to each other, then the chances of them getting crossed over together is very high. When they are on the same chromosome but little distant, crossing over chance together is there but it's slightly less. So we call this phenomenon as linkage. All the genes that are present on the same chromosome are physically linked to each other. We call that linkage. Whenever the distance between these two genes we are considering is high, possibility of crossing over is less, recombination chances are less. Whenever they are very close to each other, then recombination chances are more. Now let's assume there are two genes which are located on the same chromosome very close to each other. Then if they get crossed over together, then recombinants will be less and the Mendel's law of independent assortment will not apply there. They will not get the same F2 generation ratio as 9:3:3:1. It will deviate a lot. They will get much higher number of parental types and a fewer recombinants. Now if you think about all of the principles or laws that Mendel proposed, there's 3 of them: law of dominance, law of segregation and law of independent assortment. Law of dominance also is not universal. It does not apply all the time. It does not apply for all organisms, for all characters. What is the deviation from Mendel's law of dominance? Incomplete dominance and co-dominance. They do not follow that law of dominance, right? What does Mendel say in law of dominance? He says that whenever there's a pair of dissimilar alleles in heterozygous condition, only one of the alleles fully expresses itself and the other one does not express itself. Whichever expresses itself is called dominant and the one that doesn't is called recessive. So what happens in incomplete dominance, when there's dissimilar pair of alleles, one will only partially express itself. You get a brand new phenotype in the F1 generation. So that is a deviation from what Mendel said. And in co-dominance, what happens in AB blood group? There is a pair of dissimilar alleles, but both of them will simultaneously express themselves. So there is no recessive trait there. So those are deviations from Mendel's law of dominance. Mendel's law of segregation is universal. Wherever diploid organisms are there, in diploid organisms, when gamete formation happens, it's only one of the pair of alleles that gets transferred to the gamete. That is universal for all diploid organisms who are performing gametogenesis, gamete formation. Talking about Mendel's law of independent assortment, this was also not universal. This doesn't apply to genes that are present on the same chromosome. Even genes that are present on the same chromosome and are present very close to each other, there it doesn't apply at all. Okay, so yeah, option D is the correct answer to this question. Oh my god, only Amy has answered correctly. Here is our next question. The three principles of Mendelism are dominant, segregation, independent assortment. Linkage isn't. There are three options that have linkage. Mendel had no idea about what linkage was because all of the characters that he chose for his dihybrid crosses had genes that were present on two different chromosomes. So he had no idea about linkage. He proposed three principles, law that we currently consider as laws. They are laws of dominance, segregation and independent assortment. So Abhishek, Ritika, Divya, top three. Let's take a look at our next question. Okay, very simple. Zoology revision link, Rahul, it has not even started yet on this channel. On this channel, it will be, I think, not tomorrow, maybe day after tomorrow, I'm not sure. Zoology has not yet started here. I think it will be on the 2nd of May. In the Kannada channel, zoology revision has already happened yesterday. Over there in the live section, you might find the link. But on this channel in English, zoology has not yet happened. Okay. In AB group of blood, the two alleles are codominant because both of them express simultaneously. Option B is the right answer. Okay. Chetan, Basavraj, Abhishek, top three. Okay, let's answer this one. Take your time, understand carefully and answer. It's okay, Rocky, for as long as you know that you're answering the questions correctly, it's fine, it doesn't matter. Whoever is coming on the leaderboard top three, they are the people who are answering the quickest. There are many more people who are answering it correctly. If I keep on reading all of the names for every question, we will run out of time. So just to keep you motivated, I'm reading the top three. In the end, after each chapter, we'll go through the leaderboard ones, okay? If a child has to have O blood group, what should the genotype be? The genotype will be homozygous recessive, lowercase I, lowercase I. If this child has to have O blood group, then the parents must have at least one lowercase I in their genotype. Now, if you look at the options, this parent has a lowercase I, this parent has a lowercase I, this parent has a lowercase I, this parent doesn't have a lowercase I. So if this parent doesn't have a lowercase I, how can that allele be transferred to this baby? It can't. That's not possible. So option B is the right answer. Abhishek, Faridi, Mimo, top 3. Again, another blood group related question. Let's see how well you'll be able to solve this. Okay, a lot of you are saying A. A child with AB blood group who is born to a mother of the genotype IAIA cannot have father of the genotype. Now this little baby has AB blood group, means she will have IAIB. We know that the mother is homozygous for A, so this I must have come from the mother. So if the father has given IB, then for sure in his genotype at least one copy of IB has to be there. This has one copy of IB, this has one copy of IB, this also has, this doesn't have. So the genotype of the father cannot be option A. Abhishek, Minato, Hritika, top 3. Okay, another blood group related question. Read carefully and answer. There's one child in a family that has AB blood group, another baby has O blood group. So the genotype of this AB blood group will be IAIB, this will be lowercase I. In which case the parents should have at least one lowercase I each and they should have IAIB as well. So both of these parents are heterozygous. One is heterozygous for A, the other is heterozygous for B. So correct answer will be option C. This also has similar alleles, but if this is the case, then there is no way O blood group is possible. For O blood group to be possible, both of the parents should have at least one lowercase I. This parent doesn't have lowercase I, right? And yeah, so option C is the correct answer. Hritika, Amy, Minato, top 3. Okay, let's answer this question. A man of blood group A, this fellow has blood group A, marries a woman of blood group AB. Which type of blood group in the offspring would indicate that the man is heterozygous? Now this guy could be homozygous or he could be heterozygous. So if he gets which of these following genotype children, does it indicate that this fellow is heterozygous? If he is heterozygous, then B blood group will be possible. Otherwise, it will not be possible. Okay, so for B blood group individual to be born, IB pairs with lowercase I and then B blood group will be possible. If this person is homozygous, then there is no chance for B blood group. Regardless of whether the person is homozygous or heterozygous, if one of the parents has AB blood group, there is absolutely no chance for O blood group. AB is still possible even if it's homozygous. A is still possible even if he is homozygous. But B can be possible only when the person is heterozygous. So option D here is the right answer. Very few of you answered it correctly. Minato, Nitin, top 3. Top 2. Next question. This is simple. Let's see how many of you will answer this correctly. A monohybrid cross between two plants, one having 24 centimeter long internodes and the other having 12 centimeter long internodes, produced F1 hybrids having 18 centimeter long internodes. This is a case of It is a case of incomplete dominance where the F1 hybrid is a brand new phenotype that is intermediate to both his parents. One parent is 24 centimeters long internode, the other one has 12. This has intermediate in between, which is 18. So it is incomplete dominance. Ayushman, Joel, Wahida, top 3. Okay, so here is our next question. Genetic disorders are very, very important. Please understand all of them. In sickle cell anemia, there is change in amino acid in beta chain. At which position? At the 6th position. What kind of mutation is it? It is a substitution mutation. Which amino acid is replaced by which amino acid? Instead of which amino acid do you have? In normal beta hemoglobin, you have glutamic acid, and after the substitution mutation, at the 6th position, instead of glutamic acid, you will have valine. Like Alim is saying, glutamic acid is replaced by valine. Correct. Kirti is also correct. Option A is the right answer to this question. Ayushman, Ram, Abhishek, top 3. Next question. ಬಸವರಾಜ್ ಉಲ್ಟಾ ಹೇಳ್ತಿದ್ಯಾ? Which one is incorrect is what we are required to find out. Polygenic character is controlled by many genes. Yes. Numerous intermediate types are found in between the two extremes in polygenic inheritance is true. Height, weight and skin color are polygenic. Yes, correct. Phenylketonuria is an example for polygenic inheritance. This is incorrect. If phenylketonuria is not an example for polygenic inheritance, phenylketonuria is an example for what? Let me know in the comments. Kritika, Pranay, Ayushman, top 3. Phenylketonuria is an example for pleiotropy, where a single gene affects multiple phenotypes. What kind of a disorder is phenylketonuria? Is it autosomal or sex-linked? Is it dominant or recessive? Phenylketonuria is an autosomal recessive Mendelian disorder, right? All right. Now let's move on. Here is our next question. If a hemophilic man marries a normal but carrier woman, what is the probability of their son becoming hemophilic? Man is hemophilic, lady is carrier. Man is hemophilic, lady is carrier. So what is the probability that the sons will become hemophilic? There's 50% chance for the son to become hemophilic. So option C is the correct answer. Clear? Right, Kirti, Divya, Basavraj? Option C is the right answer. Hemophilia is an X-linked recessive trait. Prachi, Ajay, Prince, top 3. Next question. In people suffering from hemophilia, what happens? Erythrocytes are nucleated, erythrocytes are absent, blood clotting is delayed due to the lack of certain clotting factors, platelets are absent. Right, Queen Victoria's family had many people suffering from this disease. That's why it's called the royal disease. Option C is the right answer. There's a clotting factor that is not produced. Good job, 100% of you all have answered correctly. Wahida, Hrithik, Abhishek, top 3. We have Hrithik Roshan, Abhishek Bachchan in our class. Wahida Rahman also, top 3. Yeah, here is our next question. A hemophilic daughter is born to a couple if the. When can a daughter be hemophilic? Moni, I'm from Bangalore. When can a daughter be hemophilic? Daughter is female. So for a daughter to be hemophilic, she should have both defective alleles. If she has both defective alleles, one should come from father, one should come from mother. Father has only one X chromosome, so that X chromosome has to be defective, in which case he will be hemophilic. So the father has to be hemophilic. Father cannot be normal. Father has to be hemophilic. And mother at least has to be a carrier. Okay? Mother has to be a carrier. How can a mother be a carrier? Mother can be a carrier if the maternal grandfather was hemophilic. If he was hemophilic, he would have transferred his defective allele carrying chromosome to the daughter. So in that way, the mother will be a carrier. So option B is the right answer. Nitin, Chetan, Kashish, top 3. Paternal grandfather is hemophilic and mother is carrier is given. Mother is carrier is true. See, if the paternal grandfather is hemophilic, paternal grandfather cannot pass on that X chromosome to his son, right? To the father, to this daughter's father, if he should have a defective X chromosome, that has to come from his mother, because his father will give him the Y chromosome, not the X chromosome. Even if the paternal grandfather has hemophilia, this guy cannot get it from his dad. The only way these men can get hemophilia is through their mothers. Okay, so that makes this option incorrect. Option B is the right answer. Now, here is a similar question that we can skip. Okay, we'll answer this one. A colorblind man has a colorblind sister but a normal brother. The phenotype of the parents is. Very interesting question. This colorblind man has this genotype. He has a colorblind sister who has this genotype and a normal brother who has this genotype. What would be the genotype of their parents? If there is one normal brother, that means mother has at least one normal X chromosome. If the sister is colorblind, it indicates that the mother has at least one defective allele carrying chromosome. So the mother is obviously heterozygous, she is a carrier, right? So carrier is not given, instead of carrier, mother normal is given. Okay, so mother will not be colorblind. If mother was colorblind, then all her sons would have had to be colorblind, but one of the sons is not colorblind. That indicates the mother is not colorblind. Okay, Both parents are colorblind is also wrong. Mother is not colorblind. She is normal, basically a carrier. If a female is colorblind, that means the father is 100% colorblind. Otherwise, that cannot be, the female cannot get two defective X chromosomes. If both the chromosomes of hers are defective, then one of the X chromosomes that came from her father has to be defective. That fellow has only one X chromosome. If he has one X chromosome that is defective, 100% he will be colorblind. So, father will be colorblind, mother will appear normal, but she is actually a carrier. So, option D here is the correct answer. Mr. ATP, powerhouse, energy, currency. Basavaraj, Prince, top 3. Next question. Turner syndrome is due to Ritika, my name is Shalini. The name Shalini. Shalini Somshekar. Mauni, I do not like to reveal about my personal details. I'm here to teach, you're here to learn. How does it matter what is my faith, if I'm Hindu, Muslim, Christian? How does it matter where I live? It doesn't matter while you're learning something, right? So let's stick to the subject. If you have any subject related doubts, I'm more than happy to take it. So let's not discuss personal stuff here. Turner syndrome happens only in females. They will look like females phenotypically, but they are abnormal. That is a genetic condition, because unlike normal females that have two X chromosomes, they will have only one X chromosome, the other sex chromosome is absent. So, option D is the right answer. Trisomy of chromosome 21 is Down syndrome, right? It's a part of your syllabus. Abhishek, Minato, Rahul, top 3. Next question. Okay, let's answer this one. Hello, Kevin. Long time. How are you? How is the preparation going on? A man is sterile due to improper development of testes, which has an additional X chromosome. Oh, okay. He has an additional X chromosome. He is suffering from Klinefelter syndrome. Option C is the right answer. Over here, oh, good job. 100% of you all have answered correctly. Minato, Prachi, Pranay, top 3. Yeah, so this is normal number of autosomes, but trisomy of the sex chromosomes. Unlike normal males that have one X and one Y chromosome, Klinefelter's individuals will have two X chromosomes and one Y chromosome, right? Now, let's take a look at this question. Loss of a single chromosome produces a condition called, whenever from a complete diploid cell, one chromosome is lost or less because of non-disjunction, that condition, what do we call it? It is called monosomy. Option D is the correct answer. Turner syndrome is an example for monosomy. Trisomy is when there's one extra, like in Klinefelter's and in Down syndrome. Hritika, Alim, ATP, top 3. Let's take a look at our next question. Chromosome complement of Down syndrome is 2n plus 1 is the correct answer. That is a case of autosomal trisomy. Abhishek, Preeti, Rahul, top 3. Okay, let's answer this one. Correct, Abhishek. Since it has 3 copies, we can no longer call it a pair. Strength of linkage is related inversely to the distance between genes, chromatids, chromosomes, telomeres. Genes present on the same chromosome, right? Option A is the right answer. The closer they are, higher are the chances of them getting crossed over together. Abhishek, Pranay, Ritika, top 3. Next question. From the pedigree chart, find out if the parents are homozygous, heterozygous, homozygous recessive, trait is Y-linked. Pranam, Ayush. This looks like an autosomal recessive trait. Had it been Y-linked, it should be seen only among the males, but over here you can see a female is also affected. If this looks like a autosomal recessive trait where the infected individuals are recessive and the parents are both heterozygous, so option B is the right answer. With that, we've come to the end of principles of inheritance and variation. Hritika, Pranay, Alim, top 3. Let's take a look at the leaderboard before we move on to the next chapter. Ajay has reached the top of the list. I'm so proud of you, Ajay. 130 out of 150 questions answered correctly. Second, we have Preeti. Then we have Fatima, Sakshi, MR. I think he left midway and ran away. Then we have Vimala, Solo, Anam, Hritika. Hritika is also doing very well. And Danish, top 10. Is everybody ready for the next chapter, molecular basis of inheritance? What trick do you want me to explain ATP? You have problem in pedigree, Ayush. Okay, I'll tell you a simple way in which you can answer pedigree related questions. So, usually in the options, you will have autosomal dominant, autosomal recessive, sex-linked dominant, sex-linked recessive. So, when you're looking at the pedigree chart, first thing you have to check whether it is dominant or recessive trait. When will it be a dominant trait? If at least one of the parents are affected and some of the offspring are affected, then it is a dominant trait. If both the parents are not affected and you see some offspring affected, that will be a recessive trait. Unless the parents are affected, children cannot be affected in dominant, but it's not the case in recessive. Even if the parents are not showing symptoms of the disease or disorder or the trait, offspring can still show it. That will be recessive. So we figured out whether it is dominant or recessive. Now, you have to find out whether it is autosomal or sex-linked. Usually, they will ask you for to find out whether it is autosomal recessive or X-linked recessive. If it is autosomal recessive, regardless of whether it is male or female, in each generation, both of them will be almost equally affected. But if it is X-linked, it will be more common among males. Also, if you see females affected without father being affected, that is autosomal recessive. If you see every time female is affected, father is affected, that is X-linked recessive. Okay? Yes, Abhishek. It will be, if its generations are skipping, then it will be X-linked recessive. In autosomal recessive, there is no such thing. Okay? All right, let's move on. Molecular basis of inheritance. Here is our first question, which is not, okay, which is pretty simple. DNA and RNA differ by nitrogen bases and sugars, nitrogen bases and phosphate groups, number of carbon atoms in sugars, sugar and phosphate group. The correct answer is option A. Phosphate group is the same in both, there is no change. Number of carbon atoms in the sugar is also of no change. Both of them are pentose sugars containing 5 carbons. Nitrogenous bases differ. In DNA you have thymine, in RNA you have uracil, that is a change. And sugars in DNA it's deoxyribose, in RNA it is ribose. So these two will be different. Option A is the right answer. Hritika, Rocky, Pranay, top 3. Next question. Which among the following includes only pyrimidines? Only pyrimidines. A and G are purines. So wherever you have A and G, they are not pyrimidines. C, T and U are pyrimidines. So option B is the right answer. Priya, Hritika, Basavraj, top 3. Here is our next question. I will give you like, okay, take 60 seconds and answer this. Read it carefully, understand and then answer. Is there a lag, Ajay? I don't see any issue at my end. There are 480 base pairs in one spiral of DNA, one complete turn of DNA. The total number of hydrogen bonds present between the two strands in that spiral is. How many number of hydrogen bonds are present in that complete spiral? Roughly you have 10 base pairs in one complete turn. Of those 10 base pairs, 4 base pairs are between A and T. The remaining 6 base pairs will be C and G. If between C and G, one time it pairs, there will be 3 hydrogen bonds. So 6 Cg are present, then 6 times 3 is how much? 18. 4 base pairs. Between one pair of A and T, you have 2 hydrogen bonds. Between 4, there will be 4 times 2, which is 8. So totally 26 hydrogen bonds will be present. Option B is the right answer. Govind, Purnima, Ritika, top 3. Next question. नाइन्थ में हूँ, क्या मैं ये पढ़ सकती हूँ? पढ़ सकते हो, क्यों नहीं? पढ़ने के लिए कोई क्लास और I don't know, I can't speak any more Hindi. So you can study. If you find anything interesting, you can learn. There's no problem. That's the highest maximum Hindi I can speak. How many nucleotides are present in a segment of double-stranded DNA molecule having 40 cytosine and 60 thymine bases? If there's 40 cytosine, there will be 40 guanine. If there's 60 thymine, there will be 60 adenine because Chargaff's rule. So 60 plus 40 is 100, 60, 40, 100. Totally we'll have 200 nucleotides present in that double-stranded DNA. Option B is the right answer. Prachi, Chetan, Hritika, top 3. I hope it is clear. Ayush, Hindi speaking is not allowed, meaning, yeah, because this is an English channel, but sometimes no students will get confused or somehow they end up here and they ask to teach in Hindi. Firstly, this is a pure English channel. Secondly, I don't know Hindi very well. Okay, let me give you 60 seconds to solve this one as well. What is the number of hydrogen bonds in a double helical DNA of 100 base pairs with 20 adenines and 10 thymines in one of the two strands? Okay, Mauni, you're asking for motivation. Pavan Patil is throwing out some motivating quotes in the comments. You can read that and get motivated. Okay, so what they're saying is on one strand, there's 20 adenine and 10 thymine. So there will also be 20 thymine and 10 adenine. So we total have 30 adenine, 30 thymine, 30 illaito andre. Out of 100 base pairs, we will have remaining 70 na? 70 guanine, 70 cytosine. So, what are we required to find out? How many hydrogen bonds are there? Number of hydrogen bonds. So, if you have 30 A and T, you will have 60 hydrogen bonds from these two. Between these two, you will have 7, 3 times is 210, right? 210. So, we'll have 270 hydrogen bonds. Option C is the right answer. Raj, Anjali, Likita, top 3. Is it clear? Have you understood? Next question. It is kind of like an assertion reason type of question, but they have given it as statement A and statement B. Read it carefully and answer. Statement A. How have I asked you to read assertion and reason type of questions? Take each statement as a standalone statement. Find out first whether they are true or false. Does DNA have a constant uniform diameter of 20 angstrom? Yes, it is true. 2 nanometer or 20 angstrom. Statement B. A pyrimidine always pairs with a purine in a DNA molecule. That is also true. Now, is this the reason for this? Yes, that itself is the reason for assertion. Both A and B are true and B is the reason for A is the correct answer. If only a purine paired with a purine, that portion of the double helix or the base pair would become wider. If two pyrimidines paired, it would become narrower. So you don't have that narrow wide thing happening along the length of the DNA molecule. Uniformly throughout the length, end to end, it's going to be 2 nanometers. The reason is because every time there's a purine on one strand, it always pairs complementarily with a pyrimidine. That is why that uniform width of DNA is maintained. Okay, so option A is the right answer here. Vajid, Raj, Krishna, top 3. जो है अलबेला मत नैनो. Read carefully and answer. XYZ is repeatedly asking 12th syllabus how much marks can be expected. You can go check for a video on this very channel that I've uploaded where I've given you the chapter wise weightage for every 11th grade and 12th grade chapter, so you can get it from there. Who amongst the following scientists had no contribution in the development of double helix model for the structure of DNA? Rosalind Franklin's name should not have this C, but yeah. Rosalind Franklin, Maurice Wilkins, Erwin Chargaff, Michelson and Stahl. Rosalind Franklin and Maurice Wilkins, they performed X-ray crystallography of DNA and they kind of figured out that it has 2 strands and it's helical. And Erwin Chargaff said that the amount of purines is equal to the amount of pyrimidines. There is, and he suggested a base pairing between the 2 strands of DNA and how always the ratio between A and T, G and C will be equal to 1, indicating that A is equal to T and G is equal to C. Based on these discoveries or contributions of these scientists, Watson and Crick came up and proposed the double helical structure of DNA, right? So Michelson and Stahl had no contribution in this. What were these guys responsible for? They finally gave proof that replication of DNA happens semi-conservatively. Okay, so option D is the right answer. Raj, okay, Eve, Hritika, top 3. Next question. Easy. Let's see how many of you will answer correctly. Govind Purohit joined our session sometime back and he said he has forgotten everything he studied and he wants to know if in 4 days he can get 300 to 350 score. Sometime later he's asking some girls in the chat box, are you Marwadi, where are you from? Now he stooped to a level where he's asking me for my Q test name. What can you expect, Govind? How can you expect to get even 10 marks with this kind of preparation and this kind of attitude? Okay, Ayush, what do you want me to do? The combination of a pentose sugar with a nitrogenous base constitutes a sugar and a base is a nucleoside, right? Option B is the right answer. Sugar plus base plus phosphate group is nucleotide. Option B is the right answer for nucleoside. Vajid, Ayush, Sangeeta, top 3. Here is our next question. This will take you some time. So I'll give you a full 60 seconds. Answer this carefully. Game Blaze, if we are Indian, Hindi is our pehchan. It could be your pehchan, but don't force it on all of us. Don't impose Hindi on everybody. As Hindi is is not a national language, it is one of the officially recognized languages. So is Kannada, so is Tamil, so is Telugu, so is Malayalam, so is Bengali, so is Marathi. So yeah. You've obtained a sample of DNA and you transcribe mRNA from this DNA strand and purify it. You then separate the 2 strands of DNA and analyze the base composition of each strand and of the mRNA. You obtain the data shown in the table below. Which strand of DNA is the coding strand? Serving as a template for mRNA synthesis. Okay. Are we supposed to identify which is coding strand or the one that is serving as a template? This question itself is confusing. Which strand of the DNA is the coding strand? Okay. See, whichever one is similar to mRNA will be the coding strand. this and this are very similar. So you know how mRNA sequence and coding strand sequence will be identical, except for wherever you have T in coding strand, you will have U in mRNA. So this is mRNA, this is our coding strand, and this you can see how many ever A is there, roughly around the same amount of T is there, and yeah, so these two are complementary. 30 GNC, 25 GNC, okay. Yeah, U and A roughly the same number, so this is the template. Now the question itself is not clear. I'm not sure if they're asking us to identify which is the coding strand or which is serving as a template. If we assume that it is asking for which is serving as a template, then strand 2 will be the correct answer, option B. Eve, Kashish, Raj, top 3. Have you understood? Okay. Now let's move on to the next question. Griffith used 2 strains of Streptococcus pneumoniae bacteria, S strain which was virulent and capsulated and R strain which was non-virulent and non-capsulated for his experiments on mice. Which among the following results are wrongly stated? R strain injected to mouse, mouse lives. S strain injected to mouse, mouse dies. Heat killed S strain injected to mouse, mouse dies. Mixture of live R strain and heat killed S strain injected to mouse, mouse lives. When he injects only heat killed S strain, it does not die, it survives, because S strain, when it's alive, is capable of causing pneumonia, but once it's dead, it's killed by heating it, it can no longer cause pneumonia. So over here, mouse will survive. Mixture of live R strain and heat killed S strain injected into mouse, mouse will die. That is when transformation will happen. Live R strain will get transformed into live S strain bacteria. So both C and D are wrongly stated. Option B is the correct answer to this question. Sangeeta, Chetan, Tasin, Tasnimul, top 3. Okay, here is an assertion and reason type of question. Read carefully and answer. DNA fingerprinting is very well known for its application in paternity testing in case of disputes. Reason, it employs the principle of polymorphism in DNA sequences, as the polymorphisms are inheritable from parents to children. Now, if you look at it as individual statements, both the statements are true. You can eliminate both of these options. Now, whether it explains the reason assertion correctly, we'll add because here and read. DNA fingerprinting is very well known for its application in paternity testing in case of disputes because it employs the principle of polymorphism in DNA sequences, as the polymorphism are inheritable from parents to children. That is the reason why it's so, it can be used in case of parental paternity disputes, right? So, because the child will have half of the VNTRs exactly like his father's. So that way you can find out whether it is, who is the father of the child. So both assertion and reason are true, and reason does correctly explain the assertion. So option A is the right answer. Good job. Eve, Kashish, Vajit, top 3. Next question. Alright, let's answer this one. A double-stranded DNA is made radioactive in both the strands. It's allowed to replicate twice in non-radioactive medium. The result would be all 4 daughter DNA will have radioactivity. See how? You have one cell that has 2 strands. After 2 rounds of replication, 2 rounds of cell division, yeah, so 2 times it's allowed to replicate. After the first round, you'll have 2 DNA molecules. After the second round, you'll have 4 DNA molecules. Of the 4, are all of them showing radioactivity? All of them will not show radioactivity. So, because when new strands are forming, in the new medium, there is no radioactive medium present. It's non-radioactive, so it's not possible for all the 4 strands to show radioactivity. The ones that are formed, yeah, 2 of them will show radioactivity, 2 of them will not. 2 of the daughter DNA will have radioactivity and the other 2 without radioactivity. This is correct. Oh wait, let me check. 2 of the daughter DNA are without radioactivity and 2 with radioactivity in both their strands. Both the strands is not true. It can never again have radioactivity on both the strands. Hybrids will be there, one radioactive, one non-radioactive. 2 of the daughter DNA are without radioactivity and 2 with radioactivity on one of their strands. This is correct. 2 have radioactivity, other 2 without radioactivity. 2 are without radioactivity and 2 with radioactivity. Both of these are same, except here they have specified that the radioactivity will be in one of their strands specifically. So option D is the right answer. B is also not incorrect, but Eve has answered this question. I think you didn't read all of the options and answer, right? This is very similar to the Michelson and Stahl experiment that was conducted to prove how DNA replication happens semi-conservatively. But the difference is over there they used heavier isotope of nitrogen that was not radioactive. Here they are saying they are using radioactive medium. Okay, so just remember what you learned in that experiment. After the first generation, you can no longer get heavy DNA because the new medium does not contain heavy isotope, right? So the number of hybrids that you will get will be constant and the number of light DNA you will get will go on increasing generation after generation. Same way here also, the number of DNA that you will get which has one radioactive, one non-radioactive strands will be constant. As the number of generations go on, the number of DNA molecules that you will get with both having not radioactive DNA will go on increasing. Okay? Yeah. Let's answer this one. Pretty simple question. Okazaki fragments are formed during. I don't know, Ayush. Maybe the poll timer got over. Okazaki fragments are formed when? During transduction, transcription, replication, translation. It happens during replication, away from the replication fork, right? On the lagging strand, discontinuous synthesis. Option C is the right answer. Wajid, Anjali, Tasnimul, top 3. Next question. Enzyme required for transcription is RNase, endonuclease, RNA polymerase, DNA polymerase. For transcription, you require DNA dependent RNA polymerase. So option C is the correct answer. DNA dependent DNA polymerase is used for replication. Wajid, Eve, Wahida, top 3. Next question. Yes, it's a simple question, Ayush. Shashank, no, hi, hello, friendship business here. Just read the question and answer, okay? The amino acid sequence in proteins is determined by the sequence of nucleotides in the dash. Oh my god. The amino acid sequence in proteins is determined by the sequence of nucleotides in mRNA. The sequence of nucleotides in mRNA is further determined by the sequence of nucleotides in DNA. So option C is the correct answer here. Bhagya, Anjali, Ayan, top 3. Okay, simple question. Let's not run a poll for this. In genetic code, all codons consist of 3 nucleotides, which is why we say that it is a triplet code. Right? Option C is the right answer. Here is our next question. A codon in mRNA is read in which direction? 3 to 5, 5 to 3, both 3 to 5 and 5 to 3, 3 to 6 prime. Codon in mRNA is read when? During translation. So it is read from 5 prime to 3 prime direction. Not just in translation, DNA replication and transcription also proceed from 5 prime to 3 prime direction only. So option B is the correct answer. In replication, regardless of whether it is the lagging strand, leading strand, continuous synthesis, discontinuous synthesis, regardless of it all, it always proceeds in the 5 prime, from the 5 prime end to the 3 prime end. It is at the 3 prime OH group where new nucleotides will get added. Okay. So option B is the right answer here. Anjali, Raj, Eve, top 3. Here is our next question. Read carefully and answer. The number of different kinds of amino acids that take part in the process of protein synthesis in living cells is how many amino acids? 20 amino acids. Option C is the correct answer. I think most of you will get confused here and think they are asking about codons and write 61 as the correct answer. But here, carefully, if you look at it, how many amino acids is what they've asked. So option C is the right answer. I knew it. I guessed it. Raj, Ayush, Sunidhi, top 3. Okay, so this is a lengthy question. I will give you 60 seconds. Aditya Pandurangan is crying. It's okay, Pandu. Happens. That is where focus is important. Which of the following are true regarding characteristics of genetic code? 3 consecutive bases of DNA code for one amino acid in a polypeptide chain. This is true. Generally, the same genetic dictionary is applicable to all organisms. Basically, what this means is that the genetic code is nearly universal. They're saying generally, so it's okay. Some exceptions will be there. A single amino acid is often coded by more than one triplet code is also true, which is why we say the genetic code is degenerate. A single codon can specify more than one amino acid is false. There is specificity. It is unambiguous because one codon will always specify the same amino acid. The nucleotide of one codon is not shared either by the preceding codon or by the succeeding codon. This is also true. This just indicates that it is non-overlapping. So, which ones are true? 1, 2, 3 and 5 are true. Option C is the correct answer. Anjali, Eve, Sunidhi, top 3. Eve is doing really well. She is in the top, she or he. Top 3 for most of the questions. Here is our next question, which is pretty simple. So 30 seconds should suffice. Okay, I'll go back and read that question. Ayush. The entire genetic code and process of protein synthesis involves dash codons and dash amino acids. "64 codons and 20 amino acids." So option B is the right answer. Eve, Wahida, Wajid, top 3. What did you say? Where is DNA mentioned here? There's no mention, Ayush. I don't know what you were talking about. Oh, 3 consecutive bases of DNA for one amino acid. Ultimately, even if it's mRNA, where does the code for mRNA come from? It comes from DNA itself, right? And also there's no option which says only 2, 3, 5 are correct. Yeah. So the correct answer is what I've marked. What is degeneracy in a genetic code? Try to answer this one. The genetic code is degenerate because each codon has a different meaning. Each codon has many meanings. Many codons have same meaning. 3 codons do not specify any amino acid. Correct answer is option C. Many codons can have same meaning means it codes for the same amino acid. Right? So option C is the right answer. Eve, Sunidhi, Wahida, top 3. All the following statements are correct except Okay. All the following statements are correct except we have to look for what is not correct. Genetic code is comma-less, that means it's read in a contiguous fashion. Genetic code is overlapping is not correct, it is non-overlapping, which means one codon, one nucleotide in mRNA will be only part of one codon. It cannot be part of more than one codon. The previous one or the next one cannot include one of the nucleotides in a particular codon. It is nearly universal, yes. Genetic code is degenerate is also true. So option B is what is not true. Eve, Raj, Karthik, Vajit, top 3. Next question. Okay, let's see how many of you will answer this question correctly. Selvi, you can just type in the comments like how everyone else is doing. Whatever you think is the correct option, just type the option in the comments. Okay, there's nothing extra you have to do. Just give the answer in chat, Selvi. If the base sequence of a codon in mRNA is 5' AUG 3', then the sequence of tRNA pairing with it must be 3' UAC 5'. So, in how many of these? Near the 3' there is a U, it's only this one. So, option B is the correct answer. Here UAC is complementary, but look at the polarity, it's 5' and 5', similar to how DNA template and mRNA are complementary. mRNA and tRNA have to be complementary. So option B here is the correct answer. For 5' AUG in codon, tRNA has to be 3' UAC. Here they have given 5' CAU 3', same thing. If you reverse it, just check the polarity and the sequence both. So option B will be the right answer. I know a lot of you would have answered this incorrectly. Oh my god, what is this behavior? GUA. Where did it come from? You've just reversed it. Krishna, Rocky. Only 2 of you have answered it correctly. Okay, let's answer this one. This should take you like 10 seconds to answer. What is adenosine? Adenosine is a nitrogenous base of RNA, nucleoside of RNA, nucleotide of RNA, nucleotide of DNA. Adenine is a nitrogenous base that you will find in both DNA and RNA. Adenosine is adenine attached to ribose. So adenosine is an RNA nucleoside. If adenine is attached to deoxyribose, it will become a nucleoside called deoxyadenosine, right? So adenosine is a nucleoside that you will find in RNA. If phosphate groups get attached to this, then it will become adenyllic acid in case of RNA or deoxyadenyllic acid in case of DNA. Okay, so option B is the correct answer. Chaitra, Raj, Likhita, top 3. Let's move on to our very simple next question. Which type of RNA has a structure resembling a clover leaf? सॉन्ग सुनने के लिए एक और चैनल है टी सीरीज, वहां जाके सुन सकते हैं. Which type of RNA has a structure resembling a clover leaf? It is tRNA. In its tertiary structure, what is the shape of tRNA? What will be its shape? Eve, Anjali, Raj, top 3. Ayush, fast is okay. Even if you're not the quickest, it's fine. For as long as you've answered the question correctly, it doesn't matter. Okay? Just focus on whether you're answering it correctly or not. Three types of RNA involved in structural and functional core for protein synthesis, serving as a template for translation, transporting amino acids. structural and functional core for protein synthesis has to be ribosomal RNA. There's two options with ribosomal RNA. And second one is serving as a template is mRNA, transporting amino acids is tRNA. So option C is the correct answer. Eve, Shashank, Sunidhi, top 3. Okay, let's answer this one. Wajid, you have very little time left. There is no jaldi. Okay? आराम से करना लेकिन करेक्ट आंसर करना. A messenger RNA is 336 nucleotides long, including the initiator and termination codons. The number of amino acids in the polypeptide translated from this mRNA would be. If it includes the termination codon, then the terminator codon will not have any amino acids, any tRNA to bind to. So technically only 333 nucleotides will code for amino acids. If there are 333 nucleotides, it means there's 111 codons. Each codon codes for an amino acid. So option C is the right answer. Anjali, Pradeep, Vinayak, top 3. Here is a lengthy question. I will give you 60 seconds. Let's see how many of you will answer correctly. Certain events occurring during protein synthesis are listed below. Which of the following options the events are in correct order? First, okay, transcription has to happen first. 2 is 1. Second, mRNA attaches to 30th subunit, small subunit of the ribosome. Then tRNA will join with the initiator codon. Then large subunit will come and join. Then entry of first tRNA is entered to the P site, A site, second tRNA will come and join. Then formation of peptide bond, then this. So 1 is 2. 2, 3, 1, 4, 5, 6, 7. Option B is the correct answer. Shashank, Maitri have answered correctly. Transcription has to happen, right? First, DNA gets transcribed and forms mRNA. That has to happen. Without mRNA, how will translation happen? Right? So, correct. Option B is the correct answer. Next question. During translation in ribosome, mRNA is present where? The peptidyl transferase reaction occurs where? Peptidyl transferase reaction happens in the large subunit. mRNA is present between both the subunits. So option A here is the right answer. Eve, Raj, Wajid, top 3. Next question. Okay, let's answer this one. A polypeptide just released by a ribosome from an mRNA has 75 amino acids. What would be the number of nucleotides in the coding region of, what do you mean coding region of mRNA? Okay, in the mRNA, including the initiation codon and the termination codon. If it has 75 amino acids, then how many nucleotides will be present? 75 3 times is how much? 150, 225, right? 225 for the 75 amino acids plus 1 stop codon is 228 because they've asked including the termination codon. So option B here will be the correct answer. From 75 amino acids, coding codons will be 225. There will be one stop codon or a terminator codon which will not code for any amino acid. Release factor will come and bind there and complete the process of translation. So if you include that codon as well, option B 228 will be the correct answer. Vinayak, Eve and Wajid, only 3 of you all have answered correctly. Okay, we'll answer this one. Yes, Selvi, 225. See, 75 amino acids, each one codon. So, one codon has 3 nucleotides. So, from 75 amino acids, we have 225 nucleotides. But there's one codon in the mRNA, which is a stop codon, which will not code for any amino acid. Include those 3 nucleotides in that stop codon to 225, it will come up to 228, right? Consider the statements given below and find the answer among the options given. Statement A, UAA, UAG, UGA codons in mRNA terminate the synthesis of polypeptide, correct. UAA, UAG, UGA codons are not recognized by TNA, that is also correct. And that is also the reason why it terminates the process of translation, right? So there are no tRNAs that will come and bind to these stop codons. Instead, a release factor comes and binds. So that is how the whole process of translation gets terminated. So A and B statements both are true, and the reason B statement is the reason for A. So option A here is the right answer. Eve, Raj, Wajid, top 3. Okay, we'll answer this one. With regard to mature mRNA in eukaryotes, exons and introns do not appear. Exons appear but introns don't appear. Introns appear but exons don't appear. Both exons and introns appear in mature RNA. Introns do not appear because they are intervening non-coding sequences. During splicing, the introns are removed and the exons are joined. So, in the mature RNA, exons will be there, introns will not be there. Option B is the correct answer. Eve, Sunidhi, Wajid, top 3. Okay, let's answer this one. Discontinuous synthesis of DNA occurs in one strand because DNA molecule being synthesized is very long. DNA dependent DNA polymerase catalyzes polymerization only in one direction from 5' to 3' end. It is a more efficient process. DNA ligase has to have a role. Discontinuous synthesis of DNA occurs in one strand because DNA dependent DNA polymerase can catalyze the polymerization of the new strand only from 5' to 3' end, right? So option B is the right answer. Raj, Wajid, Rocky, top 3. Let's move on to our next question. Okay, let's answer this one. While analyzing the DNA of an organism, a total number of 5,386 nucleotides were found, out of which the proportion of different bases were adenine 29%, guanine 17%, cytosine 32%, thymine 17%. Considering the 𝐄𝐍𝐆 rule, it can be concluded that it is a double-stranded circular DNA, it is a single-stranded DNA, it is a double-stranded linear DNA. No conclusion can be drawn. Correct answer is option B, it is a single-stranded DNA. Had it been a double-stranded DNA, there should have been base pairing such that A should be equal to T and G should be equal to C. Over here we are noticing how A and T are not equal, G and C are also not equal, indicating that there is no base pairing. So it is likely a single-stranded DNA. Option B is the correct answer. E. Wajid Sakshi, top 3. Here is our next question. The amino acid attaches to the tRNA at its. Where exactly does the amino acid attach to the tRNA? Is it at its 5' end, 3' end, anticodon site or DHU loop? Vinayak's full name is Vinayak Teli. Okay. There's a Bhagyashri Teli also. Are you two cousin siblings or something? It attaches to the 3' end. Option B is the right answer. E. Wajid Pradeep, top 3. Okay. Let's answer this one. Which among the following is correct with reference to mRNA? Is the mRNA polycistronic in prokaryotes? Yes, it's not polycistronic in eukaryotes, it's not polycistronic in both, and it is monocistronic in eukaryotes. The first answer is correct. It is not monocistronic in prokaryotes. It's polycistronic in prokaryotes and monocistronic in eukaryotes. So option A here is the right answer. Nidhi, Anshul, Anvi, top 3. Here is our next question based on lac-operon. Match the components of E. coli and their functions given under column 1 and 2. Structural genes, code for enzymes. LAC-Z, LAC-Y, LAC-A, code for beta-galactosidase, permease and transacetylase respectively. Operator gene is the binding site for the repressor protein, right? Repressor protein. Promoter gene is the binding site for RNA polymerase. Regulator gene codes for the repressor protein. So 3142, option D is the right answer. Oh my god, why have so many of you answered it as C? Structural genes, binding site for RNA polymerase. You didn't even read it properly or what? Only Anshul has answered correctly. Okay, let's answer this one. With reference to lac operon, which among the following statements are correct? Lac operon is an example for transcriptional control. When lactose is absent in the medium, lac operon is in switched off condition. Promoter gene is the site for binding of repressor protein. Operator and promoter genes do not code for any enzymes. Over here, 1, 2 and 4 are correct. 3 is not correct because repressor does not bind to promoter, it binds to operator. Okay, so 1, 2 and 4 are correct. Option D is the correct answer. Eve, Nidhi, Raj, Karthik, top 3. With that, we've almost come to the end of this chapter. This is our last question from this chapter. Which was the last human chromosome to be completely sequenced? Is it chromosome 1, 11, 21 or X? Which chromosome was last to be sequenced? The correct answer is option A, chromosome number 1, and it was completed in 2006, I suppose. Option A is the right answer. Eve, Raj, Anshul, top 3. So before we move on to microbes and human welfare, let's take a look at our leaderboard. Ajay is still on top of our list, maintaining his position. 134 out of 156 answered correctly. Preeti is second, Fatima is third. Then we have Sakshi, MR, Vimala, Hritika, Solo, Anam, Danish, top 10. Is everybody ready for the next chapter, microbes and human welfare? Let me know in the comments. Thumbs up if you're ready. Are you all tired? Alright, let's move on to one of the easiest chapters to ever exist, microbes and human welfare. Here is our first question. Select the wrong statement among the following. We are required to identify which one is incorrect. Ecology after this, Wajid. Lactic acid bacteria are parasitic bacteria. Lactic acid bacteria growing in milk produce acids that coagulate and partially digest milk proteins. Lactic acid bacteria increase the vitamin B12 content in curd. Lactic acid bacteria play a beneficial role in checking the disease-causing microbes in our stomach. It is not a parasitic bacteria. It is incorrect. Okay, other 3 statements are true. So option A is the correct answer. Good job, everyone's answered correctly. By everyone, it's 10 students. Eve, Raj and Sunidhi, top 3. Next question. Which among the following is a wrong match? Dosa and idli are fermented by bacteria. Bread is fermented by yeast. Roquefort cheese is fermented by bacteria. Ethanol is produced by Saccharomyces. Which is incorrect is what we are required to find out. What is incorrect here is option C. Roquefort cheese is ripened by a fungus called Penicillium roqueforti, not a bacterium. Swiss cheese is bacterium, Propionibacterium shermani. Roquefort cheese is by a fungus, right? So option C is incorrect here. It is the wrong match. Raj, Rai, Divya, top 3. Excuse me. Next question. The large holes in Swiss cheese is due to the production of dash by a dash named dash. Large holes in Swiss cheese is due to the production of carbon dioxide, not oxygen, and that is produced by a bacterium called Propionibacterium shermani. So option B here is the correct answer. Raj, Divya, Rai, top 3. Here is our next question. Read carefully and answer. Biogas, citric acid, penicillin and curd are the products of microbial action. Among these, fungi are involved in how many? Biogas is produced by methanogenic bacteria. They are not fungi. Citric acid is Aspergillus niger, that is a fungus. Penicillin by Penicillium notatum, which is a mold, which is a fungus. Curd by lactic acid bacteria, which is obviously a bacteria, not a fungus. So 2 is the correct answer. Option C it is. Raj and Amy have answered correctly. Alright, let's answer this one. Which among the following is a set of alcoholic beverages produced by distillation? Which of these are distilled alcoholic beverages? Wine and beer, wine and whiskey, whiskey and brandy, brandy and beer. Oh Ajay, it's okay. Wine and beer are not distilled. So whiskey and brandy are distilled. Also rum is. Option C is the correct answer. Had they asked which is not distilled, option A would have been the correct answer, but they've asked after fractional distillation which ones are produced. So option C is the correct answer. Raj, Sunidhi, Sakshi, top 3. Here is our next question. Bottled juices are clarified by the use of streptokinase and cyclosporin, pectinases and proteases, statins and zymase. Option B is the right answer. Pectinases and proteases. Pectinases obviously will help in breakdown of pectin. Proteases will break down certain proteins, and the juice will get a clearer appearance. That's why it's called clarifying agent. Streptokinase is called clot buster. It is used in dissolving clots. Cyclosporin is an immunosuppressive drug. Okay, so that is not used in clarifying bottled juices. Option B is the right answer. Statins are blood cholesterol lowering agents. Rai, Divya, Raj, top 3. Let's move on to our next match the following type of question. Which of the following, oh wait, it's not match the following. Which of the following are correct matches is what we are required to find out. They've given name of the microbe in one column, which group it belongs to, what is its product and what is its use. Monascus purpureus is a yeast, a type of fungus. It produces statins, which is a blood cholesterol lowering agent. It is correct. Trichoderma polysporum is a fungus that produces cyclosporin A, which is an immunosuppressive drug. That is also correct. Streptococcus is a bacterium that produces streptokinase, which is used as a clot buster. That is also correct. Aspergillus niger is a fungus from which we obtain citric acid, and it is used in the preservation of food. That is also correct. So all of them are correct, right? So 1, 2, 3, 4. Option C is the right answer. Divya, Lakshman, Nidhi, top 3. Alright, let's answer this one. Biofertilizers. Which of the following is a mycorrhizal fungus? Mycorrhiza are symbiotic associations of fungi with roots of higher plants like gymnosperms and angiosperms. Which of the following is a fungus that forms mycorrhizal associations? Glomus, Azospirillum, Monascus, albugo. Glomus it is. Option A is the correct answer. Azospirillum helps in nitrogen fixation. It's a bacterium, right? Raj, Rocky, Lakshman, top 3. Here is our next question related to sewage treatment. The word flock refers to. The word flock refers to soil and small pebbles, masses of aerobic bacteria with fungal hyphae, organic matter in sewage, masses of anaerobic bacteria with fungal hyphae. I want you to read the question carefully. Correct answer is option B. We are using it in sewage treatment. In the secondary treatment, do we use aerobic or anaerobic bacteria? We use aerobic bacteria that will break down the organic matter in the sewage, right? So option B is the right answer. They are not anaerobic. Selvi, Amy, Raj, top 3. Here is our next question. Regarding biocontrol, which of the following is a wrong match? Ladybirds, aphids, dragonflies, mosquitoes, Bacillus thuringiensis, butterfly caterpillars, Trichoderma, moths. All of these are biocontrol agents. Biocontrol agents are those living organisms that can be used to control pest as well as disease. While the first 3 are involved in control of pest, Trichoderma is used in controlling disease. Okay, so it will not act against moths. It will make sure that the plants are disease resistant to an extent. So option D here is the correct answer. Good job, Rocky, Karthik, Selvi, top 3. Here is our next question. Which among the following is a wrong pair? Aspergillus niger, citric acid, Trichoderma, Cyclosporin A, Clostridium, lactic acid, Monascus purpureus, statins. A lot of you are typing C in the comments. If Clostridium does not produce lactic acid, Clostridium produces what? It produces a type of organic acid. What does it produce? It produces butyric acid, right? Option C is the incorrect match. Lactic acid, of course, is produced by lactic acid bacteria like Lactobacillus. Raj, Priya, Rocky, top 3. Next question from sewage treatment. Which of the following statements is false? Greater the BOD of wastewater, lesser is its polluting potential. Secondary sewage treatment involves biological treatment. Biogas is produced by the activity of anaerobic bacteria. Statins are produced by the yeast Monascus purpureus. Which is false here? Option A is false. If BOD is high, then its polluting potential also will be high, not low. So option A here is the correct answer. It is the incorrect statement but our correct answer. Selvi, Rai, Raj, top 3. Okay, let's answer this one. Which of the following is not a biocontrol agent? Baculoviruses, Trichoderma, Bacillus thuringiensis, Glomus. Bhuvana is asking something. Bhuvana or Bhavna? Bhuvana. Only from 2:45 p.m. video is available. After my session gets over, maybe after an hour or so, the entire thing will be available for you to watch. The correct answer is Glomus. Glomus is not a biocontrol agent. It is a biofertilizer. Option D is the right answer. Selvi, Raj, Priya, top 3. Given below is the diagram of a biogas plant. In which of the options all the 4 parts A, B, C and D are correct? A is dung and water. There's only one option that says A is dung and water. B is the actual digester or that concrete tank. C is the floating gas holder. D is the used sludge which is used as manure later. So option D here is the correct answer. Sagar, Selvi, Chaitra, top 3. Okay, let's answer this one. The biofertilizer present in the roots of legumes is Anabena, Rhizobium, Azospirillum, all of the above. Rhizobium is symbiotically associated with the roots of leguminous plants like peas and beans, where it forms nodules and helps in nitrogen fixation. It performs biological nitrogen fixation. So option B is the right answer. Selvi, Raj, Bhuvna, top 3. Anxiety. Okay, let's answer this one. Statin is blood cholesterol lowering agent. Cyclosporin is immunosuppressive. Streptokinase, clot buster. Lipase, removal of oily stains. 3, 4, 2, 1. Option D is the correct answer. Raj, Bhuvna, Amy, top 3. Okay, let's answer this one. Correct answer is option C. Priya, Selvi, Rai top, Raj, I think that was Raj Karthik, top 3. Wait. Okay, we'll answer this one. Which of the following sets is not a biofertilizer? Prem Kumar, is the paper tough? I will let you know on the 4th of May at 5 p.m. Which of the following sets is not biofertilizer? Azospirillum, Azotobacter, Annabena, Nostoc, Bacillus thuringiensis, Methanobacterium, Oscillatoria and Rhizobium. Azospirillum, Azotobacter, both of them are free-living nitrogen-fixing bacteria. Annabena and Nostoc are nitrogen-fixing cyanobacteria. Oscillatoria is a nitrogen-fixing cyanobacteria. Rhizobium is a nitrogen-fixing bacterium. All of these are biofertilizers. Bacillus thuringiensis is a biocontrol agent. Methanobacterium is utilized in the production of biogas. So these are not biofertilizers. Option C is the right answer. Rocky, where are you going? Amy, Selvi, Divya, top 3. Here is our last question from this chapter. Which of the following is a wrong match of microbes and its industrial product while the remaining 3 are correct? Which one is wrong? Yeast statins, Acetobacter aceti, acetic acid, Aspergillus niger, citric acid, Clostridium botulism, lactic acid. Which is wrong? There's a mixture of A and D that I can see in the comments. Statins are produced by Monascus purpureus, which is a species of yeast itself. This is correct. Acetobacter aceti produces acetic acid. Aspergillus niger produces citric acid. Clostridium botulism, lactic acid, is incorrect. Clostridium botulism produces butyric acid. Lactic acid bacteria like lactobacillus produces lactic acid. So this is incorrect. Option D is the right answer. Nandu, Raj Nidhi, top 3. Nandu in Kannada means it's mine. Nandu means mine. Your name is Nandu. So let's take a look at our leaderboard. Okay, Ajay is on top of our list. Second, we have Preeti. Third, we have Sakshi. Then we have Fatima, MR, Vimala, Hritika, Solo Soul, Anam and Divya, top 10. Is everybody ready for ecology unit? Give me a thumbs up in the comments. Is everyone ready? Ajay, no, I'm not getting bored of your name. Why do you ask? All right, let's move on. Let's answer this question. Temporary movement of animals from a stressful habitat to a hospitable area and return is called immigration, emigration, migration, navigation. You know, in organisms and populations, if you look at the new NCERT textbook, the rationalized one, you don't have organism and its environment part in the new NCERT textbook. But in the NEET syllabus, organism and its environment part is also there. This is a question from that, response to abiotic factors. But in the last year's NEET also, it was there part of the syllabus, but no questions were asked from this. Just to be safe, you can prepare, you can read that part also. Okay. So migration is the correct answer. Option C it is. Sunidhi is saying tata bye bye and leaving. Sunidhi, you've done really well today. There's a lot of questions that you've answered correctly. So congratulations and all the best. Bye-bye. Selvi, Amy, Divya, top 3. Next question. The pyramids A, B and C represent. If the pyramid is triangular, what does it mean? If it is urn-shaped, what does it mean? If it is bell-shaped, what does it mean? If the pre-reproductive individuals are more than the percentage of reproductive individuals, in the future the population is going to expand. If they are pretty much the same, then it's going to be stable, not much of a change in the population size even in the future. If the pre-reproductive individuals are much lesser than the reproductive individuals, then in the future the population is going to decline. So option D here is the correct answer. Raj, Shrivardhan, Bhuvana, top 3. Here is our next question. In the logistic growth equation, what is the logistic growth also called? Bye Ajay, take care, all the best. In the logistic growth equation, what does N represent? Intrinsic rate of natural increase, carrying capacity, population density at time t, logistic numerator. N is the population density, intrinsic rate of natural increase is R, which is basically birth rate minus death rate, right? Carrying capacity is K. Logistic growth, when does it happen? Whenever the resources in that particular habitat are limited. Carrying capacity is the maximum number of individuals that a habitat can support in a population, right? So, logistic growth curve, what is the shape? Who can tell me in the comments? Vinayak, Preeti, Bhagya, top 3. Vinayak, you kept saying that your name is not coming on the leaderboard. See, your name is here finally. Here is our next question. An interaction between two different organisms where both individuals are benefited is. An interaction between two different organisms where both are benefited. What do you call it? Commensalism, mutualism, parasitism, amensalism. Commensalism, one is benefited, one is neither benefited nor harmed. Mutualism, both are. Parasitism, one is positive, one is negative. Amensalism, one is negative, one is neutral. Both positive is mutualism. Option B here is the right answer. Good job, everyone's answered correctly. Priya, Selvi, Amy, top 3. As an adaptation of xerophytic plants, we've learnt about this, or even certain plants. Sunken stomata are found in xerophytes, hydrophytes, mesophytes, halophytes. I think I gave out the answer. Oops. Xerophytes, why do they have sunken stomata? To reduce the rate of transpiration and conserve more water. Raj, Vaishnavi, JD, top 3. Anxiety. Commensalism is a relationship between two organisms in which one is benefited, other gets affected, one is benefited, other remains unaffected, both are benefited, both are unaffected. One is benefited, one remains unaffected, it's neutral. So option B is the correct answer for commensalism. A lot of you have answered correctly. Dinesh, Selvi, Raj, top 3. Okay. Let's answer this one. This again is from the first part of the chapter. No, Sunita, not yet. Which of the following is the law that states that warm-blooded animals of cold regions are comparatively larger in size than their counterparts? Yes, it is Allen's rule. Option A is the correct answer. Hey, wait, no. Allen's rule is the extremities are small, right? The extremities are like the ears and the limbs are small. That is what Allen's rule says. This I think is Bergman's rule. Eve and Priya, top 3, top 2, only 2. Okay, answer this one. Logistic growth occurs when there is a fixed carrying capacity, asexual reproduction only, sexual reproduction only, no inhibition from crowding. It happens when there are limited resources and there's a fixed carrying capacity. We get an S-shaped sigmoid curve, right? Selvi, Raj, Rahul, top 3. Top 2. Yeah, top 3. Here is our next question. There exists a close association between the alga and the fungus within a lichen. The fungus fixes atmospheric nitrogen for alga, provides protection, anchorage and absorption for alga, provides food for the alga, releases oxygen for the alga. Correct answer is option B. What happens here? Alga is the photosynthetic partner, that is what will provide food for the fungus. In return, fungus being filamentous will help in providing shelter and also helping in absorption of water and minerals and pass it on to the alga. So option B is the correct answer. Fungus is not photosynthetic, so it cannot provide food for the alga. Alga being photosynthetic itself does not require food from any other organism. Oxygen is released only by photosynthetic organisms, so the fungus does not release oxygen. Like us, it also releases carbon dioxide. So option B here is the right answer. Selvi, Dinesh, Divya, top 3. Okay, let's answer this one. When population reaches carrying capacity, what happens? Mortality rate will become equal to birth rate. Mortality rate will become greater than birth rate. Mortality rate will become less than birth rate, none of the above. When it reaches carrying capacity, the population growth will become steady, stationary. That means it will no longer grow exponentially, it will not drop either. It will remain at pretty much the same level, very close to the carrying capacity. When it's remaining constant, that means that the number of births is almost equal to the number of deaths, which means mortality rate is almost the same as birth rate. If mortality rate becomes greater than birth rate, then there should be a drop in the population size. If mortality rate is less than birth rate, there should be an increase in the population size. But that doesn't happen, right? It remains steady because birth rate and death rate will become equal almost. Shrivardhan, Raj, Bhagya, top 3. Okay, let's answer this one. Which of the following would necessarily decrease the density of a population in a given habitat, which will decrease the density of population? Natality is birth. Natality will increase the population. Mortality is death, it will decrease. When birth is more than death, then obviously there will be an increase in the population. Immigration increases population size, immigration decreases the population density. If there's more immigration than immigration, obviously the population density is going to increase. Mortality and immigration both contribute to a decrease. Natality and immigration both contribute to an increase. They're asking which will cause decrease in density of population. So the correct answer here is option C. Selvi, Bhuvna, JD, top 3. Here is our last question from this chapter. Mutualism. Both interacting species are benefited. Commensalism, positive, neutral. Parasitism, positive, negative. Competition, negative, negative. Predation, positive, negative. Mutualism example, orchid, Ophrys and bee, so 5. Commensalism, epiphyte growing on a mango branch. Parasitism, sparrow, wait, that would be predation. Parasite, ticks on dogs, that is ectoparasite. Competition, Balanus and Cathamylus. Predation, sparrow and seed. 5, 4, 1, 2, 3. What? There is no option like that. The options are wrong. 5, 4, 1, 2, 3 is the correct answer. There's this one option that has 2 5s. There's a mistake. Okay. Just randomly, I'm selecting as C. Rahul Raj Vinayak, I don't know why you marked as C, but yeah. Let's take a look at our leaderboard. Ajay is still on top. Then we have Sakshi second, Preeti third, Fatima fourth, MR fifth, Vimala, Hritika, Divya, Solo, Anam, top 10. Sure, take your grace marks, Bhuvna. Let's move on to our next chapter, ecosystem. Here is our first question from this chapter. The consumers in a pond are represented by. The consumers in a pond are represented by zooplankton, free swimming forms, bottom dwelling forms, all of these. Please read the question carefully, read all the options before you answer. Producers are your phytoplanktons and other plants like the submerged plants and the marginal plants, right? Consumers, zooplankton, floating forms, free swimming forms like fishes, they are also consumers, bottom dwelling forms are also consumers. So all of these, option D is the right answer over here. Selvi, Wajid, Priya, top 3. Next question. What happened? Okay. Secondary productivity is defined as the rate of formation of new organic matter by producers, plants, consumers, none of these. Secondary is always consumers. If it's by producers, which are plants, you don't call it secondary productivity, you call it primary productivity. Correct answer to this question here is option C. Jesus saves. Divya, Selvi, top 3. Okay, we just answered this one. Okay, let's answer this one. The amount of biomass or organic matter produced per unit area over a time period by plants during photosynthesis is called secondary productivity, primary production, dry mass, and respiration. When they talk about the rate of formation of biomass, it is productivity. If they are talking about how much quantity or amount is produced, it is production. For as long as it's about plant synthesizing organic matter, it is primary. Okay. So here it is plants, how much biomass is being synthesized. So it's primary production. Option B is the right answer. Shrivardhan, Raj, Selvi, top 3. Here is our next question. In an ecosystem, the rate of production of organic matter during photosynthesis minus respiratory losses is called. If you minus the respiratory loss, what do you call it? All the biomass produced, total, the rate of that is gross primary productivity, minus respiratory loss, whatever remains for the consumption of consumers or herbivores, is what we call net primary productivity. Option C here is the right answer. Selvi, Jesus, Raj, top 3. Let's move on and take a look at our next question. I wonder why same question is repeated 150 times. Okay. I don't know where the other slides are related to decomposition. It has gotten deleted or something has happened. I'm not able to find it. Many questions are repeated. Okay. So let's quickly summarize what decomposition is and about ecological pyramids. For some reason, the questions are deleted from my PPT. I don't know how. Decomposition is a process where organic matter is broken down into inorganic substances, and the raw material for decomposition is detritus. Detritus is the dead matter of living organisms like plants and animals. There are 5 steps in decomposition. First is fragmentation. The detritus is broken down into fragments by organisms like earthworms, which we call the detrivores or detritivores. Then we have leaching. Whatever water soluble substances are there, they will dissolve in water and leach down the soil and percolate down the soil, and it will get precipitated as salts which are not available for plants. They cannot use it. Then we have catabolism, where microbes like bacteria and fungi will aerobically act on the detritus and break it down further. Then we have humification, where an amorphous dark colored substance gets formed, which is resistant to microbial action and colloidal in nature. That acts like a raw material. That acts like a reservoir of nutrients, so that humus is very difficult to break down. Finally, you have mineralization, where microbes again break down the humus and release inorganic nutrients. So this is the whole process. Decomposition, there are many factors that affect the rate of decomposition. It can be the nature of detritus. Whenever it has substances or molecules or compounds like sugars and nitrogen containing compounds, it happens faster. If it has lignin and chitin, it is slow. Whenever the temperature is warm, it happens faster. When it's too hot or too cold, it is slow. Whenever there is sufficient oxygen, it will happen fast. When there's less oxygen, it will slow down. Then what else? Yeah, just about that. Moisture also is important. It has to be moist. Then we have ecological pyramids, and ecological pyramids are of three different types. You have pyramid of numbers, pyramid of biomass, and pyramid of energy. It's important that you know what are the different shapes. It can either be inverted, it can be upright, or it can be urn-shaped. Whenever you take pyramid of numbers, generally in most cases it will be upright. In all of these ecological pyramids, at the base we always consider producers by convention. Right on top of it you will have primary consumer, secondary consumer, tertiary consumer. Decomposers are not included in the ecological pyramids. And yeah, pyramid of number in a grassland is upright. If you consider a tree itself as an ecosystem, then the pyramid can be urn-shaped. Talking about pyramid of biomass, in a pyramid of biomass in a forest will be upright, but in a sea it could be inverted. Then pyramid of energy is always, regardless of which ecosystem, it will be upright because of the 10% law of energy transfer. So remember all of that. And about food chains, there's two different types, detritus food chain and grazing food chain. Grazing food chain is the major conduit in aquatic ecosystems. On terrestrial ecosystems, it's mostly detritus food chain. Okay. Correct. Selvi has answered your question correctly, Wajid. Now let's move on to our next chapter because I have no more questions in this. For some reason it has gotten deleted. Oh wait, okay. I don't know why one question is repeated so many times. Okay, let's answer questions from biodiversity and conservation now. Here is our first question. According to current records, most species-rich taxonomic group among vertebrates is. I want you to read the question carefully and answer. A lot of you are typing A in the comments. I said vertebrates. Insects are not vertebrates, right? Fishes are the most species-rich taxonomic group among vertebrates. Among invertebrates, it is insects. So option B is the correct answer here. Apurva, Devdas, Chaitra, top 3. Next question. In India, we find mangoes with different flavors, colors, fiber content, sugar content, and even shelf life. The large variation is on account of. You know how all of them belong to the same species? Within the species, same species, we're finding variation. What is the reason for that? Is that species diversity, genetic diversity, or ecosystem diversity? The correct answer is genetic diversity because we have different kinds of genes. They differ. There is diversity there. So option B is the right answer. Selvi, Srivardhan, Jesus, top 3. Here is our next question. It is an assertion and reason type of question. The assertion statement reads, India has a greater ecosystem diversity than a Scandinavian country like Norway. What is the reason? At ecosystem level, India has deserts, rainforests, mangroves, coral reefs, wetlands, estuaries, alpine meadows, while Norway doesn't have such diversity. Over here, both the assertion and reason statements are true, and the reason correctly explains the assertion, so option A is the correct answer. Right? Jesus, Divya, Chaitra, top 3. Next question. This is regarding sacred groves. Examples that are given in NCERT. In Rajasthan, we find Aravalli Hills. In Karnataka, we have Western Ghats. Madhya Pradesh, Sarguja, Chanda and Bastar regions. Meghalaya, Khasi and Jaintia Hills. So 3142. 3142. Option C is the correct answer. Like most of you are saying. Good job. Ayan, Amy, Srivardhan, top 3. Here is our next question. Match list 1 with list 2 to find out the correct option. Evil quartet. It is the 4 major reasons why biodiversity loss or extinction happens. Rivet-Popper hypothesis was proposed by Paul Ehrlich to explain the importance of species richness in the maintenance and stability of an ecosystem. David Tillman's experiments helped us understand the relationship between species diversity and productivity. Alexander von Humboldt, species area relationship, right? So 3142. 3142. Option D is the correct answer. Vajid Varsha, top 2. Let's take a look at our next match the following type of question. Lungs of the planet. We call Amazon rainforest the lungs of the planet. Endemic species are species that are found in one geographical area and not found anywhere else on the planet. Biodiversity hotspots. They are very species rich and they have a lot of endemic species. Keystone species are species that drive major functions in an ecosystem. So the correct answer is 2143. Option B is the right answer. Jesus Selvi Maharaj, top 3. Narrowly utilitarian because we get many products from nature that are of commercial importance. Bioprospecting. Wait. This is 2. Bioprospecting is exploring diversity that could be of commercial importance. Broadly utilitarian is services like pollination, which are priceless, which are free of cost. So 2, 3, 1. Option B is the right answer. Yes, most of you have answered correctly. This is based on why we should conserve biodiversity. Ammu, Selvi, Rohit, top 3. We are almost done. We have very few questions left. This is an assertion and reason type of question. The assertion statement reads, a forest in a tropical region like Ecuador has up to 10 times as many species of vascular plants as a forest of equal area in a temperate region like the Midwest of USA. Reason, in general, species diversity decreases as we move away from the equator to the poles. If you look at this, both statements, assertion and reason are true. Why is the tropical forest in Ecuador more species rich compared to Midwest of USA? Because that is closer to the equator compared to the Midwest of USA. You know about latitudinal gradients and how the species richness is highest at the equator, and as you move from the equator towards the poles, the species richness is going to progressively decrease, right? Why is reason false? Species diversity decreases as we move away from the equator towards the poles. It is true, right? So correct answer is option A. Both assertion and reason are true and reason correctly explains the assertion. Option A is the right answer. Maharaj Tritirtha Chaitra, top 3. Next question. The protection of hotspots could reduce the ongoing mass extinctions by about. If we protect biodiversity hotspots, we can reduce mass extinction by 30%, 2%, 13%, 3%. This is a statement right out of NCERT. The correct answer to this question is option A, 30%. Vedarth Selvi Tritirtha, top 3. This is our last question for today. Answer this. It was held. The Earth Summit was held in Rio de Janeiro in 1992. So option A here is the correct answer. Hamid, Vedarth, Selvi, top 3. With that, we've come to the end of the session. Let's take a look at our leaderboard. Ajay is still on top of our list. Then we have Sakshi and Preeti and Fatima and Physics, MR, Vimala, Divya, Hritika, Solo, Anam, Amy, Raj, Dhanish, Nafisa, and the rest of you. Some of you joined late, but yeah. All right. Tomorrow you will have, I think, some other subject, chemistry or physics. Okay. And what time it will be scheduled and all of that you will get to know under the live section. So you can come prepared accordingly. There is very few days left. Stay in a positive mindset. Don't doubt yourself. Don't worry too much and overthink. Okay. Whatever is your effort so far, proportionate to that you will get the results. So if you overthink anything, you can't change much in 4 more days. Okay. So all the very best. Stay positive. You worked hard and good will happen. Good things will happen. Okay. I hope that all of you will get to achieve your dreams. I will see you again. I don't know when. So take care of yourselves. All the best. Bye bye.